Yes. So it will be starting by 15 minutes late, right?
No, no, I think we can start now, right?
Okay.
So, so it's Catherine and Seema who are in session in charge. Okay, it's all yours.
Yes, sir. I'm waiting for the chair. मेरे हाफ चेयर मिलेगा ना सर मैं उधर बैठूंगी चेयर बैठूंगी पूरी चेयर पे Good morning, everyone. Thank you for joining us for next session. So there is a river somewhere. Somebody needs to mute it. Okay. Uh, so welcome, Dr. Dharani. To introduce you to the group, he is currently the group head of Marine Microbiology Group at National Institute of Ocean Technology. He is also an open water scuba diving instructor and first aid instructor. So, a very true blue marine biologist with over 30 years of experience in the area. So, he will be talking on biodiversity and prospects of seaweed cultivation in the Andaman and Nicobar Islands. So, sir, the platform is all yours. Please share your screen now. And you will be having 20 minutes for the talk, and we want to keep a few minutes for discussion. So, floor is all yours, sir.
Thank you, ma'am. 1 minute. Before starting, can I have a check? My slides are visible to you full screen?
Please make
Hello,
it full screen.
hello.
Yes, sir. Hello.
It is not appearing full screen. Sorry,
So it's not full screen.
it is moving. One second. I will just unshare it and again do again.
Okay. So the option on sharing, share
Yeah,
your
yeah.
entire screen and go to
Yes.
the slideshow.
So I
Yes.
hope you have my full screen,
Yes, sir. We are at full screen now.
and it's yeah. I can see it moving also. Shall I start, man?
Go ahead.
Am I clear and loud?
Yes, sir. Yes, sir. You're visible.
Yes. A very good morning. Namaskaram. Um, Respected Director ILS and the organizer of this wonderful conference. I'm extremely thankful for the Director ILS as well as the organizer of this conference for inviting me. My sincere apologies, I could not make out today because of we are having a tight schedule. We ran to some other station and came back Monday, and so could not able to make out of the frequent travel and other things. But however, it is my great pleasure to share our research work, what we are doing in Andaman Nicobar Islands. Um, Of course, as the introducer said, I'm Dharani. I'm working for this Marine Biotechnology Division of National Institute of Ocean Technology. We are having a field center at Port Blair at the presently Sri Vijayapuram, and we have also field center at Andhra Pradesh, and we also do have a working station at Lakshadweep. With a small note, I would like to start. Of course, a lot of speakers could have spoken earlier about the importance of seaweed. I don't need to re-emphasize on this, but still, I like to say that the seaweed market is picking up so drastically and so fast, and it is going to be a huge economy, blue economy, in terms of the present days of blue economy concept. This is going to be one of the boosting aspect in the blue economy concept, and that is definitely a growth. And we are having an severe impedance of expanding this to cope up the blue economy requirement. That is what I want to emphasize this. And of course, you must be knowing the marine algae adapted to survive in marine surrounding habitats, some multicellular, and it has a different kind, and generally divided into chlorophyceae, phaeophyceae, rhodophyceae, which has essentially producing three major compounds of agar, algine, and carrageenan, which pitch the market and which drive the market of this blue economy program. They produce in various ways, and there are very fascinating organisms on this earth, which we cannot contemplate with any contemporary plants. Though it is not a true plant, but it is a wonderful, marvelous magic plants which can do wonder in coming future. So, about the seaweed diversity, it was general. I'm not into seaweed taxonomy, but based on the literature available, they say that approximately 11,000 taxa of seaweed reported worldwide. And in terms of marine environment in India, about roughly 5,000 and odd kilometer length, with it's recorded has 865 taxa of seaweeds, out of which India also has a very good margin in this. India has about 7.5% of taxa on this, and these are some of the distributions of the taxa state-wise. Out of this, you would have seen this very clearly. The Gujarat coast, particularly Maharashtra and Gujarat coast, and Tamil Nadu coast, have the huge diversity. And coming to the next is the Andaman Nicobar Islands. So these are the major biodiversity reserves of the seaweeds, and which has very potential immediately to blow up the seaweeds into a very larger expanse. So what we do in the Andaman Nicobar, being a station at Andaman Nicobar is consisting of 336 islands and about 1,962 kilometers of coastline and about a geographic area of 8,200 square kilometer encompassing 0.6 million square kilometer of our EZ, has documented so far 244 different taxa belonging to 100 genera. This includes BSI as well as all the reports inclusively, and which has of course the Phaeophyceae is a very large group which has 587 species, followed by Rhodophyceae of 107 species and Chlorophyceae of 79 species. So this is the richest diversity of the Andaman Nicobar Island. And this is our center called Atal Center for Ocean Science and Technology of National Institute of Ocean Technology in currently the Sri Vijayapuram, named as previously known as Port Blair. And what we do there is, first we are monitoring the sea areas. We have another program called Coastal Sea Water Quality Monitoring, and we also do an continuous resource management survey. We have completely digitize this Andaman Nicobar maps, and since we have a huge number of huge data sets on the coastal water quality and the bathymetry of these islands, we have generated and mapped to identify the suitability of the depths, suitability of the seaweed cultivation. That means to identify a potential area of the seaweed cultivation in terms of depth, sea surface current, and mean wave height, TSS, temperature, salinity, DO, chlorophylls, chlorophyll, and other nutrients. These are some of the first six parameters is very important in terms of FAO protocol to have an economic culture. So we have identified the total area of the seaweed cultivation in Andaman and Nicobar Islands across and from North Andaman to the South Andaman, and this is the quantity of areas, one area available, and we have divided into three major areas: least suitable, suitable, and highly suitable areas based on the major set parameters earlier. And these data sets are shared with the Ministry of Fisheries and other institutes for the use for the. And also we simultaneously recorded, we are closely working with BSI, Botanical Survey of India in Andaman, and of course the team of BSI and NIOT jointly do surveys in this entire island. And we have recorded an occurrence of out of 244 taxa, we could able to record 110 taxa out of this. And we have, these are the standing biomass in each islands, whatever the stations that we have done. And these are some of the locations what currently being continuously monitored on every quarterly basis. So these are the stations covering from North, Middle, and South Andamans, which has a continuous monitoring program. And these are some of the representations of the different species. Different species of chlorophytes in orders. You have see it was really marvelous having a large diverse set data. I could not able to present all the pictures because there are a huge number of species available, and this is a glimpse of the chlorophycean, and this is of Phaeophyces, and this is about the Rhodophyceae of red algae. And out of this selection, we continuously do the biodiversity assessment and the standing crop. And by this thing, we bring the species to the Port Blair for further cultivation. So for this, initially, based on the based on the availability of the seaweeds, the growth rate, and based on the market requirement, we selected at least 5 to 6 species of Gracilaria from the Andaman Nicobar Island. You must be knowing there was a controversy of discussion in Andaman to whether introduce a foreign species like Apophagus. They still say they consider as a foreign species to introduce in Andaman and Lakshadweep. That is because of that, to have the things before we complete the debate with the fisheries as well as with the forest and come to a common conclusion to bring other species, we initiated with the indigenous species, which is in Gracilaria. And we have identified number of species Gracilaria inside an Andaman native species, and also we have transported Gracilaria species from mainland like Gracilaria debilis and Gracilaria filiformis and Saponema, all those species we have transported from mainland to simultaneously study the growth rate of these strains in the coastal waters of in the deep sea waters of Andaman. And this is one of our test site in Chiriatapu, where we do different kind of cultivation system, starting from a simple bamboo raft to a long line, from the shore to a 20 to 30 meter depth. And this are the different types of cultivation systems what we have attempted since from the beginning, from the simple bamboo raft to a PVC long line, to a HDP long line, to a full-fledged long line without any rafts, with a grid mooring system. And also we have tried in a 20 meter depth, and we have various degrees of success of growth rates. And what is the aim of the study is to have an 1 hectare of study, 1 hectare of seaweed farming studies throughout the year to see the annual productivity and see what are the troubles and problems that we are having inside the in the culture system, as well as what is the annual productivity. So this is what some of the experiments what we do every month that we have for example, from January to February, what kind of growth rates what we achieved. And essentially I want to share what kind of the problem that we are achieving. We will come to the production of the species. Every month has some problem of environmental vagaries, including cyclones, weather conditions, and the animal infestations. There are some organisms which I particularly want to say in January to February, there is a big source of grazers, which like nudibranchs, different groups of nudibranchs comes inside the seaweed cultivation system, which has a large impact on the cultivations. And similarly, when it says from May to June, it has a luxuriant growth, but still has some growth and remnant of epiphytes and sedimentations are picking up in this region in this duration. And again, from July to September, the growth is very good. There are no much epiphytes or other things, but due to the wind and wave conditions, there are the time the organisms grows well and luxuriantly grows into big bunches of maybe 5 to 6 folds time. And this time what tends to have a breaking of the seaweeds, which get washed ashore, or which we lose the product. And during this October to November also, the growth rate is excellent. Of course, there are epiphytes like juveniles of crabs and other things, which is not of great order of destruction in this seaweed species. And again, coming to January, February, again, the monsoon starts at the time. And again, epiphytes like hypnea and other things, decapod starts, and holothurians also settle down, because you must be knowing seaweeds is one of the very potential site for this holothurian breeding. And this is how during the rainy season the stage starts from the day 2 to days 30 to day 65, how the seaweeds grows. Yes, we have, you can see very visibly on the 45th day how much the full raft. From the first day to the 45th day, you can see the difference of the seaweeds, which completely occupy the rafts. And this is a general of a 6 cultivation cycle. This is a general growth rate of the seaweed what we have recorded, and we have recorded maximum of 6-fold increase. That means, which is 6-fold increase in significant times of the year, which means it is meeting the standard requirement of commercial growth. That means 5 to 6 times is considered as a standard doubling time which is required for a commercial cultivation. Yes, it is happening, but not for all the months. That is what the annual cycle of culture says from our experimental culture systems. And once this has been done, this all the parallel. There are some of the preliminary work what we do with Central Agriculture Research Institute in Andaman. We give the seaweeds and help them to extract all this product and to study the biostimulant property of various things. That is well known, but in Andaman themselves, in ICR's Agriculture Research Institute is doing this kind of analysis, and it is giving promising results there also for island group. And because one of the problem in island is there is less nutrition and acid sulfate soil, there is less nutrition in the land. Something of this kind of order will support the agriculture for Andaman Nicobar, because agriculture product is very less and Andaman is very interested. And this product also we attempted to do the extraction of bioplastics in an industrial way. This we attempted with 2 species of 2 species of seaweed, one from Gracilaria, another from the reference of the commercial Cappaphygus, and we able to make it in industrial scale. We had an industrial partner with us. We have an industrial partner who can do a commercial scale culture. We have attempted from the blending of the seaweeds. We replace the total starch of various concentration, and we started from the blending, then to extraction, to making pellet, and to blowing to a sheet with the seaweed component, and we could able to get a very huge quantities of seaweed based bioplastic. You can see on the right hand side, these are the plastic sheets, bags which has been extruded out of this, which is very much equivalent to any other bags. And also we did a very detailed the testing of this plastic extruded from the seaweed based polymer. We did the parameter analysis in both the Plastic Engineering Institutes and IIT Madras. It is giving more or less very close to the commercial available plastics. And the important things is, one of the things is it's fast degradable. It is degrading in a very fast manner. And second, also we try to attempt the production of lactic acid, which is one of the commercially important product, which we cannot extrude the plastic. This is one of the method. We attempted with the Gracilaria aquatica with various degree of success. We are producing the lactic acids in a small scale fermentations, and by trying to optimize the culture conditions, because the lactic acid production is maximum of say 0.3 or 0.06 weight by volume. And this has to be increased because we are using the MTC strains for this cultivation. But we are on the process of developing the own strain which is coming from marine, which may able to give more production on this lactic acids. And this is a reference species of alba which they have used for production of lactic acids, which also give somewhere 0.4 to 0.3 weight by volume of this lactic acid production. And we have also completely optimized the lactic acid production using the kappa figures, using the MTC strain with a multi-parametric analysis. And we have a complete data set of this production, and we are confident that which can go up to scaling of the lactic acid production in industrial fermenter. And also this has been attempted for hydrothermal liquefaction with IIT Madras, and we have an data sets of which is in various combinations of hydrothermal liquefaction as well as different species. We have a data set which can be shared with our colleagues. And these are some of the problems what we saw earlier. I have shown you like in the January months we have a huge infestation of this nudibranch, which is like a pest. And also other challenges in seaweed cultivation includes the accessible to the cultivation sites. Most of the generally the Andaman Nicobar, the eastern side is only habitated. The western side is not habitated. Sorry, the western side is only western side is not habitated, and only we had to use the eastern side. And there are a lot of difficulties in accessing the site, and a lot of reserve areas, and the non-availability of the seaweeds. I mean the commercially important seaweeds in the wild, grazing of herbivorous disease, various physical parameters, and undesirable growth of marine organisms, and also the roughness of the sea. And the bottleneck, what we have felt, this is the wild seed commercial preferred species required in a large quantity must be made available in Andaman. And there are no industry or seaweed processing industries, with no industry for cultivation or no industry for seaweed processing working. And there is no local demand for consumption or any use of seaweeds until we produce and usage. And there is no connectivity with the neighboring countries, Andaman. Of course, recently there are some connectivity started for, if you see in terms of export from Andaman, either it has to only brought back to mainland Chennai, mainland India, or now there is some connectivity to Malaysia, but we do not know how it is going to work. And there is very poor local awareness of the seaweed and cultivation, any kind of cultivation in Andaman. And currently also there is still the seaweed policies has not been framed, but government is working slowly on these aspects. And these are some of the recommendations. We require high quality, the Andaman require high quality seedlings. Identification promotion of native species, one of the important, considering the Andaman biodiversity and the fisheries, as well as the Ministry of Forest norms and regulations. And identification and testing of all the feasible sites is very important, though we have done a test study, but practically every site has a unique problem that has been tested at least for a year so that we can do a large scale cultivation. And establishment of a small scale seaweed industry in Andaman is very, very essential, at least to make the raw material, I mean, at least finish the raw material, semi-finish the raw material, so that we can separate into value added and non-value added and transfer, because the transferring of the Andaman from any biomass from Andaman Andaman to mainland is going to be a costly method because of the either ship or air cargo transportation. And the transfer of technology for development of livelihood opportunities, one of the important things required. And there is also a small window of non-cultivable time, that also we had to have a seed bank to hold this. And the eco-friendly approach has to be concerned, and development of other infrastructures is very much required. And of course, we are also working tightly with Fisheries Department. We have a MOU with the Fisheries Department for testing and large-scale cultivation of the seaweed in 1 hectare farms in 1 hectare offshore cultivations. And this is our area where we do the test cultivation. And thank you very much for your time. I'll be happy to take any couple of questions.
The Dr. Dharani, am I audible?
Yeah, clear and loud, sir.
Yeah, I just wanted to focus on the two slides in the website. One is the bottlenecks of the seaweed economics, and the other one is the
Sir,
recommendation.
sorry,
And
sir, can you please repeat, sir? Your voice is very low, sir.
okay, am I audible now? Better?
Yes, sir. Clear, sir.
Yeah, the bottlenecks, the challenges that you showed, and the recommendations by NIOT.
Yes, sir.
So one of the places you said there is a lack of demand of seaweed
In Port Blair, sir.
in Port Blair. And do you think
In Andaman now.
yeah, the export market is not looking for seaweeds, or do you think by that time the cost escalates and it is not
So
economically viable?
export market is again all these things done from the mainland, particularly from Chennai, from Madurai. So the transportation is there. Someone has to set up the facility there and try to export directly from Port Blair to other Southeast Asian countries or wherever it want to be done, because the cost involved by ship or air is very huge.
And the other point was that identification of particular seedlings for the farmers.
Yes, sir.
Do you think help of high technology such as sequencing or barcoding or markers would help, and where scientists can play a role there, or it is already been done?
Sir, we are there are only few scientific institutes in Andaman. Though you have a lot of institutes, but science-wise, only National Institute of Ocean Technology or otherwise ICR Agriculture and Research Institute, and is there, and Ministry of Forest is there, but it is majorly with the conservation preservation aspect. So we are doing primarily this identification and barcoding, but still we would like to have collaborate with DBT partners like ILS and other institutes to have a very effective and large scale works.
This is what precisely I wanted to hear from you. Thank you.
Yes. Of course, we are all the technical partners now. We will have collaborations in every area that what we are working.
Hello, sir. I have a question.
I'm sorry, sir. Again, the voice has gone down.
Hello, sir. There is a. I am Shubhabrata from ILS. I have a two question. Like you have monitored the Andaman and related to all seaweeds, but have you found anything like which is commonly grown in any area of Andaman? Means which one? If we ask you give us seaweed which is good for Andaman, then which one? Because it's very specific to location wise. But have you tested any seaweed which is overall can be grown in Andaman? So we can work on that and try to improve more of that.
Sarah, we have a very detailed data of standing biomass of seaweeds diversity and standing biomass in middle, north, and south Andaman, middle and south Andaman up to port locations, and there is uh, generally there is a large population of seaweeds which belong to the green algae are large, I believe. I will just check the data. But we have not tested in large number of locations. One of the fast we have selected waste area because waste area is the standing biomass is bit large, and this was one of the potential species what has some industrial value. So this species we thought we will take into a large scale cultivation which can grow in the deep water cultivation to test whether this can form an potential species for uh, to bring into an industrial product. So here also we require seaweed industries seaweed industry as well as other institutes like us to have a formulation, because the seaweed Gristleria edilis now only coming up in the market, people are buying, but still there is a full process package is not available to make this seaweed as an economically viable model. Except except this Gristleria edilis area of Edilis and other 3 to 4 species of Gracilaria, and particular to the Chidiatop or North Bay, we do not have data on much other aspects, other islands. So if we want to do in North Andaman and Mayaband and other area, we had to do the test sites there. Initially, CSMC had tried in but there was an Coprophagus species, so the experiment was cut short rapidly, and because of the environmental problem and other things, it has been done, tested, but could not be continued for a long time.
Okay. And have you able to identify any physical marker like appearance related to carrageenans or related to agar quantity? Is there anything like which are in terrestrial plants, something we have a physical marker you can see
We
and
have, we have a couple of strains which is very close to Coprophagus species,
okay.
and which yields carrageenan, but the growth rates are very poor, and it is not growing. We are now on the test site, so we are yet to publish those results on the things. We have set up the test, but still data is not curated.
Okay. Thank you, sir.
So we can take questions from online participants also, if there are any questions.
Yes, I had a quick question, Dr. Dharani.
And Namaskaram, Dr.
Good morning. Good morning. I saw your fermentation of seaweeds to lactic acid.
Yes.
I just wanted to know, are you breaking down the polysaccharide into individual sugars before fermenting to lactic acid?
That is a very established, conventional process of breaking down to sugars by various alkali and chemical treatment. Then
Okay.
only going for fermentation, because you know that, and that is a lot of things to optimize to bring down the product into the sugars, and use of less, reducing the harsh chemicals and the quantity of chemicals is also one of the important topic to be addressed.
Okay. Thank you. Thank you, sir.
Yeah, we are trying. We are trying with right from bacterial to very common chemical alkalis to break and clean it.
So, Dr. Dharani, I just have an extended question to Narayanan. So, I know that the breakdown product will have mixture of sugar. It will be galactose as well as sulfated galactose as well as anhydrogalactose. So,
Yes.
do you know whether all of them are being taken up for the lactic acid fermentation, or there are only few of these, or just galactose or some of the glucose are being taken up? Just curious.
Sir, generally now we are concentrating on a couple of species which has majorly carrageenan, and the chief product is of galactose from this carrageenan, I believe, and we are yet to see the results, or I have not presented the results of the different usage of different combination of sugars in this type strain which got from MTCC. So again, it is on the type strain which is going to use, which has a specific preference on the specific sugar moieties. So we have 2 type strains from the MTCC culture collection. So we also try to screen more number of bacteria from the marine source itself, which can use a variety of sugars.
Good morning, sir. Good morning, sir. I am Bhu Prasad from ILS. Sir, my question is, how can we store the indigenous species of fluid? Is cryopreservation is a good idea?
Sorry, I couldn't hear you. Can you please repeat again, sir?
Yes, sir. Sir, my question is, how can we store the indigenous species of seaweeds? Is cryopreservation is a good idea?
Sir, I don't think seaweed can be cryopreserved because of the fragile cellular nature and high water content and the glucose and sugars content, a huge amount of polysaccharides, and we have not attempted, and I also not seen much publication on this. There may be better experts that need to based on this preservation, but in generally we have done preservations of various preservations for bacteria and from fish eggs we used to do, but I don't think this will be feasible to do cryopreserve because of There's a different kind of sugar content, but it has to be attempted.
Thank you, sir.
Thank you, Dr. Narendra, for that question.
Yes, Namaskar.
I'm Karthikeya from DBT. So my question is, how big is your germplasm collection, and what are the challenges you foresee in scaling up the germplasm collection?
Sir, this is a very common question, and of course, everyone is facing. Uh, we also do not have a big land-based infrastructure. Of course, we are doing a very fish rearing facility in approximately budget of 20 crore. And it may be another 15 months we'll have that. But generally, we are preserving all the seaweed species in the sea itself, and the set of culture is being cultivated on the surface. And during the rough weather season, we are having a small cages of metal cages like a chicken box. You must be able to see those cages has been, the seaweeds has been packed and lowered in the sea 10-meter depth of 5 to 6-meter depth, and that has been held for to tie over the rough weather seasons. And we are currently holding, say, some 5 to 6 species of this Gracilleria and couple of other species what we have bought from other locations, and we have a very handful of species. So we do not have a big germplasm or anything like that.
Thank you, sir.
Good morning, sir.
Namaskaram, sir.
I am Rajit from ILS. Sir, I have a question that by which mechanisms do you stimulate the plant growth?
Sorry. Again, I repeat. There is some voice.
Sir, by which mechanism seaweed generally stimulates plant growth? Is there any mechanism?
Seaweed generate plant growth. Of course, still I'm also not an expert on this. There is a lot of experts sitting in front of you. Generally, seaweeds extract, which has huge amount of sugars and has lot amount of plant promoters like gibberellic acid and other things are available in the seaweed. We have a signature of those things in the HPLC analysis. And this, there is a large amount of phytochemistry people are doing, which use surface foliage kind of things are being used, and that is fully a cup of agriculture people. I am not an expertise in this. We are working closely with and of course Dr. Sri and other people who are ex-whale salvers who are working on the seaweed, seaweed aerial fertilizers will able to answer the question properly. I believe we are mostly concentrating on identifying a species, identifying the green environmental settings, and to set up the large-scale cultivation systems to feed feed the industry, because there is a front and back, everything is available. The seaweed demand is there, the process is there, and the product is there. Only we want the source, and we want the source to get large scale cultivation. That is what we are trying to do to expand the seaweed in offshore culture conditions.
Thank you, sir.
Thank you, Dr. Dharani, for this very. Okay, one more question we have. Last question. Hope you don't mind.
Please.
Hello, sir. I'm Samiksha from ILS. So my question to you is, how did you identify the seaweeds which will be helpful for making bioplastic, and what are the characteristics features to make bioplastics?
Of course, you must be knowing that seaweed harbors, as we said, 3 types of seaweed, from green, brown, and red. In terms of color and in terms of product, it has carrageen, algin, and agar. All these 3 compounds are essentially an biopolymers. Any one of this can be used as a biopolymer. It depends on what is the what is now on what quantity that in what quality that we can produce, and what area you can use it application. So you can use agar to produce an bioplastic because it is also an polysaccharide. You can use carrageenan, you can use algae. All these things are polysaccharide, and definitely in the bioplastic production we can substitute this. But what is the cost? Where we are going? If you are able to sell your bioplastic of very high order, you can use even agar. Currently, we are pitching carrageenan because that is one of the less value product then compared to the agar. So we think if we produce carrageenan surplus, which is not coming to a direct food like agar and which is not having a big, the cost is not so high as agar. Carrageenan can be one of the molecule once we produce a large quantity, but still we have for pharmaceutical and other lot of other industrial supplement, carrageenan is a base material. So we want to go from a cheapest material, cheapest this one to use as an biopolymer. So that is the crux. It is a known fact, nothing, a great secret inside this. And of course, bioplastic, if you say the lactic acid is one of the ultimate, it can be done by the biopolymers. But again, the trade-off is how much lactic acid we produce per unit area, per unit quantity of the stuff that we bring in. So that once you strike and balance, you can do this by the both approaches. One, you use and base as a total polymer to use and to produce in bioplastic, or convert completely into lactic acid entirely, then use as base material of bioplastics is going to be for the application and for the cost and the demand.
Okay, sir. Thank you.
Now, thank you very much, Dhani, for joining online and giving your very valuable thoughts on this. I'm sure it was very enlightening for the students and for all of us also. I hope you'll stay online with us for the rest of the day. Thank you very much.
Definitely, it will be a pleasure to watch the proceedings and share in the proceedings. Thank you very much. I once again thanks Director of ILS, Dr. Sri sir, for inviting me to meet. Thank you very much, sir. I'm very sorry I couldn't able to make up in person. I will come back. Will be due, and I'm very glad to work in tight with ILS and Dr. Sri in various aspects of this. And I require everyone of the elite audience sitting there to support, to promote Andaman Islands in terms of seaweed cultivation. Seaweed cultivation, of course, we will able to do these things, but for a product development and for marketing and so many things, we require a huge handful of people, handful of scientists to promote this inside the island itself. So that is a great demand we have in front of us. Thank you, sir. Namaskaram, sir.
Thank you, Dr. Dharani. So the second speaker for today is Professor Valsama Joseph from Cochin University, and now she is the head of the Cochin University of Science and Technology, the National Center for Aquatic Animal and Health. And she has to her credit many academic and administrative qualities. And her main research interest has been omics for understanding marine bioresources. And today she will be talking to us on nitrogen fixing bacteria associated with the tropical seaweeds. Professor Valsama.
Thank you very much. A warm good morning to each one of you. So I thank Dr. Debashish Das, Director ILS, and the ILS family for this opportunity. And I am also thankful to Sri Kumar Sajunarayanan and Dr. R.A. Narayanan from C6 Energy for introducing me to this event. So today I will be speaking on nitrogen fixing bacteria associated with the tropical seaweeds. As a student of marine biotechnology and marine algae, I have been looking more into what seaweeds are and what they require. So what are our experience that we will be sharing? As we all know that furthering knowledge of seaweed growth and development is required for sustainable production, because basic research is the key. We don't know much about what seaweeds require. They are growing in the intertidal areas, and they are growing in very harsh conditions where always this wave action is there. Their reproduction is happening there in these tough seas. And what are the growth factors required to facilitate the various life cycle stages? This is a very challenging question for both biologists as well as biotechnologists. So seaweed microbiome is very important. because mostly people look at bacteria as pathogens, but just like in any other case, like humans or any other animals, fish, cultivated aquaculture species, we know there is a beneficial microbiome. And that is true the case with seaweeds also. So preventing seaweed from going from a healthy microbiome to dysbiosis is the key. And they are very important. Seaweed microbiome is very important in providing pathogen resistance and also making the nutrients and vitamins available and providing nitrogen to this. And also they are very important in there are seaweed beneficial microorganisms which we need to identify, which is very specific to each different kind of species and different family of seaweeds. So there are a lot of biotechnological applications. I need not expand too much on this because everybody sitting here know they're all working on bioprospecting of seaweeds. Even microorganisms associated with seaweeds also have various biotechnological applications, functional foods, bioremediation, plant growth promoters, medicinal compounds, fertilizers. They too have a lot of actions. So it is very important to study their distribution and what is their role, whether it is parasitism, mutualism, endosymbiotics, some or and we have to isolate them, study, characterize them, and apply them for various field applications. And we know oceans are oligotrophic; they are limited in nutrients. The moment nitrogen, phosphorus, iron, all these are released, it is immediately taken up by the microorganisms. So oceans are always oligotrophic and limited. So mostly seaweeds depend on nitrate, ammonia, or urea at times, and there are intensive studies on going on the nutritional physiology of seaweeds. But a recent study shows that apart from, we are very much familiar with the marine nitrogen fixation, especially the cyanobacteria are very much important, reported in marine nitrogen fixation and in nitrogen biogeochemical cycle. Now people are trying to close, but we are finding that recent studies show that there are new players which are involved in marine nitrogen fixation. And the nitrogen is fixed nitrogen is very important because nitrogenase is the key enzyme which is very oxygen sensitive. So they are microaerophilic organisms, and this nitrogen is first converted into ammonia and finally to glutamine, so which is used by the cells. So this work which we initiated with conceptualized along with during a informal discussion with C6 Energy, having Sri Kumar Suryanarayan sir and Ms. Shailaja Nori and Professor I.S. Price sitting at our lab. Then we were thinking that these oligotrophic oceans, this Kappa ficus is growing vigorously. So then they must be harboring nitrogen fixing bacteria. So nitrogen fixing bacteria is isolated in nitrogen free seawater based mineral medium, because anytime there is any organic constituents like a beef extract or peptone, then the heterotrophic bacteria will grow. So we have grown them and we have isolated several bacteria and characterized them for NIFH gene. Finally, we have screened them for ammonia production. That is because of the pH change, there will be ammonia production in those cultures which are producing this. And we have tried various approaches: immersing clean, sterilized tissue in the medium, extract, inoculating serial dilution and extraction. And finally, we could have very distinct colonies, and many of them are novel species as well as reported species like Pseudomonas tatseri, which is already known in in other rice and other plants reported. So we have characterized them, and this is this is the first culture because they are microaerophilic. We have to stab them into the culture, and once they develop, again we are streaking. But we have to study a lot about these organisms because we should not be biased by thinking that they are very sensitive to oxygen. So only some of the, even though all of these strains were producing ammonia, only some of them were having NIFH gene, and we are not sure whether we miss the NIFH gene while amplification. So this also we have to conclude, and we have sequenced some of these organisms, and it is found that they are producing indole acetic acid and phosphate solubilizing. They have this plant growth promoting properties. And we could see also that we have, since it is isolated from Kappa ficus, we have provided carrageenan, a different concentration. We did different permutations and combinations, and some of these producers are very well. In fact, these nitrogenase activities are part of an ongoing work. So we can see that in the presence of this carrageenan, this nitrogenase activity is very much in the potential isolate. So we are planning further studies with validation with the C6 energy for how we can use in the field conditions, or even we have to validate it under laboratory conditions. So this is our work on this. And another work under DBT marine bioresource work, we have been working on cell and developmental biology of seaweed, Sargassum whitey, identification of unique metabolites, transcriptome analysis during different growth stages, and isolation of endophytic organisms, and development of micropropagation protocols. So for the past 4 years, we have done so much of field sampling from Kovalam, and we tried, and we have gained a lot of experience. Not all the cases, even isolating nucleic acids from seaweed itself is a challenge. Isolating transcriptome itself is a challenge, because we are getting all these phenolic compounds or polysaccharides. So we have with several trials and errors, we have standardized these protocols. These are some of the images showing the reproductive structures of Sargassum whitey. You can see the air bladder, and the right is this receptacle on which these conceptacles or the reproductive cells are formed. So we have collected from Kovalam and cleaned the thallus, and we are bringing it in for different applications. For chemistry, we are bringing in 50% isopropyl alcohol, and for molecular analysis, we found that bringing in RNA later always degraded the sample. So we are bringing directly in liquid nitrogen, and wrapping this seaweed tissue in clean wet cloth, and then bringing in ice is the best way to storage, bring them to the lab. So we have tried with several experiments. Even with the Kappa ficus, we tried several combinations of maintaining them in the lab in nitrogen-free seawater and nitrogen-free mineral medium. Different combinations we tried. And all these trials shows that after some time, automatically it will degrade. And plant tissue culture based methods were not working. Now we are basing, just like Dr. Michael showed, we are trying to clean the seaweed thallus and recycle the water. So this way we are trying to maintain it. And from Sargassum also, we have tried all different microbial media. Even under dilution also we can try, because on the field, when we collect organisms, suddenly putting it into full strength media will be very toxic for them. So we have isolated, and this we are characterizing right now. But very unique observation that this, from the growth media of the tissue culture medium, there emerged several, after few months, there emerged various fungal and bacterial species. Many of them were colored, and we can see that except malic acid, there are no other organic components in this growth media. So we identified many of these unique species as Exigobacterium, Cinnamomicrobium, Paracoccus, and there is Pseudomonas tarseri also that is missing from here, that is very potential, and also very unique fungal species. And we are also doing metagenomic studies also to study the microbiome. So it is found that many of them are producing indole acetic acid. So this screening is right now going on, and there are nitrogen fixing also. So nitrogen fixation, the life in the ocean is a story of cooperation, because without symbiosis, life is not possible there. And recent reports are there that this from Sargassum birds, there is intense nitrogen fixation reported, mainly based on the epiphytic cyanobacteria and all. So the nitrogen fixation reported mainly in all these eukaryotes. There are studies also going on, all because of the symbiosis organisms. If you look at many of these land plants and sponges, or any of legumes, and all these take, different species of nitrogen fixing bacteria residing in various tissues. So we are yet to get a clear idea about how this cooperation is happening with respect to the bacteria we isolated. Much studies are required. So in plants this is there. So now recently we know that one nitrogen fixing organelle is identified, a nitroplast is identified in marine eukaryotic algae. Like that, we have to look intensely into this. So nitrogen microbiome manipulation in seaweed culture is a very potential area, and we need to study the seaweed diseases. We need to have the diagnostic techniques, because under farm conditions, under ocean conditions, we know nothing is under control. But to control the same, anything is there, we need to have very good understanding. So this microbiome manipulation in every stage is very important. So for that, understanding bacteria is a key aspect. So potential areas which is of interest to us: basic seaweed biology, karyotyping, even we are doing genome sequencing. And as we all know, microalgae are formed by endosymbiotic events, primary, secondary endosymbiotic events. They are very complex and distinct in genetic content because of the endosymbiotic gene transfer as well as the horizontal gene transfer. So it is very important: genetic diversity and development of genomic resources, understanding the developmental biology, micropropagation, strain improvement, host microbe interaction studies, and developing microbial formulations for growth and enhancement, and micropropagation of this. These are all potential areas. So we look forward to new horizons in aquatic animal health and marine biotechnology. And our motto of our institution is relentless effort for the health of aquatic animals and their environment. So I profusely thank Department of Biotechnology, Government of India, for all the funding. We have carried out almost 30-40 projects since we are celebrating 25 years. And Professor I.S. Pricing, founder of our center, and he's a well-known microbiologist, and Sri Kumar Suryanarayan and Shailaja Nori for having conceptualizing along with us on this work on nitrogen fixation. Dr. R. N. Narayanan, head Ocean Agri-Tech in C6 Energy, with him we are collaborating now. Dr. Sina, Dr. Varsha, Dr. Jayesh, and several project fellows, and our DBT PG students, Ms. Sweta, Ms. Aparna, Mr. Agash. All these were students who have been continuously working. They initiated this. Thank you. And if the chair permits, 1 second I'll take. We are working in in various aspects. We are running several education programs, especially related with the DBT PG program, Skill Weekend, Bioinformatics Center. And I would like to share that we have developed the first world's crustacean cell line over 15 years of DBT support, because nobody believed with the fusion cell line, and which is deposited now in NBFGR. We are working on various product development, and these are some of the probiotics, and this we have been transferring to aquaculture farmers. Around 1,000 to 1,500 farmers take our services yearly. So we are working in very close interaction with them. And we have also, this is also initiated with the DBT support nitrifying bioreactor, which is installed 100 unit for high density fish culture. This has been installed. Thank you.
Thank you, ma'am. Thank you so much. That was very, very informative. Since we are running behind, we have time for 2 questions maybe.
Thank you, Dr. Valsama. Very insightful talk. So we know about the application of seaweed extract as a biostimulant for agricultural crops. And you mentioned that there are several endophytes and epiphytes associated with seaweeds. So can a part of this activity, which we see in agriculture crops, the growth enhancement, be contributed by the nitrogen fixing bacteria that are present in the seaweed, and they are able to colonize the rhizosphere and promote plant growth once you put in the extract in the
yeah, that we are not sure, but major part of this extract will be from seaweed only. But when with respect to seaweed, what is known is that this gibberellic acid, indole acetic acid, all these are present in reported in this. But when we do the laboratory level experiments, when we have tried with all these extracts and all these several combinations, but nothing worked on seaweeds. Maybe these plant growth promoting factors are also there in the seaweeds. That might be the major reason. That is what I feel. But the role of these associated bacteria, we have to study, then only we will be able to say. Another is polyamines are thought to be very important in seaweed growth and reproduction. So it is thought to be produced by the associated bacteria, but in some seaweeds also have the spamine, spermidine, pruttacine. But we tried this also, but something is there which we all need to work together to crack, what is, why we can't grow them. See, because some people are asking about the germplasm maintenance, because Dr. Michael is maintaining, but I don't think that anywhere, in India, any lab, whether we are able to maintain, because now we have to develop that method, because after we bring from the lab, we maintain several trials. After some time, it is getting decayed off. That is our experience. But I think we have to follow that method which he is doing. Thank you.
Thank you, Professor Joseph. Excellent work. Just want to ask, those associated microbe that you had screened, and thought mainly for the nitrogen fixation. Have you also looked at other activities like enzymatic hydrolysis? Because I'm sure some of those might also be producing enzyme to hydrolyze those kappa figures.
Yeah, some of them, because even in the tissue culture medium, what we have observed is that, because we are putting, sterile, type of this or blade or hold fast, all this we tried. But after, 1 month or 2 months, we have seen these microorganisms emerging. And in and around this agar, in fact, it is a big hole, so it means that they are producing, so that we have to check,
Sure.
Yeah.
Hi. One curiosity-driven question. Let us do a theoretical experiment of having a 20-meter long string vertically dropped from the surface of the sea towards the bottom. And every 1 meter, I plug some seaweed material, same genetic material, 1 meter. Do you think at the end of 45 days, the growth will be different? The transcriptomic changes will be there, or chemical changes are there? Because as we go down, less sunlight and more nitrogen on more other things that. So do you think there will be difference in the?
See, light is less, but even we don't know, because for example, if you take brown and red algae, they have accessory pigment. Even if it is slightly deeper, they can, diatom, cyanobacteria, all these can take a little deeper. But regarding transcriptome, I think that there will be changes. But regarding growth, I'm not able to answer that. Yeah.
We'll take one final question.
So, just I'm curious. Actually, you have shown one slide that NIP8 gene.
Yeah.
The nitrogen.
Yeah.
Convert.
Yeah.
Atmospheric nitrogen to ammonia. Have you done any acetylene reduction assay?
No, that we have to do, because what we have done is only qualitative test and ammonia production. That is not enough. We have to do GCMS based acetylene reduction assay to exactly quantify this. That we are planning now.
So you have estimated the ammonia production.
Ammonia production we have estimated.
Using
it's very difficult,
Nestler reagent.
Yeah.
So have you seen any HDK gene there? No.
No. Only this NIFH we have, but all these strains are producing ammonia, but not all we are seeing this NIFH,
Okay, thank
that
you.
we have to look into. Thank you so much.
Okay, thank you, ma'am.
Yeah.
Okay.
Thank you very much, ma'am. It's a very nice presentation. So I have a question. So have you ever thought of working on disease-causing bacteria? Disease-causing bacteria of seaweed, because it gives lot of loss to the cultivation. So if we can generate some value out of that, then that will also be beneficial for the seaweed community. So have you ever worked on that, or have any plan of doing that?
We have not worked so far, but we are thinking, because we are approved diagnostic laboratory for aquatic animal health diseases, but we are thinking that we must include seaweed pathogens, seaweed disease, seaweed health. In fact, we have included this as one of the areas, but we have not worked so far. But I understand it's very important and we will be working on that.
That will be a very good
Yeah,
initiative,
yeah,
ma'am.
yeah.
Thank you.
Yeah.
Okay, so I understand there are many more questions. Everybody is encouraged to submit your questions to the email address we have shared, and we get back to you. So, without any more delay, thank
Yeah.
you so much,
Thank you
ma'am.
all
Thank you for
for
that wonderful
patient
talk.
listening. Thank you so much.
Our next speaker is Dr. Shams Yazdani. He is the group leader in Microbial Engineering Group and has been the coordinator of the DBT ICGEB Advanced Bioenergy Research at ICGEB since 2012. He is working on ways to break down the recalcitrant biomass from non-food based bio sources for biofuel production. And in fact, his work was recently highlighted in a DBT Spotlight meeting. So today he'll be talking on chemical and enzymatic processing of cellulose in the context of seaweed polysaccharides.
Thank you very much, organizer. Thank you, Dr. Devasish Shri, for inviting me. After attending almost 1.5 days of this conference, is a huge learning, and also I learned why I was invited here. Dr. Devasish wanted somebody who is more ignorant than him on the seaweeds. so, so you understand that I'm actually completely zero in the seaweed, but he kind of tried to save me by giving this interesting title, and he gave me this title, and I appreciate for that, which is basically chemical and enzymatic processing of cellulose. So lessons for processing seaweed polysaccharide. So whatever in the last little more than a decade I had been working in this area, whatever lesson I got, can I transfer to you so that you don't go into the wrong path? You really follow the path which will give you a application based output. I'll start with the cellulose and then I'll come back towards the end for the seaweed. And if I cross my timeline, probably you can stop me. so Cellulose is the most abundant polysaccharide on the earth. I'm not going into the ocean at this moment. So therefore it is, it has a great value by itself. And, almost all the agricultural residues, if you take out the grains or the juices, the leftover is what we call it as a lignocellulosic biomass, and it will have a plenty of cellulose in it. Now, in those agricultural residues, a lot of them are being wasted, so it will be a great value if we can take it out. And Shri in his presentation already pointed out that these residues may not be enough in the future. So that's why seaweed is going to be very, very important. But at this moment, even we are not able to use these residues. So we are trying to see how quickly we can facilitate the whole ecosystem so that these residues can be used for a various purpose, not really being burnt like currently it is being burnt. So if you look at the structure, basically it's composed of the lignocellulosic biomass or agricultural residues broadly will composed of cellulose, hemicellulose and lignin. And there's a general flow diagram to suggest, and this flow diagram I'll come back to you for the seaweed as well, which gives me basically a bird eye view of how you process any biomass to a certain product. So if you look at the agricultural residues, the first thing is the hardening of it, which is coming from lignin, which fortunately you don't have in the seaweed. And that lignin normally is removed at this moment by thermochemical pretreatment. There's no good biological method at this moment available. If it is developed, that will be great savior to the environment as well. At this moment, it is thermochemical treatment, which removes lignin. And then you still have a cellulose and hemicellulose, which are difficult polysaccharides. Cellulose more because it's a crystalline in its nature. Hemicellulose is still not that difficult to hydrolyze. So you need a cellulases and hemicellulases. And at this moment, they are very, very expensive. There's a huge monopoly in this field. There's only one company, which is Novozyme, which supplies enzymes for the biomass hydrolysis. So therefore, it is greatly needed to have some indigenous technology. And then once you have hydrolyze these sugars. So remember, cellulose will give you glucose, but hemicellulose will also give you C5 sugar, which is xylose. So typical yeast will not ferment. So you have a mixture of sugar. So the problem that you will have in seaweed, you also have here, that you have a different varieties of sugar. and then you need to engineer host so that it can take all sorts of sugars that are present. Typical yeast that ferment glucose to ethanol will not take xylose and all that. So therefore, there is a metabolic engineering part that are involved here, which will be involved in the seaweed fermentation as well. And then, once you have engineered the host to produce ethanol, you also think about now you have a backbone or the chassis which can take up various sugars and convert them into ethanol. You can manipulate it to convert it into many other products. So for example, here I've listed, because these work mainly coming from the bioenergy center, so fuel molecules, which are also used mainly for the cosmetics and other chemical industries. You can think about manipulating the host to consume those various kind of sugars to produce by working at the end pathways. So that's what we had been doing it. I just made this slide to show you the difference between the chemical routes and the biological routes in terms of converting cellulose into monomeric glucose. So whatever way you take, normally it goes through the oligomers or dimers and then to the glucose. If you're taking the chemical routes, so right now there are various ways, but the typical is this supercritical water at 400 degrees centigrade and very high pressure, or you take concentrated sulfuric acid and with a high temperature. And of course, these are going to be not very environment friendly techniques. And eventually you will see that along with the glucose, you will see a lot of byproducts also coming up. And your metallurgy to handle these kind of chemicals are also going to be very, very expensive. And therefore, preference to the enzymatic route comes where you need a series of enzymes that will convert first to oligomers or dimers and then to the glucose. So now that people realize that the enzymatic route is a better route to convert cellulose into glucose or hemicellulose into glucose and xylose, they also realize that it's not a work of one or two enzymes. So for example, you can, by analogy, think about starch, which we chew, and in our saliva, there is a amylase and glucoamylase. These two enzymes will completely degrade starch into glucose. On the contrary, these cattle have to sit and chew those lignocellulosic biomass hours and hours and hours, and then only their rumen is able to digest these. So of course, these comes because of the crystallinity. By nature, these are structural polymer. They are not a storage polymer. So therefore, digestion becomes very, very difficult. And what I've listed here are, we know them as a core enzymes like exocellulase, endocellulase, LPMO, lytic polysaccharide monooxygenases, beta glucosidases. These four enzymes are, we call them a core enzyme. They are necessary, but they are not sufficient to give you a very, very optimal hydrolysis of the biomass. So there are, if you will start looking at it, there are hundreds of different auxiliary enzymes that help those rumens together to digest those biomass. So the classical problem here is that if there are one or two enzymes, you can recombinantly express it in a well-known host and produce and express it, secrete it in a high quantity. But if there are so many enzymes, you can't do genetic engineering of those enzymes. So you go back to the nature and ask, what is the best host that can give you the composition of these enzymes? Although we started a little early, we were also, just like today we are talking about seaweeds as early as today. About 12 years back, I was at the same situation when I started working on the enzyme. And then the best literature at that time, which came in Nature and Science, suggested that the best enzyme you will get for cellulose hydrolysis in the gut of the termite or the insects that digest the biomass. Well, that made sense, those insects like rice stem borer, pillbug, termite, they were kind of digesting those lignocellulosic biomass, and we thought that okay, that's the best place to get enzyme. So we started looking at or bioprospecting the gut microbiota and looking at which one can give me the best enzymes. And fortunately, we had a very strong insect resistant group at ICGB, so they already were having all the infrastructure to isolate insect microbes and all that. We took help of that. So sorry about that. Oh. Okay, yeah. So, a lot of good science happened. We not only isolated microbes, we also adapted them to digest the rice straw for a very, very long time. And we shown that actually the rice straw digestion, can actually increase over the period of time with those evolved microbes. And we also discovered many, many new things during the process. We realized that the rice straw is also very nicely organized with the xyloglucan, in addition to so branching of that. And there are some carbohydrate binding modules that will bind to those branch. So we discovered this CBM46 CBM3 combination that used to bind to those branching part of it, which is xyloglucan branching, and help the xyloglucanase enzyme to digest those branching part. So we expressed, characterized all of them. We made many of the bifunctional enzymes. And to our luck at that time, and thanks to Dr. Shri, we had a collaborator called Dr. Arvind Lali, Professor Arvind Lali at ICT Mumbai. So whatever we claimed very good, we used to send it to him, that you test it, because he had access to Novozyme enzyme. We didn't have Novozyme, refused to give us enzyme. So we used to send it to him. And after, whatever best we used to say, he used to, the response used to be that you are not best. You are only 20 to 25% of what Novozyme is giving. So that after many years of this struggle, we realized that probably somewhere we are not right. If we have to compete, publication, as Dr. Sau said, was not really the aim. The aim was really to give a hard competition to this monopoly, which was Novozymes giving. So we actually did some survey and realized that perhaps the best platform to look for enzymes for cellulosic biomass is going to be fungal platform. So we then went back and screened many fungal ice. So we kind of changed our whole hypothesis. So the way we were screening earlier were based on the enzymatic assay, some of the cellulase, endocellulase, or exocellulase, or beta glucosidase. Then we asked the question, now that we know that these are only the core enzymes, there are many other enzymes that play a role. Should we really look at the individual enzyme assay, or should we look at really a combination of assay and find out which combination gives the best biomass hydrolysis? With the help of DBT support, we also had a very good high throughput screening system. So we thought, okay, let's ask this. And we had a very talented PhD student who was MTech. He knew some of the mathematics, which I didn't know. So he developed this mathematical model, he called it as a weighted sum matrix score. He said that I will not only consider the enzymatic assay of these key enzymes like endoglucanase, exoglucanase, and beta glucoside, but also look at how much sugar is released from the pre-treated biomass. So we got some pre-treated biomass from Dr. Arvind Lali, and we also incorporated all these metrics in our weighted sum matrix score and started screening the fungal species that we got from the culture collection as well as our own soil isolates. And then based on all this, we figured out that one of them is giving higher weighted sum matrix scores as compared to others in terms of getting into this equation. So we picked this up. Then these are different because as a scientist, you keep on asking questions, and questions will keep on giving you the publication. So while you're doing application research, you also get side by side good publications. So that's something that I wanted to highlight. So basically, first thing was that if this is a good strain, then what is it that is producing? So you have to go to the genomics and proteomics. The unfortunate part about the new strain that you're isolating, if you want to go to a mass spec is data based mass spec. So you do proteomics and you will figure out that only 30 or 40 proteins are giving you a hit. That's not good enough. So then you have to build your own database. You do the genome sequence, build your own database. Then you go back to the mass spec and ask questions, and then you will see that there are 300 and 400 proteins are getting secreted outside. So these are the things that you have to learn over the period of time, when you go into the deep science. So we learned that what are the different kind of proteins that are getting secreted by this particular fungus through this protein, and we realized that there are many different, majorly almost 80% of them were responsible for biomass hydrolysis as compared to as coming in these sections. But also most important enzyme and the highest one was this GH7, which was one of the exocellulase. To prove just the hypothesis that okay, this strain is really giving us good enzymes, we purified one of this highest expressing enzyme, which was exocellulase. Exocellulase is the very interesting. It's the most important enzyme for hydrolysis of crystalline cellulose. So it really is a real workhorse. Pull up the chain, it will walk over the chain, it will keep hydrolyzing. We call this a threading processing cycle, dimer of glucose, and keep walking until the end of the chain. Now, this particular enzyme, they were known to be best so far from Trichoderma ricci species, and Novozyme was playing big on this basically. So what we said, we got the enzyme from Trichoderma ricci, we purified our own enzyme, and we did some of the kinetics, and we realized that in almost all criteria, our enzymes were behaving better than the Trichoderma ricci enzyme. And the most important part was the inhibition part. So most of these enzymes, if you have a high amount of cellobiose, which is its product, it gets inhibited. We found that Ki of our enzyme was much, much better than the Trichoderma ricci, and the major reason for this was this extra loop that we see at the product expulsion site. That was helping the product to come out faster as compared to the Trichoderma ricci. And when we published this, just about a year later, so this was a model, then NREL published a proper crystal structure of this 1 year later and published in Nature Communication. So that also supported that, okay, we are going into the right direction. Now, once we made our platform, now we have to go further because efficiency and titer both are going to be determining the cost of enzyme production. So we have to beat Novozyme. So we have to go to the molecular biology of this. Now, the first problem you will face with any natural isolates that you have collected from nature is the tolerance or resistance to most of the antibiotics that are used to manipulate the host. So for example, in the fungus, typically these are the antibiotics that are used for the transformation tool. Now our fungus was tolerant to all the antibiotics, so we were not sure what to do about it. Now we went to the transcriptome analysis, and we tried to understand why this is tolerant to antibiotic. And the reason we could guess was that it was expressing a lot of efflux pumps. So as soon as antibiotic was going in, it was thrown out. So we thought, okay, let's block, because there are some blocker of efflux pump known. So during the transformation system, we added those blocker of efflux pump, and we realized that, okay, now it is able to, we can make it sensitive to antibiotic. And then once it is sensitive to antibiotic, you can develop a lot of tools to, including CRISPR-Cas9, to transform this host. So after we were, confident that we can transform, the first target for increasing the enzyme expression is the repressor of enzymes. So this is a transcription factor. Homolog in the yeast is called MEG1, which will, as soon as you have some enzyme expressed, there will be some glucose produced in the media, and that information will go that there is no more need of enzyme. So this repressor will block the enzyme production. So we annotated the repressor in the genome, and we deleted it and found that we can improve the enzyme production by more than twofold. Now it opens up further new things. Because you have tools, you can add more important enzymes. You can study what are the important enzymes that could be added. And some of these, for example, beta glucosidases and all that, I will also come into it. So the out of the two most important enzymes, I told you about the exocellulase, which is cellobiohydrolase. Another enzyme that is more recently discovered called lytic polysaccharide monooxygenase. Now, endocellulase does not work on the crystalline cellulose. It only works on the amorphous cellulose. For the crystalline cellulose to work as an endo, that means to break the chain in the middle, there's another enzyme which is called lytic polysaccharide monooxygenase. Now remember, there is a cellulose chain, very long chain. Now only exocellulase is going to work, which is towards the end. So the efficiency will be very poor. So if there is a lytic polysaccharide monooxygenase, it will keep on making a nick in the middle and middle and middle and generating more chains for the exocellulase. So therefore your overall efficiency increases. So what we did was we actually made a recombinant strain where we express both of these enzymes in a I mean, we dosed copies of more of these enzymes, and we showed that when we dosed individually as compared to individual, the copying both of them together showed a lot of synergy. So actually, this combination along with the repressor gave us a very good efficiency of production as well as activity, and we kept on improving enzyme activity. We also, discovered some of the enhancer of enzyme production. Now, this is the uh, you get cellobiose by the activity of LPMO and cellobiohydrolase, but eventually cellobiose will have to be converted to glucose, and the beta glucosidase is the enzyme. Now, the unfortunate part about the beta, so most of the biomass hydrolysis takes place in 3 days. 72 hours is a typical time at 50 degrees centigrade to completely hydrolyze lignocellulosic biomass into glucose. Now, if it is happening, and this is over the various period of time, people have realized that this is the best condition you can have, and maybe because of the thermodynamics or various reasons. Now, for that reason, 50-degree your enzyme have to survive till 3 days. Okay. Now, cellobiohydrolase and LPMO, fortunately, the one which we had in our strain, were not that bad. They were able to be mostly active in the 3 days period. But beta glucosidase, which is the enzyme which you need towards the end, when these two enzymes have worked, you have a lot of accumulation of cellobiol and then you need it, and this is the first enzyme that get inactivated at 50 degrees. So we realized that what do we do, and again, because remember, when you are developing and you are competing with a commercial partner, you have to remember that you have to be generating new IPs as well. You can't take any known beta glucosidase which has IP or known host. For that reason, we didn't work on trichoderma. So we thought that let's look at this host only. What are the different beta glucosidases that it is producing? And can we take one of them and engineer it? So we profiled all this. We realized that this host can produce 8 different beta glucosidases. And through the exoproteomics, we realized that 3 of them can come out. Okay. Now we deleted those 3 of them individually to see which one is making most impact in the activity, and we realized that, and you showed that this is basically a MUG assay, but cut story short that 2 of them were showing a lot of impact in terms of beta glucosidase activity, BGL3 and BGL3. BGL3B was a little strange enzyme because when we deleted this enzyme, not only beta glucosidase activity went down, but also cellobiohydrolase activity went down. So we didn't really understand why is this happening. So for the time being, we didn't focus on this. We started focus on 3A. So these are the basically the structure of all these 3 enzymes. So 3A is this, and we took help of, we have a very good bioinformatic translation group. They took help of him that look, we don't want to disturb our catalytic side. We just want to do a rational engineering so that our thermostability is maintained, activity is not lost. So there's a various molecular dynamic simulations and different kind of structural in silico study has taken place, alignment with different mesophilic, alignment with different thermophilic enzymes, and making, trying to make some enzymes that could be thermostable. So they came up with the two kind of varieties of BGL, mutant A and mutant B. Mutant A had 6 mutations, while mutant B has 13 mutations. So they said try both of them and let's see which one works. Now, when we tried mutant, so this is our wild type, which is BGL3A. Mutant B has lost its activity within 1 day. So that means this more mutation wasn't really happening. But this mutant A actually maintained its activity until 7 days without much loss. So that was really a music in our ear that, this is a mutant that can survive more than 85% of activity until 7 days, and that's the one that we wanted. With a lot of these kind of changes that we made in the fungal host, we try to convince, and in our with the help of Dr. Arvind Lali, we could also test at that time that these enzymes are very good in terms of activity, very close to the novozyme enzyme. We could convince DBT there is a very good host to scale it up, because any industry, if you go, they will ask for whether the host is scalable or not scalable. So we had a 20-liter fermenter at ICGB. We showed at that, we developed all the protocol. I had some previous experience of scaling up malaria vaccine in E. coli for the GMP production at, in the industry. So I used all the technique, and we hired a facility in Bangalore which had a 30,000 liter reactor. So we scaled it up to 15,000 liters scale. We made some finished product, and then we send it to Praj for validation, and the validator showed that it's fairly close to Novozyme. We also tested our enzyme on with the help of Center for High Technologies and Ministry of Petroleum. So the most advanced 2G ethanol plant. So remember, these enzymes are going to be only used by oil manufacturing company, which has a 2G ethanol plant. Now, private players are almost not existing in India. Government has given mandate to oil manufacturing companies to build 2G ethanol plant. Indian Oil Corporation is almost very close to functioning this 2G ethanol plant in Panipat. Center for High Technology helped us in getting the biomass. They shared all the standard of Novozyme and all that, and then they asked us to test it. So we tested it. So our enzymes were very close in terms of activity to the Novozyme enzyme. And when we add those engineered BGL, thermostable BGL, our activity further improved. So that gave us a lot of confidence. Further, Praj became our validation partner, and they actually visited ICGB, and they spent 2 weeks to completely validate. They did techno-economic analysis, and now we are realizing that for 1 day, so the requirement, I mean, you should understand the volume, and that's probably going to be a similarly true for the seaweed. So 2G ethanol plant in I mean, so they realized that unless you produce 100,000 liter of ethanol in one 2G ethanol plant, it's not going to be commercially viable. So scale is 100,000 liter in one plant of ethanol. 100,000 liter of ethanol will need 500 tons of biomass. 1 day, so I'm talking about 1 day. 500 tons of biomass will need 10 tons of enzyme every day. So 10 tons of enzyme every day in one plant, and you can multiply by 365 per year in one plant, and there are 4 plants to be operational by next year, and there are 12 plants to be operational by 330 40, and all of them are dependent and have signed agreement almost with the Novozyme to import enzymes. So it's going to be a huge opportunity. So we told that at least give us 20% of the opportunity. So they said, okay, if you kind of follow our tender, it says that you should have a 3 years of demonstrated ability to produce enzyme at a commercial scale. So these are the policy that we have to actually work upon to change. And I think there's a lot of secretary level talk discussion. But I said, okay, if not, then at least give us enough enzyme for our plant to run for 1 day. There's a 10 ton of enzyme. So where do I produce 10 ton of enzyme? In India, there's no facility to produce 10 ton of enzyme. So we are actually, talking to some of the industry outside who can actually make and send it to us. So this is a situation at this moment. I'll quickly, these are something. So I mean, why I put this, that remember, IP is one thing that you should keep in mind every time when you are generating something. You are going to compete with some of the multinational. So you should have an IP in place every time. I'm going to quickly shift my gear because I realize that seaweed, you will get the sugar, but some of the presentation from yesterday and today, what I heard, we're not sure whether we are utilizing all the sugars or not. So there are actually galactose, but there are anhydrogalactose and there are sulfated galactose. Therefore, metabolic engineering will become inherent problem to you in the future. And I'll just give you some of the case study that what we did. As I told you that, this part is the fungal part, but the bottom part, because of my training, I actually did all this in the bacterial host, where C5, C6 engineering I did, and some of the long tail hydrocarbon I'll just quickly put. So if you take E. coli, for example, and you grow it under anaerobic condition, it will grow, but it will produce mixed acid fermentation. It will produce a huge amount of lactate and other thing, and the ethanol yield will be very low. So our challenge was, I mean, the interesting thing about E. coli is that it can consume glucose, xylose, arabinose, galactose, all of them without any problem. So what we did metabolic engineering to convert all of these sugar into ethanol with a very high yield. And then to improve the productivity, we did the what is called adaptive laboratory evolution. And then we produced 50 gram per liter, which is close to 6.5% of ethanol in E. coli via xylose fermentation. And because nobody had done this kind of work, so you can get a US patent grant. A lot of these work are really focused on yeast. Because we have worked on E. coli, we could get the US patent granted. Well, second challenge was that even if they are able to consume glucose xylose, but they're not consumed simultaneously. So we did further engineering so that both of these sugars were consumed simultaneously. And there are some science behind it, which I will not go further. But this is the last part of my talk in terms of metabolic engineering. We also, in the event of Bioenergy Center, try to produce some of the advanced fuels. We went into jet fuels and cetryl alcohol, basically pentagecane and cetyl alcohol is hexadecanol or octadecanol. Now, all of these can be used in the fuel, but also has a very good market in the cosmetic industry. And, these are the pathways, quick pathways. But basically, this inspiration came from a science paper which published first time the pathway from the cyanobacteria they imported in E. coli coming from the fatty acid synthesis cycle. And what they could not decipher in that paper was that there was a alcohol corresponding to aldehyde in E. coli. Where is it coming from? So we discovered that in this paper. We discovered what are those aldehyde reductase that was responsible for alcohol production. And we further did a lot of metabolic modeling. And that's another thing that I wanted to tell you, that huge amount of metabolic modeling will also be needed along with your metabolic engineering to convert it. These are some of the interesting paper which in the cyanobacteria, a bit of work that we have done. And there's some, again, making thermostable, one of the alkane producing enzyme. Now, this is just to not to it's just to warn you that a lot of metabolic modeling would be needed if you want to do a very good metabolic engineering. And we could show the highest production of fatty alcohol, which is also called cetryl alcohol, and alkane so far reported. So that time when we published, Hindu took up this article. Now, coming to this is not my paper, but I wish it was my paper. Okay. So just to inspire you that, it's a tremendous, potential in the seaweed area. Innovative science can lead to not only potential application, but also a very good publication. So this was, when Shri, we were discussing in the earlier time, this paper came, and this really motivated me, although I didn't go into this direction. So basically this paper, where an engineered microbial platform for direct biofuel production from brown microalgae. So they've taken brown microalgae, basically alginate, and converted into the ethanol. There's another paper. So this is 2012, this is 2014 in Nature, same group published another paper when they imported a similar kind of pathway in yeast. So there is E. coli, there is yeast, you have a science paper and you have a Nature paper. Okay. So basically, I don't want to go into the detail, but I wanted to tell you that complexity that these people have solved, and that's the reason why they got a good paper, is how they taken the alginate breaking enzymes and the sugar that was eventually made the metabolic engineering to convert those sugars to eventually ethanol in E. coli. So they could produce 4.6% of ethanol in E. coli from this particular pathway. So they actually had discovered through a lot of in silico works. A lot of in silico work was there. The Vibrio from the NCBI search realized that the Vibrio can produce a series of enzymes that can degrade alginate. There's a 32 kilobase of the genome size of Vibrio. They put all the enzyme together in the E. coli and showed that it can. So a huge opportunity here, and this is because we had been talking about gracilaria for long. Since yesterday I'm hearing there's one host that can make a lot of difference, and we should focus on this. I tried to search a bit of a literature and realized that, you know, couple of literature really made a sincere effort to characterize the biomass. The first thing, the ability, if anybody wants to see your research, we need really a full characterization of the biomass. And I think compositional analysis, people have done FTIR, 1 and 2D NMR spectroscopy, 13C and proton. These are something that must be accessible to do a full characterization, because remember, we are talking about, I mean, she always says that, ethanol is a bulk volume. If you're talking about fuel, you can't leave one sugar molecule and just focus on another one. You have to consider all of them. You have to consider that we have to accept the fact that this particular R could be H or methyl group. This particular R could be methyl group or sulfate group. And therefore, these sugars will be a mixture of all those. We need enzymes that can digest all of these together. But we will need enzyme, we'll search enzyme only when we know exactly what we are looking for. So compositional analysis is very important. Once you have all these mixture of sugars, then we certainly need and of course, as Dr. Joseph said, we need a very good bioprospective enzymes which are basically high tolerant, rational engineering for high activity thermostability. These are something that is certainly doable nowadays. And because I realized that there are some cellulose component as well, you can try to add my enzyme and see how it works with the cellulases. And then you need, some of those engineering of transporters and metabolism for all the sugars, co-fermentation, so that you're not missing out the toxicity of one sugar over others, and of course the high tolerance. So I'll stop here by thanking my team, my leaders. They are the leaders. They guide us what to do basically. Extremely beneficial team I have. I'm lucky to have them. A lot of good collaborators, and thanks to certainly DVT for funding in a amount that we really required, and there are some fundings from other sources. So thank you very much. I'm happy to answer.
Yeah. I won't say excellent talk, Sam. You put me into depression right now. So before the chairman says that no more questions, I would like to ask, are you still interested in the science or nature paper in the seaweed biology? I think the time has come. Lots of 10 years of efforts you have put in and understood step by step. Some of it can be directly translated to the seaweed biology problem that we have just discussed. I may not have tons of enzyme for you, but I have tons of question for you. The one being, you said that some enzyme that works at 50 degrees centigrade, and to discover that enzyme, it took some time. From normal physics, do you think by increasing the pressure, we can reduce the required temperature, and the same experiment can be done at a low temperature?
I don't think anybody has done this. Provided these enzymes are not sensitive to shear pressure, because the 3D structure of enzymes.
I'm not talking about a huge amount of pressure. Just we can do the experiment in a vessel where we can steadily increase the pressure and see
if it
at a 5 degree lesser temperature, maybe the same experiment happens.
Sure. I'm sure it's doable. Things can be tried.
So we'll take one or two more questions.
Sir, I'm Chandrakant from ILS. So my question is like I have observed some of bacteria those are utilizing complex sugar. So how what system should I use for isolation of this like any enzyme responsible for it, and how to proceed?
So Again, ask the right question. So if your idea is just to discover enzyme, so there are two aspects. One is discovery of enzyme and production of enzyme. These are two independent things, could be independent, could be together. Now, if your aim is just to discover good enzymes, so you look at the specific enzyme that you're looking for. If this bacteria is, you should have a good assay method to screen. Many times the problem is that we believe, let's say we have screened 100 bacteria and we found one of them giving best, and we believe it is the best. But how is it better than the others? Commercial enzyme or the one who has published earlier is very, very important. So think about a benchmarking. You know, how do you really classify your enzymes compared to the already a benchmarked enzyme? And that will give you a better direction, you know.
Thanks.
Sham Sabin, this is truly impressive. I have two questions. One is, you mentioned the characterization of biomass is really important, right? Who are the groups in India that could do this? Do you know of any groups in India who could we give them a seaweed biomass and they will actually be able to tell you exactly what the carbohydrate structure is, whatever? Are there any groups in India who can do this? We should collaborate with.
I think I don't, I mean, there are carbohydrate chemistry people, but I think I'm not sure whether they have access to all these high-end equipments. I think we'll need to collaborate with the people. For example, ICGB has NMR, but the people who are using NMR do protein chemistry rather than carbohydrate chemistry. But the people, I think NIA has some of the good carbohydrate chemistry people, like Gopalan and all are there. We can convince, they don't do, I mean,
So we'll have to
classical.
really put together, stitch together a group
We
that
have to
will can
stitch
take
together.
a Russell area and tell us, oh, it's got this is a
Exactly.
sulfation. I think without that, we'll be working blind,
Exactly.
right? Okay. So my next question is about, you mentioned the E. coli, which bioarchitecture lab did. And now this was truly impressive, right? 2014. You know where bioarchitecture went is that they bet on the wrong biomass. Okay, there is no way that I see that temperate algae, which will be grown 2 times a year or once a year, can ever get down to the level of the cost of the biomass, because that's that. I mean, I've talked to them, and that was the problem. They were always looking for cheaper biomass. We don't have that. I believe we have a very unique situation in the tropics, because only in the tropical countries you'll get this. But we shouldn't discount the fact that in Puerto Rico, the US is very strongly looking at it. But this is quite a race right now, that we can, with our advanced farming systems, we think we can bring down the price. But we really need this holistic approach that we're talking about. One of the things I wanted to check with you about Bioarchitecture Lab, did their E. coli have saltwater resistance?
I don't think they've tested.
It's because that, I
Yeah,
believe, is going to be a very practical consideration. Okay. Now I know that there are saltwater tolerant E. coli. Okay. And so when we do our whole process development, we should actually keep that in mind. Okay. A high salt, capable of working in the kind of saltiness and yet produce the ethanol. Now that will be an extra challenge, because you have to tolerate 5 to 6% ethanol plus 3.5% salt. This is not trivial. Okay, so it could be totally exciting when I see several science nature papers coming out of this.
No, absolutely, I agree, Shri. I think they didn't consider this aspect at all. It was a proof of principle. So, yeah, totally agree with you. Yeah.
So, sir, I'll just get to the question. So, there are many methods for taking biomass to energy, like for example, HTL, or even in case of this agriculture byproducts, you can go directly to charcoal. So, how do the energy balances compare, and which method currently has the best positive energy balance?
Yeah, I mean, very difficult for me to answer. Probably a good chemical engineer, or maybe, yeah. I think, yeah. So Shri would be a best person, actually.
It's an extremely good question, and that's something we have grappled with. Okay, and the answer came to us very recently. There is a certain amount of energy in biomass. That is roughly, biomass has about 9 to 14 megajoules per kilogram, depending on the kind of biomass. Okay, if it's seaweed biomass, it's 9 megajoules per kilogram. If it's starch, it's probably 14 to 18. Correct me if I'm wrong. Now, when you convert it to ethanol, actually you'll find out that you'll recover most of the energy into the ethanol. Even though you recover only 3 carbons out of the 6 carbons, the other 3 go off into carbon dioxide. But you will recover because ethanol has a high calorific value. When you do something like HDL and crude oil, again, you'll recover quite a lot of the energy, up to 80% of the energy. Okay. So then the consideration is not about energy recovery, the consideration about what is the practical use. Okay. Now, if you want to, and that's where it gets very interesting, because ethanol is very interesting because it's already available in bulk quantities. Many people have now adapted their processes to take ethanol, put it into fuel, or dehydrate it, make it ethylene, and make polyethylene from there. So the large oil companies which control the access to the customer, okay, you want to give them a product that they can use. So when we talk to them, they say, give us ethanol because hamara factory already ethanol se chal raha hai. I said, can I give you crude oil? He said, wo bhi acha hai, but you know, then so both are possible. Both are possible. Crude oil, if you can make bio crude by HDL, it will plug in upstream. You start blending it with the crude oil and it will go down. So both approaches are valid. But the ethanol approach to ethylene is somehow sensed as cleaner. It, you know, not all companies do prospecting for crude oil. They buy their crude oil and maybe they make ethylene out of that. They can plug in. Both are valid. That's all I want to tell you. Both approaches.
A great talk, Shams. One question is, with your vast experience on this enzymes, are we close to synthesizing an enzyme artificially that can do the job for us?
Okay, so we are close, I would say. So far, what we have done is really a rational engineering. But recently, we have started some new project on plastic degrading enzyme, and there we are really taking a very, very, I would say a jump in terms of synthesizing enzymes, because what we realize is that the known plastic degrading enzyme so far is a PETase, but the main plastic is polythene. For this, there's no enzyme. The reason being that there's no enzyme that efficiently break carbon-carbon bond in the polythene. And for that reason, it has to sit properly on the polythene. So there are some enzymes that can modify the end chain of carbon hydrocarbon, but that doesn't sit on the solid surface. So we're trying to design some of those enzymes completely new and see if it can work. But this is still in the designing phase, I will say. If we get success, we'll come back to you.
And then the other thing is, like, can you use a hybrid way of digesting this cellulose, like enzymatic as well as chemical? Initially, if we can, because getting tons of enzymes, it's I mean, after doing such a hard work and then eventually coming up with a requisition of, can you give me a ton of an enzyme in a day? It looks ridiculous.
No, the thing is, Novozyme is willing to supply you enzyme. Okay. So the problem with the chemical, people have already tested a lot. If you have adapted chemical, you are adapting a lot of many other things. See, your metallurgy is going to be completely different. If you're going to use concentrated sulfuric acid continuously in your vessel, you need to have very expensive metallurgy to tolerate the concentrated sulfuric acid. All the nuts and bolts will go corrosive. Your supply from one vessel to another vessel will be a problem. I mean, I was part of the team for one company called Quantum paper mills, which BRIC funded, and their protocol was basically the acid hydrolysis. So they face this problem, and the second problem is gypsum. So concentrated sulfuric acid at the end of the day will become gypsum. So you will realize that ethanol तो कम बन रहा है, gypsum ज़्यादा बन रहा है, gypsum की factory खोल दो. So I think people have tried and tested that pathway, and they certainly believe that enzyme is the only way forward. Even if we do less now, let's stick to the less. and try to see if we built our inherent capability to produce enzyme. But at the same time, Novozyme is willing to supply you enzyme as much as you. They have a huge facility in China. They can, supply you bulk of enzyme shipment. At this moment, you won't believe, even without cellulase enzyme coming, the maximum shipment import in the world is to India from China for the enzyme. So enzymes in India is already has a huge market in your detergent, in your food industry, in cosmetic industries. So it's going to be just added up. You have a double of that.
But what kind of fermenter size is required for that?
So I tell you, we made some techno-economic analysis. So we realized that you will need 100,000 liter 7 reactor of that. That would give us most of the enzyme requirement in India. So right now we have 30,000 liter reactor. That is maximum.
I think we have an online participant who has a question. One last question, maybe we can take from there.
Hello, sir. This is Bala from BRIC-Institute. It's a very interesting talk. I had a question regarding to your discovery of LPMOs, especially the lytic polysaccharide monooxygenases, and employing these for biomass degradation. So I was just wondering how efficient this enzyme is going to be useful for pre-treatment of hemicellulose versus cellulose, because LPMOs will be more specific to cellulose. So in that case, if we lose cellulose, then we don't get more, biofuel out of it. That's bioethanol. So is your idea to convert a LPMO to be more specific to hemicellulose so that you do a pre-treatment, or what is the rationale for using LPMOs in your study?
I'm not sure I understood, but let me just try to answer your question. I think you were asking whether LPMO that we have discovered is useful for cellulose, or it is good for hemicellulose.
Hemicellulose, yes.
So for hemicellulose, see, the use of LPMO is mainly to hydrolyze crystalline cellulose. So hemicellulose is not crystalline, right? It's amorphous. So you can get inside easily. But the problem is for the crystalline. So cellulose chain, if you see, they sit over each other so tightly because of this beta-1,4 linkage. Every cellulose chain microfiber sit on another microfiber and very tightly linked with the beta-1,4. And that's why, all our shirt and pants with the cotton, they're all cellulose basically. They don't get digested or degraded so easily. They are basically because of the crystallinity of the cellulose fiber. And this cellulose crystallinity comes because of the very nice hydrogen bonding between each of these chain, which are made up of beta-1,4 linkage of glucose. Now, because of this crystallinity, many other enzymes will not work. Only exocellulase work, and that also work towards the end. Now, if you want to make exocellulase function more actively, you need to generate more end. Now, that is what the role of LPMO is. LPMO is a monooxygenase enzyme. So it oxidizes. It's not a hydrolytic enzyme. It's a monooxygenase. So it oxidizes the chain in the middle. So it will generate more and more. So it's really needed for the cellulose. For hemicellulose, you don't, I mean, there may be some monooxygenases for hemicellulose, but you don't really need them, So
Okay, thank you. I have a second question. This is regarding the biomass characterization. So you have showed about the various enzymes which you have used for degrading, for example, xyloglucan in your biomass. So I was just wondering, do these structures differ between different biomass? For example, let's say a rice straw versus a wheat straw versus any other biomass, or within the wheat, do you see structural diversity in these biomass that we need to, emphasize on structural characterization and then going for a designer enzyme to create those structural glycans? Is there, would that be a feasible way?
So basically, cellulose chain remains same in all of them. There are beta-1 linkage of glucose. That doesn't change. So the variation happens in the hemicellulose portion. You're right. The glucose and xylose linkage, there will be some arabinose, there will be acetylation, and there will be linkage to the lignin. So these are going to be variable. Now, therefore, the pre-treatment technology actually play a very important role. So there are some, for example, if you're doing a dilute acid pre-treatment to loosen the biomass, that takes care of most of the hemicellulose. So that will convert hemicellulose into monomeric sugar. in the dilute acid pretreatment. So even if you have a variation in the biomass structure a bit, of course there will be, those pretreatment technology takes care of most of them. These enzymes that we develop would be more or less working with maybe 5 or 10% of difference in the efficiency to all the lignocellulosic biomass.
Okay. Sure. Okay. Thank you so much.
Thank you, sir. But I would request you to please introduce yourself so that we know.
From the
Thank you so much, sir. Thank you for that very informative talk. So our next speaker is Dr. Smita Midbhavkar from NIO. She is a senior scientist in the Department of Biological Oceanography, and her work involves microalgal taxonomy, ecology, and bioprospecting. She is very enthusiastic about the community structure and nutrient requirements of the phytoplanktons of coastal India. And today she'll be talking on marine microalgal bioprospecting, biodiversity, and biotechnological applications.
Okay. Good afternoon to all. So, at the outset, I would like to thank ILS for giving me this invitation. And today, my topic is marine microalgal bioprospecting, biodiversity, and biotechnological applications. Okay. So, we all know that global challenges like environmental degradation, population growth are putting immense pressure on our food systems. So for this, we need innovative solutions and alternative food resources. So the ocean food resources are a promising avenue, and marine microorganisms, they are providing us innovative food products and supplements. So amongst these marine microorganisms are the marine microalgae, and they are a promising resource because they have a fast growth rate and they are rich in nutrients. And by using these marine microalgae for various value added products, we can aid in the accomplishment of UN Sustainable Development Goals, that is the number 14, life below water. Now, microalgal bioprospecting, it involves the search for novel genes, then natural products, and also the novel microalgal strains. And these strains, they should be rich in the various bioactive compounds which have applications in pharmaceutical, nutraceutical, food and beverage, and also the aquaculture industry. Now, first of all, we should know the microalgal biodiversity. Now, microalgal biodiversity, it is very rich and it ranges from around 2 lakh species to several million. So, and in this, just 10,000 have been described. And we have only a limited number of species that have been explored for bioactive compounds. Now, why is this? In spite of the biodiversity being so high, only a few species have been explored for the bioactive compounds. Now that is basically because of the lack of expertise. Like we do not know how to get the sample, isolate, and also the processing of the sample. So that is a big problem. And also maybe there is a lack of access to the habitats like the open ocean or the coastal regions. And so what most of the researchers do is they rely on these collection centers for getting the strains. Now there is a big problem in getting the strains from the microalgal collection centers because we don't know when it was isolated and from where it was isolated. So if we do it from our local sites, so we know what are the environmental conditions. So accordingly, we can play with the different environmental factors when we are culturing that microalgae. Now, another problem is that in the course of our experiments, we may tend to lose the particular strain. So then also the culture collection may not have that particular strain at that time. Whereas if we isolate it from our local sites, then we can get it back. So also over long term, these cultures can change their characteristics and also that can affect the production of the valuable compounds. Now let me give a brief introduction about microalgae. Now these microalgae, they can also be found as epiphytes on the seaweeds. So there are different habitats. They are found in the coastal waters, the brackish waters, estuarine, and even in the open ocean in the euphotic zone. So they are basically microorganisms with a size of 0.2 to 200 micron, and they are unicellular autotrophic organisms. So since they are autotrophic, they also form the base of the food web. And during the process of photosynthesis, they take in CO2. So that's why they also help in the mitigation of global warming. And they contribute to almost 50% of the total photosynthesis on this globe. So now, what about the compounds that they produce? So there are various proteins, carbohydrates, vitamins, pigments. So here you see the different kinds of, in these pictures, you can see the scanning electron micrographs and also the light microscopy pictures of the different kinds of microalgae. Now they occur in huge diversity, as I told you all, and also different shapes and either single or in colonies. So you can see the solitary forms as well as they can be found in colonies. And there are the spiral forms. There are also the cylindrical, spherical. So there are various shapes and sizes. Now, what is the classification of this microalgae? Based on size, they can be classified into 3 different types. So the smallest one is called the picoplankton, where the size ranges from 0.2 to 3 microns. Then comes the nanoplankton, where it ranges from 3 to 20 microns. And the next is the microplankton, where the size ranges from 20 to 200 microns. So the smallest one, now they include both the prokaryotes as well as the eukaryotes. So the prokaryotes are represented by the cyanobacteria, and the eukaryotes are represented by various classes like Cyanophyceae, Chlorophyceae, Rhodophyceae, Prasinophyceae, and others. Now there are, based on the habitat, they can be classified into the planktonic as well as the benthic. So planktonic means they are found in the water column, they are free living, whereas the benthic forms are found attached to the substrates. So like I told, it could be a living substrate like the seaweeds, or it could be an artificial substrate like rocks, sand, etc. So now I'll just discuss about the main microalgal groups which are valuable in the bioprospecting. So the first comes the diatoms. This is the most dominant microalgal group that you find in the marine ecosystem. Now there are 2 kinds of diatoms. One are the centric diatoms with a radial symmetry, and the other one is the pennate diatom with the bilateral symmetry. So the centric diatoms, as you see in the top picture, so there are many, it is designed with many ornamentations, and those are usually for the exchange of nutrients and also to release the waste products. So the most primitive ones were the centric diatoms, whereas the most advanced are the pennate diatoms. So the pennate diatoms, they have a structure called as raphe, with the help of which they can attach to the substrates, and they also release exopolymers, which is not the case with the centric diatoms. So as you can see the SEM pictures, so each diatom, it has two valves, the epitheca and the hypotheca. So that ornamentations you can see, and based on that we can identify the different species. So based on the different kinds of ornamentations. Now the next group are the dinoflagellates. You all must have heard of this. So these are the different pictures of the dinoflagellates, and they can be identified with the help of the various grooves and the cell wall. So some of the groups, they have a cell wall, they are called as thecate, whereas the others, they do not have a cell wall. And as the name suggests, they have two flagella with the help of which they can move about in the water column. Now these dinoflagellates, they form harmful algal blooms. So they produce toxins, and these toxins also have various biotechnological applications. Now the next comes the green algae, which are the chlorophytes. Now they are two types, that is the chlorophyceae and the prasinophytes. And they have a cell wall which is hard. Those are the prasinophytes, whereas some are the soft in nature. So these usually are good for bioprospecting because the cells can be broken easily. Now these cells are rich in chlorophyll B pigment. And the next are the cryptophytes. So as the name suggests, they are hidden. So it is very difficult to identify these organisms because they are very soft and they cannot be preserved because they are very delicate forms. So these also have two flagella on the basis of which you can identify them, and they have a asymmetrical body with the invagination. So these have various pigments like the phycobilly and as a result of which you can find them in different colors like brown, red or blue-green. Now last but not the least are the cyanobacteria. So as I told you all, these are the smallest forms which we find, and they are the smallest smallest photosynthetic organism. So that is the Prochlorococcus that is found in the marine environment. And next are the Synechococcus. The size ranges from 0.8 to 1.5 microns, and these cells are rich in phycoerythrin or phycocyanin pigments and hence valuable for bioprospecting. Now, as I told you all, they are found even on the substrate. So you can see these greenish patches. These patches are nothing but the diatoms or the cyanobacteria that you find on the in the intertidal regions or attached to rocks or any other artificial or natural substrate. So why marine microalgae? Marine microalgae, it can serve as a resource because they have a very rapid growth rate and they are rich in nutrients. Then they can be cultivated in closed photobioreactors where the conditions can be controlled like temperature, light, nutrients. And so they also provide renewable, sustainable and eco-friendly products because they can grow all the year round. They are not seasonal. Now let's see about the bioprospecting, that is the upstreaming. So the first step that is involved is the isolation of the particular species. So about the isolation, it can be either done with serial dilution, which we usually follow for the bacteria, and or it can be done through agar plating also. So when we see the colonies, we can isolate it and we can streak it for further purification. And also there is an automated method where with flow cytometry we can sort it out based on the fluorescence and the size of these different cells. We can sort the different groups, but of course there are limitations. It cannot be done for all the groups. Those groups which are fluorescent, it can be done for that. And it won't happen at one go. So we have to do it again, repeat the process till we get the pure cultures. So what is the next step? So before isolating, we should also know which species is going to produce a particular kind of the value added product. Here I'm going to concentrate on the pigments. So if we know where the organism, what is the habitat of the organism, what kind of pigment it produces, so we need not waste time just collecting samples from all the ecosystem. You can directly go to the particular ecosystem and collect the samples, and then isolate the particular organism. Next, you have to grow it in under controlled conditions. So for that, this is a multi-cultivator which we usually use for exposing these cultures to different environmental conditions. And in this multi-cultivator, we can control the temperature, light intensity, which is very important for the particular kind of species. So it is species specific. Now through trial and error, we'll get the optimized condition for increasing the biomass, and we can then characterize for the various compounds that are present in the particular microalgal species. And these compounds will have various activities like anti-diabetic, anti-cancer, anti-inflammatory, etc. Now, what is the difference between the natural and the synthetic pigments? Now, presently, the pigment market is dominated by the synthetic form. Now, you see the natural pigments, they are biodegradable, whereas the synthetic forms, they will persist in the environment for long times, leading to pollution. And the next is about the toxicity. The natural pigments are non-toxic, whereas the synthetic ones, they can remain in the environment, they can cause pollution. And also about the carbon footprint, the natural pigments, they have a lower carbon footprint as compared to that of the synthetic pigments. And the resource efficiency, they can be grown under non-arable land, whereas the synthetic ones, they require extensive chemical processes and raw materials. So hence, looking at the advantages, these microalgal pigments can be sustainable and eco-friendly choice for the industries which seek greener solutions. Now, what are the different kinds of pigments that are found in the microalgae? One is the carotenoids, and that is the carotenes and the xanthophylls. These are the two types. Now, what is the difference? The carotenes are hydrocarbons, whereas the xanthophylls, they have oxygen in their chemical structure. And the different examples are like beta carotene, alpha carotene, and then xanthophylls are like lutein, zeaxanthin, fucoxanthin, etc. And the main important point of these carotenes is that they serve as provitamin A, which get converted into vitamin A after consumption. So you can see over here the various pigments that we have produced. So the first, it is dominated by the so by the chlorophyll pigment, whereas after incubating them under the controlled conditions, we have the pigment production. So these are two different kinds of pigments. One are the carotenoids and the other one is the xanthophyll. And these are the different organisms that we have used for the production of the pigment under controlled conditions. And the next pigment are the phycobiliproteins, which are water soluble, and they are of three types: phycoerythrin, phycocyanin, and allophycocyanin. So you can see the pigments over here and the organisms from which we have got them. So mostly it is cyanobacteria, red algae, and cryptomonads. So for optimizing the pigment production, we should always know the pathway. So here, for example, I'm showing the fucoxanthin pathway. So it goes through various stages and then it enters this cycle, violoxanthin cycle, and then the final cycle where you can get the fucoxanthin. So this is the diadinoxanthin is the precursor of fucoxanthin. And if you supply it with the low light intensity, you will get the fucoxanthin, because fucoxanthin is a photosynthetic pigment and it gets produced more under low light. Whereas if you subject it is to high light intensity, you get the diatoxanthin. So that is not the pigment of our interest. So always, if you want the fucoxanthin production, the low light is the best option. So here we have used diatom. So it settles on its own. And so we don't, it is an economical process. Then we have extracted it in the solvent and dried it and subjected it to silica gel chromatography. So we have extracted, eluted the different kinds of pigments that were present. So you can see here chlorophyll A, chlorophyll C, fucoxanthin, and diatoxanthin. Then we have evaporated it and subjected it to HPLC analysis to know the concentration, and LCMS analysis to know the structure, purity, mass, with NMR. And this is for going for bulk production. So this is another kind of pigment which we have grown to up to 60 liters. And but this particular species, it could not be settled on its own. So we have used some coagulants, and then we have separated out the biomass, air dried it, and we have got the powder that is rich in the particular pigment. So now if you want to go for the industrial scale production, either photobioreactors are used or the open raceway ponds. Now what is the problem with raceway ponds is that you will get a huge biomass, but the problem of contamination persists because a lot of protozoans infect these microalgae, and that can cause a culture crash. Whereas the photobioreactors, they are closed, and you can avoid the contamination and get a good pigment yield, and also you can control the various conditions. Now what are the benefits of these pigments? So there are various benefits, like they can serve as antioxidants, they can also help in the skin health, eye health, where like lutein and zeaxanthin are the two pigments that filter out the blue light so as to protect the eyes. Then also the cardiovascular health and detoxification, then anti-inflammatory effects, and also the immune system support. Now they can also help in the aquaculture industry, where they can improve the pigmentation of the fish and the crustaceans. So they are more appealing to the consumers. They can boost the immunity and also improve the reproductive health of the fish and serve as an alternative for the synthetic additives. So about the market value of these pigments, we can see that in 2010, so these are all the different pigments obtained from microalgae. The market value is around 250 million US dollars. But then in 2023, it has almost doubled. And you see that it is around the selling price around 300 to 1,500 per kg. And in 2010, actually fucoxanthin was nowhere there, but recently this is into the market, and it costs around 40,000 to 80,000 USD per kg. So now what are the latest advancements or the bottlenecks? So we have to advance the photobioreactor designs so that we can improve the light utilization. On also the biomass has to be increased, and so that we get a better yield of the pigment. Then also regarding the co-cultivation system, so we usually think that the microalgae has to be made exenic, but it is not so, because the bacteria helps the microalgae in the production of the various pigments. And also we have to follow the omics technologies so that we can better understand these organisms and engineer certain strains that have a better pigment yield and also which are more stress tolerant. Now also we can use the advanced extraction techniques like the pulsed electric field, ultrasound assisted, and also the enzyme assisted, because some of the cells, they are very hard, like diatoms, they have a silica wall, even though they are rich in the pigments, but they have to be broken open. And also in the carbon capture integration, whereas as I told you all, the microalgae, they utilize the CO2, so this can be combined with the industrial process where they release the CO2, so that will help in improving the biomass as well as improving the reducing the greenhouse gas release. Yeah, that's all.
Thank you, ma'am. Thank you so much. In the interest of time, we will take one question. Very structured, no subsections, one question only.
Hello, myself Subindu.
Yeah.
I think you can recognize me.
Yes,
We
yes.
met last time in IO. So, ma'am, my question is that how this technique means co-culture or co-cultivation of different kind of seaweeds. It could be possible to maintain those kind of culture in laboratory conditions for the seaweed for the beneficiary of seaweed cultivation what kind of approach we can take if we try to?
For seaweeds.
Yeah. Seaweed.
Because always the bacteria will be there with the seaweeds. They will be attached. Even you will get the microalgae attached to the seaweeds.
Yeah.
So that is the co-culture, because the bacteria, they produce certain cofactors like vitamins, which help the host to produce the particular value added compounds.
Thank you.
Thank you, Dr. Smita, for this
Thank you.
interesting talk. So we are just in the same corridor and we work together. It's nice to listen from her. Any questions from the online participants? We get back to the seaweed business again now. And we have Dr. Kirti Mishra. She's not very new. She's the CEO and a women entrepreneur of Brahma Kamal, where they grow seaweeds and then in a sustainable way directly to the coastal community. And today, and she integrates R&D and then training programs for the coastal communities for commercial and in a remunerative way. She's trying to cultivate seaweeds along the Odisha coast. And today in her talk, she'll be expressing the challenges that she's facing and whatever options and suggestions we can give, she's welcome to this event. Thank you, Dr.
Thank you. A very warm good afternoon, everyone. First of all, I would like to thank the Director ILS, Dr. Das, and the Senior Scientist, Dr. Sahu, for inviting me to this noble platform. So actually the topic for my topic was seaweed cultivation and challenges. So I did a little modification to that by adding this rationale of going for seaweed cultivation, particularly in the state of Odisha and the country as well, and the current status from where we are going to start, and the necessity of doing it, and what is the importance of doing it in context of not only Odisha state but also the country and globe as well. So before going deep into the subject that has been given to me, I would like to invite your attention to few slides which will give you a background that how seaweed cultivation has become important in our in context of our country and state as compared to the global scenario. So these slides I have taken from the presentations of the then Secretary of Fisheries Department, Government of India, Sri Rajiv Ranjan. Why I am saying so, it is most authentic thing which I would like to present, and where we are now we can estimate ourselves. So if we can see the data, the table, right side table, India is contributing only 0.02% of the total to the total global production, whereas the other Asian countries led by China is they are producing almost 94% of the total production, global production. So there is high regional imbalance in seaweed production across the globe. And coming to this slide, this is showing that seaweed locations in India, and we can very well see that where Odisha stands, and having such a long coastline of 480 kilometers, and with recent addition of some bays and other areas, it is going to be now almost 522 kilometers stretch of coastline where we stand today. So there are a lot of scope, as this slide says, and this is a very important slide which shows how, there is already the directive of Government of India and support of Government of India, and the guideline is already there. It shows a very clear year-wise action plan and a very well-defined roadmap where year-wise target has been fixed for the country for different regions of the maritime states. So there we can see that Odisha has been given a target of producing 1 lakh ton of seaweed by the end of 2025, and with a budget allocation of 57.14 crore. So the next slide is more interesting to see that by 2021, many maritime states have submitted proposal to Government of India, and still there has been no proposal from the state of Odisha. So this data really prompted us to, I will say rather validated our concept to work on seaweed in the state of Odisha, as we found that after so many years it has been remaining so unexplored. So, and there is so much of government support is there and motivation is there from the government side. So it will be a nice field to start the cultivation in the state and bring the revolution in the state, as we have 9 districts having coastline, vast stretch of coastline, and people are normally depending on none other thing other than the fishing activity, and which is also giving them a very poor income. So coming to about us, we are Brahma Kamal Research LLP. It's a startup and focusing on seaweed cultivation and its value chain development in the state of Odisha, and with a plan of providing additional livelihood option for the rural coastal community. So we work with that objective, and our product lines we plan that is biostimulant and hydrocolloids and the animal feed supplements. So the problem statement what we see to start our work is the large coastline remaining underutilized. We see that people go empty-handed to the coastal waters and come with some fish, and which is a very give them a very irregular kind of income and catch. So most of them are leaving their villages and their station, and they are going Tamil Nadu and other states in search of livelihood. And the other one is the dwindling livelihoods from fishing, what I said. Then there is always a demand supply mismatch in seaweed, as we have been discussing for last 2 days. And impact of climate change is very pertinent for a long time, quite a long time. And this loss of natural carbon sequestration due to fossil fuel and other things, and the mass deforestation that can be addressed by the seaweed cultivation. With these problem statements in mind, we have started working, and the solution we propose is that to create a sustainable ecosystem where the coastal fishermen and the farmers, they will cultivate seaweed, and they will be organized to form groups or organizations so that they can take the responsibility of not only aggregating the seaweed but also have a semi kind of processing at their level, so that they will feel more empowered. And we will be simultaneously having factory set up for the development of seaweed derived product, so that the whole chain will be sustainable. And in the process, there is a plan that we will be providing them training and giving them inputs to take up the cultivation, and there will be 100% buyback things from them. And then we have a plan of producing, commercializing the seaweed-based biostimulants, animal feed, etc., by having contacts with different companies. So it is broadly, I can say that we dream of having a Amul kind of thing in seaweed in the state of Odisha. And uniqueness is that it is a first kind of initiative by our startup, and before that nobody has taken up. So along with this, also the socio-economic impact associated with seaweed cultivation and its value chain development is known to all of us. And opportunities we find for seaweed cultivation in the state is that the market potential for seaweed and seaweed derived products is very high, and there is always a gap in supply and demand, and mostly we are depending on the imports. And vast coastline with inlets, bays, and estuaries, for which Odisha is very blessed in terms of having all these things. And identified sites, as I was astonished to see that it's a CM4I data that says that around 26,000 hectares in Odisha, with an estimated huge 254,000 dry tons of seaweed yearly. If we take the biomass of seaweed 10% of the wet biomass, then we can imagine the huge potential Odisha is having as per the CMFRI data, which would translate into a market opportunity of US dollar 95 million US dollar. And the other factor is a strong desire and drive for economic empowerment in the state and need for additional livelihood option for the coastal community and the rural communities, which is always there. And most importantly, a conducive ecosystem and government support, and which I will attribute to the present, the organization of this present workshop and the leadership being taken up by ILS in this direction to address the research gaps in the subject. So all these are the beneficial points and making a very good ecosystem in the state, which was not before. So going to seaweed cultivation, as we all know that there is already a paradigm shift in the approach. Previously, it was mostly depending on the cultivation from the natural bed, and which has its own negative implications, and which has been now changed to a cultivation mode. So benefits, as we all know, have been discussing, that is increased production and import substitution by adding more seaweed biomass to the country's load, biomass load. So we can import substitution in that way. Then sustainability is another thing which is coming under benefits, and controlled growth condition, which is always there when we go for cultivation of seaweed, and year-round availability, which is not there for when we go for a collection from a natural bed. And employment generation is the most important opportunity we get from seaweed farming. And farming innovation, there is always scope, as I have been seeing different innovative methods taken up by different regions, different areas, and different organizations for making the seaweed cultivation suitable, best suitable, befitting to their area. And reduced environmental impact, it is always there associated with seaweed cultivation. So coming to the different methods of cultivation, so there are various methods. These are the common methods, and this may be a redundant information to all of you as you have been seeing it. So floating raft method, tubular method, long line method, net bag method, polypropylene net method, and single row floating method. It depends on the type of species we are going to cultivate and the nature of water body where we are going to cultivate. So seaweed species and cultivation methods, you notice what we do. We are growing Capaficus alvarezi and also Gracilaria species, and mostly edulis. We are also trying to culture adapt the Gracilaria verrucosa, which is natural to Chilca, and simultaneously also we are having a cultivation system for Gracilaria salicornia. So we know the commercial importance of this species, and methods of cultivation are floating bamboo raft method and monoline method. Long line methods are followed for K. alvarezi cultivation, and we follow a tube net method for all Gracilaria species in our system. So process of seeding, as most of you may be knowing, just a pictorial form I have tried to give you an idea. So how the seed materials are put inside the net bags and how the is done before it is floated in water. Then process of seeding, this is the monoline method, how the bunch looks, and to get a bunch like this, what are the processes stepwise. And this is all these photographs in our natural field site from where we have collected these photographs. And the process of seeding in a bamboo raft method, that is preparation of raft and floating of raft, and again seeding of the seed materials in the raft, and harvesting and reseeding out of that harvest. All of this we are doing. So in this slide, I wanted to show how it looked like a cultivation before seeding. I mean, during before planting in water body, and during harvest, how it looks. And it is in Odisha waters, this kind of growth we are getting, both for tubular net method and also in bamboo net method, bamboo raft method. So somebody was asking about the economics yesterday. So this is a kind of economics we get out of our 2.5 years of experience in the field. So this we calculate that per season per raft a person is getting around 850 rupees, and we calculate, and this 850 rupees is not enough to to say farmer and to motivate him to join for cultivation. So we have calculated in that way that one person who is having maintaining 20 rafts, he is going to get around 10 to 12,000 per month. So in that way, we motivate the farmers, and we already have done 2 rounds of reseeding, and we have also started paying to the farmers. And we are happy to see that the interest is coming from other persons in the field, which was not there before 1 year. So quality analysis we did at CSM CRI for our Capophycus alvarezii sap and the residue, dried residue, and it was found that it is as good as its normal counterpart what we received from Mandapam, even better than that in terms of gel quantity and other nutrient and other biostimulant contents. So recently we have purchased the technology from CSM CRI for production of biostimulants out of Capophycus. So another small experiment we did, we have done in collaboration with Odisha University of Agriculture and Technology with their seed technology division. So by doing a simple priming assay as compared to a normal hydro priming and as compared to a positive control by taking a biostimulant available, seaweed biostimulant available in local market. So the result is very encouraging, and they, as per their result, they have got very encouraging result from Gracilaria verrucosa extract, which gave the best result, producing highest average seed yield of 47.64 quintals per hectare, which was 36.3% higher than control. Over control and control, here we have taken a very standard seaweed based biostimulant available in the local market. So Capophycus alvarezii also extract also gave a good commercial yield, which were 26.4% and 22.8% higher respectively over the control. So 4% concentration was found to be giving the best result out of the all three trials tried in triplicate. So what are the common challenges we encounter during our seaweed farming? And we have classified that that is natural, that is prone to natural calamity, failure of monsoon, sometime extended monsoon, environmental factors, which are very common in Odisha coast. Then ecological, which is seasonal impact, vigor loss due to seasonal impact, and biological, of course, it is grazing disease and epiphytes, as we have been discussing since yesterday. And there are few social factors which is also very difficult, posing very difficult challenge for us. Of course, we are addressing it and getting good result out of our the approach of addressing the problem. Then middleman interference in local conditions is really intervening in the process, and gradually we are trying to sort it out, and we are also getting good response from the community over a period of time. Then variations in growth we found in different seasons, as my previous speakers have been showing it, and it gives me a kind of relief in one sense and validation in another sense that whatever problems we are finding in our coast, that is not very, I mean, different kind of challenge we are facing, because always we are apprehending that we are doing it in a compromised condition. We are not able to enter into the open sea. So we are using the bar mouth areas where river enter the sea, and we get a sea-like condition for 6 months over there. We are doing the cultivation there. So whatever challenges we are facing during cultivation, we feel that we may not, would not be facing when we would have gone to sea. But I got that idea that in all presentations related to cultivation, the similar kind of challenge they are facing. And the one which impressed me during last presentation of Dr. Sri Kumarji, that how the chlorophyll content and the temperature, they are interdependent, and when both of them coincide to give a good culture, a very luxuriant cultures. At that time, epiphytes and other thing, grazing issues, how it affects. So throughout the year, there are risk of challenges, and I have summarized few. So healthy growth with good pigmentation observed during winter, January to March. It is so impressive that we have shown all the premier institutes who are dealing with this seaweed, CSMCRI, CMFRI, they are very impressed by seeing the luster and color of the thing. That means Odisha water is supporting it, supporting its growth. So good growth, what change in pigmentation observed during early summer. We see a change in pigmentation. Early summer in April shows crop loss due to grazing and sudden rise in temperature. And again, I will see after seeing the presentation of Sri Kumarji yesterday that we were thinking whether jellyfish could be a problem, because local people every time they face that challenge, they attribute it to the population of large population of jellyfish coming into the cultivation site. But yesterday I saw that yes, it is a problem. Jellyfish population, rise in jellyfish population is also a challenge to the seaweed cultivation. That we faced during end of March and middle of April till mid of April, and after that that spell goes away. Then stunted growth is observed during late summer, that is towards June and July. We find the color is there, the pigment is there, the main meristem is very hard, growing very well, very fleshy, but the secondary meristems are very showing very stunted growth. That is typical in Chilka area and also in Jatadhar area. So that is also one of the very very close observations we have made. And excessive monsoon rainfall causing the salinity dilution, as we are working in the sea mouth areas where river enter the sea, so that entire track becomes completely fresh water. Almost the salinity goes down to as low as 0 to 4 it remains entire months of from mid of August till November. So inadequate rainfall during monsoon sometimes extends the post monsoon rain, which lowers the salinity till November. So even though we try to have restart the cultivation in the month of November after 15th, then also we find the same problem. The low salinity condition persists, which makes a crop loss. So these are few photographs I have given. And another observation I would like to add here, as experienced persons are here, when we are getting very good growth and luxuriant growth, particularly with long line method. So with the weight of the biomass, that long line takes a concave structure. In that condition, the middle of the things, bunches, they almost go down, they come to the close proximity of the ground. So that's why in that case, we observe a kind of pigmentation growing from the lower end, distal end. So that is also a problem we are facing. And that's also, of course, a operational thing that we are taking care, and also we are getting good result out of our remedial measures being taken by us. So biological challenge, that is epiphytes grazing a disease, as we already have discussed and we all know. So grazing is a major threat. These fish species are really threat to the problem, even though we put a fish net below the things, but we are constantly taking other measures to keep our cultivation away from the fish grazing. Then herbivorous fish, crabs, snail or sea urchin are the most likely creatures we find inside our cultivation site and farm site. Capaphycus is more sensitive to jellyfish infestation. We saw in both the years, last year and this year also, in all cultivation sites, the similar observation is coming. And microalgae population over our seaweed cultivation is really a hindrance, which also grows during that particular salinity and temperature condition. And so for regular cleaning is a problem, even though we sensitize our farmers to do that. And you may be knowing, so there are lack of that commitment sometimes witnessed in field. So that is a challenge for us. Then change in salinity, high temperature and irradiation, particularly in Odisha. Even though in the month of April and early part of May, there is heat is so much, and the light intensity. that also affects the seaweed cultivation, lowering its pigments, intensity of pigments. Other challenge, that is heavy tidal action, prevents us to enter into open sea cultivation, and which we want to do, but it's really a problem. For that we need help from the state government, and freshwater influx during monsoon in the bar mouth area. So high heat and irradiation affects growth and acts against pigments. Seed crisis to pursue the activity after monsoon. It's a really a challenge, because we again, for restarting the whole thing, we have to take the seed material from Tamil Nadu, and it will take a longer time for acclimatization. In that way we lost another 1 month in that. So if we could have a seed preservation facility in our state, in our marine system, that will be really a help for. We are, from our personal point, we are exploring at different port sites, requesting them to lend us some water body so that we can keep our thing during monsoon time, but that is not working. But still we are trying for that. And no maritime policy yet yet in the state for area allocation, though we have been visiting fisheries office. So due to, though we have been said, we have submitted an application under PMMSY also, but we have been the given received a comment that we have to give a CAA approval for that. But there should be equally the initiative from state side to give a clearance, at least facilitate the things to get us a clearance. So whatever is planned, that the purpose of saying is that whatever directive and guideline is there from the highest level, but that very poorly percolates at the lower level where the things happen. So ignorance on seaweed farming, we strong believe that affect the fishing resource. It is one of the phenomenon we have been witnessing with some groups, and we are trying to address that and sensitize them through various meetings and other things. So support we require. I was expecting, as Sasikant sir was there yesterday, so I was expecting from the state government, as he will be here, so this will give him some insight. So how a seaweed farmer entrepreneur can go ahead with the state government's help. So these are few things I have noted. Clear state policy for seaweed cultivation, provision of monsoon storage site for seed materials around 2 tons to be stored for 4 months, at least that provision could have been made. State support for initiating seed bank and elite germplasm production facility. So as I was going through, there is a press release by Government of India just 7 days back that there is the states like Tamil Nadu, Dadra Nagar Haveli and Lakshadweep are going to have seaweed seed production facility, and that's a good thing, and we hope that Odisha government will also pursue in that direction. And development of biomolecular probe, as the research community is here, and I have been listening to the various advanced researches being conducted and being planned for the next couple of years or decades. So this will be a good thing if biomolecular probe can be developed, as we are using for human diseases, for monitoring the crop health and preempting the disease condition in plants. That will be a good idea, what I think. And support for combining seaweed with shrimp on land-based cultivation and IMTA framework. We have been taking initiative in that direction, because these 4 months when there is a break in cultivation of seaweed, we want to have, and we are also working in that direction, to have some low saline crop or an IMTA system to have a shrimp and seaweed at a common facility to be grown, so that the farmers, they will get their engagement during those 4 months. Then create awareness on seaweed application and importance of farming. That will help us to go to the community and get better community support. So importance of seaweed seed bank facility, it is all there. I just wanted to put it in graphically that if we have a seed bank facility here, so there will be no interruption, and if there is any crop loss or monsoon break, which is very obvious in a state like Odisha, and as I saw also in other states, so having a seed bank facility will be big support for the seaweed entrepreneur and the farmer. So this is our journey. We have incubated our startup in KIT TBI. So we have taken training from CSM CRI, and this is our farm site. Our farmers are engaged in cultivating seaweed, and they are so enthusiastic, and it is becoming really difficult to convince them that after 2 months there will be a break in the cultivation due to the monsoon. And this is our journey. Some more photographs from the field, how we are harvesting and reseeding, engaged in the process of doing the things. And I will try to place this. Very interesting and very soothing to hear this sound, your seaweed raft, floating in water so happily. And this is another thing from the directly field side. This is from the farm side of Jotadhar, where farmers are engaged in cultivation. Thank you so much.
I think she deserves a very big round of applause. Hats off to you, Dr. Kirti Mishra. So if we have any quick question over the lunch, any online participant wants any question? Only I will suggest, if are you measuring DGR?
Yes,
The growth rate,
yes,
you're
yes, for
measuring?
both the
Okay,
spaces.
both the spaces
Yes.
you're measuring.
We are doing it weekly and
Okay.
previously fortnightly, but because people were not very confident of taking it out, they were not
every
knowing the
time.
process, because finally we are dealing with the people who are not having that technical things. But now we are doing it on weekly basis and converting it to get an easier.
Yes.
Madam, you said you had some problems with jellyfish. How are the
Yes.
farmers handling the jellyfish? Don't they get stung when they go to work with the seaweeds?
Actually, they are, I will say that they are the experts of water, system, water body. When I experienced it, when I took them to Mandapam CSM center, they certified that our farmers, we are having the best set of farmers, they can be the master trainers. And in terms of their experience in working a long time in water, they can differentiate two types of jellyfish. They say that when they get a red a jellyfish of having red color, they get itching in their body, and they say that that is also majorly affecting the cultivation. And for that the steps they are taking, like putting fence around the things, but as we have now shifted from bamboo raft to the long line culture, and that too again as per the recommendation of the farmers, because they find it more convenient and more less time consuming. So in that case, when keeping a fence around such a long line of 80 feet and 100 feet, it is really difficult in area. So that is really giving us a big challenge, and currently we are going through that phase in two of our sites. Even I was taking information from other fishing side. Fishing is also being hampered by the overpopulation of this particular species of jellyfish.
Thank you, ma'am.
Thank you.
Thank you, ma'am. Thank you so much.
Are would it be possible for you or your teammate to visit the Brahmakamal sites and see where technological intervention can be made to make the farmer's life bit easier, safety, and more
Absolutely.
productive?
We will be more than happy to do that. We'll coordinate with madam and we'll try to do that.
Yes.
Okay, thank you.
All our
Thank you.
farmers are very well oriented, and you will feel very happy to meet them, how enthusiastic they are for this.
Thank
Sure,
you, ma'am.
thank you.
Thank you so much. We will break for lunch right now, but before that, please join us for a group photo right here. We will reconvene exactly at 2:25. Please don't be late because we have to start by 2:30. So online participants, can you please all switch on your cameras? So we have a collage.
Online participants, please switch on your video if you want to be in the photograph. Come, quick.
Good afternoon, everyone. Before we start our next session, I would request Director Sir Dr. Debashish Das to felicitate the speakers of the last session. So, first of all, Professor Valsama Joseph, ma'am. Dr. Shams Yazdani. Dr. Smita Midbhavkar. And last but not the least, Dr. Kirti Mishra. जी सर, थैंक यू सर.
सीवीड का बायोस्टिमुलेंट चाहिए स्टूडेंट लोगों को, अपने दोस्तों को बोलो जल्दी से खाना खाते आ जाओ ऑन व्हाट्सएप और व्हाटएवर मीडिया का है.
Yeah. Are, if you can introduce the speaker, yeah, please.
All right. Thank you. Good morning, Folka. Welcome. Nice to see you. Just a short introduction to Folka. Dr. Folka Sieber is the head of department of the Department of Chemistry of Biogenic Resources at the Technical University of Munich. And he's also head of the Fraunhofer Institute of Interfacial Engineering and Biotechnology, Bioelectron Chemocatalysis at the Straubing branch. So he's super versatile. He knows everything from electrochemistry to enzyme catalysis to biology to fermentation to all kinds of stuff. He did one of his, I guess he did a postdoc or a research stint at Professor Frances Arnold's lab at Caltech. She was the first chemical engineer to get a Nobel Prize in chemistry for directed evolution. So we have an expert in directed evolution and enzyme catalysis and everything. I hand over to you, Folker. You can go ahead and give your talk now.
Yes, thank you for the kind introduction. I'm still struggling with what you told me. I would have to set here the picture-in-picture.
Yeah, you can go to menu and click on more settings.
There's the menu. Okay.
Yeah,
Yes, I see. Okay.
And then disable the picture-in-picture while sending screen share. Then you can share your screen.
I don't have that. Well, I start sharing the screen before maybe that will work.
Yeah.
So
There we go.
do I still have to do anything, or is that okay?
This is okay. You can start and you can continue. Yeah.
Okay, so yeah, thanks for the introduction, and I'm happy to share some of the work that we do to give you insights of what you could do with seaweeds. And so generally there are many different types of seaweeds, and you've heard a lot of possibilities of growing them in the conference yesterday and today. I'm looking at the polysaccharides that you can produce with seaweeds, and there are different types. Today I will concentrate on the red seaweeds, and here specifically on the carrageenan-bearing seaweeds. And if there's some time, I will also shortly talk about alginate, but the focus of the talk will be on carrageenan, with the different seaweeds that you have producing different types of carrageenan. And I don't know whether you had this as a topic, but I'll start now with what carrageenans there are out there, which are important. So first of all, there's a kappa carrageenan, which forms very strong stiff gels if you put potassium in there. Can also form gels with calcium, but they're a bit more brittle, and that's the main application. Chemically, the carrageenan has in one repeating unit one sulfate and also this anhydro galactose bridge. And kappa carrageenan is very, very, I think, the most important carrageenan that's out there in the market. Then there's iota carrageenan, which forms more elastic gels, clear gels, quite nice application also. It differs to cover that it has an additional sulfate group here in this repeating unit. And finally, the third commercial one, that's lambda, that's lambda carrageenan, which has three sulfates, this one here, and now the red is missing, so here's another sulfate, and the third sulfate here, and there's no cross, no bridge here, and this is just for forming high viscosity, no gel formation. And now if you look at the different seaweeds that are out there, that grow in different regions of the world, they produce different types of carrageenans and very often mixtures of carrageenans. So Yoshima, which is what you have more in the tropics, yields typically kappa and iota, but sometimes also mixtures. And then there are in other world regions, Gargachana or Kandros, which have then mixtures of kappa lambda and iota, or Kandros just kappa and lambda. And so one question that is then possible to be raised, whether you can interconvert these different carrageenans into each other. And what we were looking at, so if you now look at the applications that you have, some are very important for kappa, others iota is better, whether you can modify the sulfate pattern and by that maybe create here new applications or new properties for new applications, but also maybe interconvert any of these into each other. And by that, starting from mixtures of carrageenans, have just one type of carrageenan left. And yeah, this is what we were looking at. And to achieve this, what you do have is modify the sulfate. So release sulfate here or this sulfate here, and then get new carrageenans. And to release sulfate from the polymer, you can use strong acids or base, but typically this would cleave the glycosidic bonds and you would depolymerize your polymer chain. So chemical means of removing sulfate are not very good, and that means enzymatic method that are more mild might be the better choice. And this is why we were looking here for enzymes, sulfatases specifically, that would release the sulfate and then create new polymers. And these sulfatases, the widely distributed class of enzymes, they are very specific, chemically specific in the mechanism. I'm not going to detail here. It's just that they have a very unusual active sites, and you need specific organisms to produce these sulfatases, but we created these organisms, and so we were able to produce these sulfatases enzymes recombinantly and apply them for this reaction. However, not many sulfatases were known to do this. And so what we first did was looking at environmental samples that we got from marine isolates, whether they would produce any of these enzymes. And what we were looking for the release of sulfate. We have a very fast detection method, a colorimetric detection method. So we have a green color formation if there is a sulfate released. And so we identified some enzymes that would release sulfate from iota and also from kappa. And so we finally also did some genome screening and identified a few sulfatases that are really active on the polymers. And that is a very important part. So we're talking about release from polymers, because we do not want to degrade for this application the carrageenan. These enzymes will then purified and applied, and what you can see here, so basically from iota carrageenan you release this sulfate group here, and you create an alpha carrageenan. So this is also something that is appearing slightly in nature. It's not very common in nature, if at all, but we can create this. And this is just a IR spectrum to see that here the sulfate is gone. And the same sulfatase is acting on kappa carrageenan and makes a beta carrageenan, which is a sulfate-free carrageenan. It's almost like an agarose. And again, you see that here this specific peak is reduced. It's not completely gone, but reduced. I'll come to this. So activity is there, and then we did apply this to commercial carrageenans, and what you see is you get first an iota alpha hybrid, because the enzyme first acts in making just releasing every second sulfate, and only if you wait it longer you get to the alpha carrageenan. And the same here, you can create a kappa beta hybrid, and then the beta carrageenan is sulfate-free. So 4 new types of carrageenans that you can then see whether it has a good property for certain applications. And so certainly if you would go for high gelling ability, alpha carrageenan is not the product of choice that you want to produce, because so we created then these different hybrids having different amounts of alpha, different amounts still of iota, and you see this linear relationship between the gel formation. This is basically what's shown here, and the concentration or the content of alpha. So you completely lose here the gel formation. However, there will be possible applications for a sulfate containing non-viscous polymer. So if you need that, then this is your product to go. On the other hand, if you start with carrageenan and you go to beta, you see this linear increase in gel formation, the more beta you have. And that is a specific experiment where you have a low metal content, so not very much of the calcium or potassium here to help the gel formation, and so with beta you still have gel formation. Now this point here actually is not 100% kappa carrageenan, because it was not beta carrageenan, because it's not possible to create 100% beta. And this is because then gel formation was too strong. This point is actually agarose. And you see that you can now produce an agarose type polymer from carrageenan, and same linear relationship here. We looked at different properties of the gels, the different hybrids also. So you have here some rheological characteristics, storage modulus G prime, loss modulus G2 prime. I'm not dwelling on this, and here the different dependence on the salt concentration. And yeah, you see that with low salt, the beta is much better here than the kappa. And also, again, here shown now the concentration dependence that you can, with beta, get the same gel formation with not even half the amount compared to kappa. So you can have here much less of the carrageenan you can save basically on the product in the application by applying beta compared to kappa. We were also looking at the production of these modified carrageenans, and you can imagine different forms. One is that you first extract your carrageenan, you prepare it as we did in our experiments, and then from this apply your enzymatic process to get your modified carrageenan. So this is what I've shown before, but this is production-wise maybe not the most efficient route. So one way would be to apply the enzymes on the washed and dried seaweeds, have the transformation on these, and then do the extraction later. And well, if you do not want to have here the alkaline extractions, the alkaline modification, which is sometimes necessary to form here this specific anhydrous sugar, then another option would be to just apply the sulfatase on the washed and dried biomass. And so what I showed you before, if you now use the kappa fugus and do the extraction and then apply this, you get the beta carrageenan. We did that. We also applied here the enzyme as I described on the seaweeds before the alkaline modification. And since there is a lot of hybrid beta and mu carrageenan and the enzymes will not be active on the mu, you get a beta kappa carrageenan hybrid specifically if you want to apply that. So different results here. And yes, we did this, and both approaches were basically successful. So you see that the blue curves here are above the red curve. So you see that again, gel formation much stronger when you apply here the sulfatase. And we then also applied this to different seaweeds that we obtained from Alan Critchley, who might be he's also part of this conference I saw and partner that he organized for us. And so indeed here with these seaweeds direct application on these different forms, we were then able to obtain these different types of carrageenans. And yeah, again, you see here, if you work with the carrageenan containing ones, also having mixture of lambda here, you have this increase in the blue curve above the red curve in gel strength. If you just work with iota carrageenan containing seaweeds, you have this reduction here for this very still sulfate containing but low viscous polymer. Okay, now you can also see here on the picture, if you apply the sulfatase as gel, no sulfatase, it's liquid. and also you see that the melting temperature and trend temperatures are increased if you introduce here the sulfate into the system, the sulfatase, the enzyme. Okay, so to finish the first part of my lecture, I think the sulfatases and the application do offer here an opportunity to achieve modified carcinogens, and by that have further ways of commercial utilization of the carcinogen that you have. And now, last 10 minutes, I'd like to use to talk about another approach of utilizing seaweeds, and that is production of chemical building blocks, and specifically complete degradation to obtain here this 3,6-anhydro-D-galactose, a very rare sugar with bioactive properties. It has anti-inflammatory and skin whitening and carcinogenic property, it is said, but getting this molecule in high amounts is not very easy. And again, you see that starting from the sulfatated kappa and iota polymers, you do have to use here some sulfate-releasing enzymes. But in this case, the application is not necessarily on the polymers, but can also be on the oligomers. And so this is basically what you need to do. So you start out here with the polymers, you degrade them with an hydrolase that will make oligomers. And then you can apply different sulfatases to get to this basically a beta type carrageenose or neocarrageen, basically oligomers. And so you need hydrolases here, glycosylases, and sulfatases. Here an enzyme, some enzymes are known. We work with one that is known to break down both polymers. But we were looking again at the sulfatases, and in the first part I was looking at sulfatases that would act on the polymers. Now it's some that has to act on glucosylsaccharides. And coming from here, you need another enzyme that would then take off here the anhydrogalactose, and yet still a galactosidase that would take off the galactose that you can basically chew yourself away and release these polymers. So this enzyme also had to be identified, this glycosidase that releases this rare sugar. And we looked here at different bacteria that have these polysaccharide utilization locus. Many genes, every single arrow here is a gene, are in their genome. And we use this to identify here the right or possible enzymes. And this is just to show with sulfatases it's possible. You get activity here of many different ones on polymers, but some also on the oligomers. And the same for kappa to beta, so more active on oligomers than some on polymers. And so this basically is the first time that this desulfation from iota to kappa neocarriose could be shown. So activity on iota going to the kappa in this case. And so what was certainly important is to measure here the sulfate release, and you see some of the enzymes are very active. However, the most important question is whether you do actually produce the anhydrous sugar with these sulfatases. And here, in the combination of different enzymes, then different sulfatases were actually better. The CA and EP here, we characterized them, showed that they are applicable. This has good salt tolerance. This has a high thermostability. Could be applied at higher temperatures, which is certainly helpful. And then we were able to apply these into the cascade for the degradation of iota carrageenan as well as beta carrageenan, as well as several different hybrids. Even phosphorylated can be used here as a substrate. And so this basically is a one-pot reaction, all the enzymes together. and DA can be released, and yields is about 50% or 30% to what is theoretical yield would be. So since you do release also sulfate and you also release galactose, so the complete possible yield is somewhere in the range of maybe 25 or so percent. So well, not that bad. One challenge certainly is that when you want to apply the enzymes at this temperature, then you have some gelling formation already, and you cannot increase the concentration of the substrate very much. So we are now working on getting here higher concentration, by that having the higher concentration also of the released monomer to make a chemical process out of it, and then have a scale up here. That was what I want to talk about carrageenan. There's one maybe very short, since the time is running out. This alginates, how you can apply enzymes there. Alginates have different polymers and different monomers, and still form gels here with specific types of within the more polymer, and that is shown actually on the next slide. You have here guluronic and manuronic acids, and only the guluronic acid allows formation of the gels. And with enzymes, you can actually interconvert manuronic into guluronic acid, changing the position of this part here, moving it from up to down. And by that, the sugar will reform and will make then the gel formation possible. And these are enzymes that can be also produced. So you produce them, and when you look at different algae, different seaweeds, you see that they contain different amounts of G. So if and this is what you need for the formation of the polymer of the gels. So if you, for example, have here parts of the seaweed which have a very high M type, with the enzymatic treatment, you can increase the G type and then allow them to form gels. This is also something that you could do, and I could talk about this maybe another time in our work here. Finally, a bit of a different issue. I'd like to shortly introduce an association that we have formed with different institutes and universities worldwide, Global Bioeconomy Alliance. That is an association to increase cooperation, and by that create a higher impact and accelerate the research to commercialization, because we do work together with many different companies in this alliance. And one of the focus areas, one of many here, is also agriculture, marine bioeconomy. So if you are interested, you can look here at the webpage. And with that, I'm through. I think I kept my 25 minutes. Most of the work that I described today was done by Alexander Fuchs, a great PhD student that I had. The seaweeds I obtained from these people, I mentioned them, and here with the sulfatases, I worked with Professor Lara Sete from Brazil. Actually, coming back to this, the whole project, the whole work on seaweeds was basically also part of this Bioeconomy Alliance work. And with that, I'm through. And if there are questions, I understand that there will be later discussion panel. Thank you.
I think we can take one or two questions.
Hello, can you hear us, Dr. Walker?
Yes, now I can hear you.
Hi, very nice work. This is Shams Yazdani from ICGB, International Center for Genetic Engineering and Biotechnology, New Delhi. A remarkable work on the enzymes. So I had a few questions on the carrageenase enzymes that you mentioned. Specifically I saw there were two glycoside hydrolase family, 16 and 127. Are these the only two family known to hydrolyze carrageenan, or this is what you found in your portfolio?
We did not done extensive research. I mean, there are so many families out there, and I don't have all. We haven't looked at all of them. So there might be some still out there in the other families. We just didn't look extensively.
Okay. Are these mainly bacterial enzymes that you looked at, or
Yes.
they're fungal?
Well, we also looked at some yeast enzymes, but we currently work with bacterial enzymes.
The bacterial enzymes, because I could see they are mostly working in the neutral range, so I could guess it's a bacterial enzyme. And I guess, yeah, sorry.
They should work in the neutral range because they work outside. At least the sulfatases, they work in the ocean basically.
Right.
So there is not much pH regulation.
Right, exactly. And if I understood correctly, you said if you treat them with sulfatases first, then the glycoside hydrolase works better. Or is there certain sequence of event that you focus on?
That's exactly what
Oh, well, that depends on the actual reaction. So in our application, we did first the treatment with the hydrolase, with the glycosylase, and then applied the sulfatase for the degradation. So the sulfatases generally work better, or we have more sulfatases that work if you have degraded the polymers.
Okay. And last question. So we're still looking for the final enzyme that makes the monomer from the oligomer, or you have a complete set of enzymes?
No, we have a complete set of enzymes. We can make the anhydrogalactose, as I've shown. We have all the enzymes, and so we produce the anhydrogalactose, we produce the galactose and the sulfate. But only the anhydrogalactose has some value. I mean, galactose you get some other way.
And what is the typical reaction time in terms of hydrolysis to get
Oh,
the?
well, this is not optimized. We typically just do it overnight. And you can certainly, with increasing the amount of enzymes, have it within a couple of hours or even faster. But this is due to optimization.
All right, great. Thank you. Thank you very much.
Professor Polka, thank you very much. My name is Sri Kumar. We've been in touch before with email, but I've not really.
I remember the name.
Yes. So my question is about anhydrogalactose, and I know that that's the most valuable thing that we can get there. But I'm just curious, have you ever come across enzymes that will break down anhydrogalactose into galactose?
No, and I don't think that nature is using this as a degradation, but it will rather degrade it differently, oxidatively, I would assume. And so you will not get galactose, but you would get sugar degradation products.
Okay, so you believe that anhydrogalactose can then be used to, it could be fermented into further ingredients if we needed to completely.
I think so. I'm not sure whether this is the best way to utilize it. But yes, certainly there will be some pathway, and I have also a kind of an image how this should work that would degrade it further and then produce things that you can make ethanol or whatever out of this.
Mm-hmm. What would be the most valuable applications of anhydrogalactose, you feel? And if you degrade carrageenan, you get one kind, a D or an L, and if you degrade agar, you get the other kind. What would be some of the most valuable applications apart from pharmaceutical and nutraceutical applications?
I would assume that's within utilizing the bioactivity of it. So this anti-inflammatory properties would probably be good, but the amount would not be very much. If you use it as this browning agent for the skin, that would be very helpful for cosmetics, and I think that the market is higher, is larger. So I would think that it might be not as expensive. You cannot sell it for such a high price, but you can sell more. So I would probably think this would be the best application.
And one last question. What it was very intriguing that you actually tried the sulfatases on the whole biomass instead of isolating the carrageenan and then trying it. So do you think apart from the carrageenan, there is significant quantities of other kind of carbohydrates that are in the biomass or related carbohydrates that may not have been structurally characterized? Did you see any difference in the rates of reaction or access? How did because in one case, the extracted carrageenan is easy to access, right? I mean, you make a solution out of it. But a biomass is typically insoluble. So how did you find the reaction rates? Did you just make a fine powder out of it, or did you?
Yes, definitely. You have to make a very fine powder of it, and the accessibility is not as good if you have a carrageenan. But if you make a powder of the seaweed, and you do have it already in an aqueous environment. I mean, you do have to get water into it. Enzymes somehow can attach to it and work with it. And the reaction certainly is slower than when you have the purified carrageenan. I can't give you now a number, but we were happy to see that it's actually working at all on this ground seaweed.
Yeah. Okay. Thank you. And these sulfatases are thermostable, right? Or
well, that's how you define thermostable. Some of them are to 60 degrees Celsius, and you can engineer them to make them more thermostable if you want to apply them at 80 or 90 degrees Celsius.
Okay. Thank you very much. Really amazing.
Professor Bholkar, fantastic talk. I'm Debashish Das, currently director of ILS, Institute of Life Science. My question is that do we know the genome sequence of various types of kappa picus, and also do we know the syntenic relationship between the genes, the cascade of genes in various types that we have?
So, I mean, many genomes are now analyzed, and it's very easy to get a genome of a new caragenolytic organism that you identify. So this is not a problem at all. Not all the genes that you see there in these PULs, in these utilization loci, are known, and so you can quickly identify an enzyme being a sulfatase, but you never know what the substrate of the sulfatase is. And so also, if you looking for the exoglycosidase that will take the DA unit of the oligomer, that's not clear at hand just from the gene. So then you have to express them and analyze them for the activity. So you always have a good guess what they do, but you don't really know the actual substrate.
Okay. Thank you.
Hello, my name is Dr. Shamim. I am a postdoc here. A wonderful talk. I just wanted to ask you simple questions, which is, as we know, the carrageenans, their sulfation levels change their chemical and physical properties, right? So instead of changing one carrageenan to other by desulfating or by adding another sulfate group, as we know that every carrageenan with different sulfation levels have different gelling properties. Why can't we create individual carrageenan as a different material for different purpose. Let's say I'm trying to make a sticky like a gum which is very sticky and a gum which is not that so sticky. Both are used for different purposes, right? So the carrageenans with different sulfation levels, let's say the kappa one which has one sulfate group, that forms a good gel with potassium ions, right? And strong gel. Then whereas the iota one, which has two sulfate groups, gives you a small soft gels in presence of divalent cations. Whereas the other one, which has the lambda one, which has three sulfate groups, does not give you a gel, but it is used in the food industry as a thickening agent. So their physical nature changes based on the sulfation levels and the physical nature, the chemical, yeah. So can't we just have a different product based on the different sulfation level that we are getting originally in the nature, instead of adding cost by making desulfating and making individual product.
Definitely, certainly yes to your question. You can mix different carrageenans and this is what's done in industry to then get the properties that you want. The question is more if you have a certain mixed carrageenan and you have a certain seaweed in a certain year with certain properties which are not right, and you want have a bigger market for a certain property and you just cannot fulfill this, and then you just change the sulfation pattern and you get new properties and you can then align your product and sell the carrageenan that you have produced. So it's just another possibility to modify the product and to basically make a standardized, pure, whatever product. And also specifically for the production of beta carrageenan, you will not get the properties of beta by mixing other carrageenans together.
So instead of you basically separate them to individual carrageenans, and then instead of trying to convert one carrageenan to other by desulfating, why can't we just separate them into as is and then
Oh,
market
you
them?
cannot separate them. You cannot, if you have a mixture of carrageenans, you cannot
That is true.
make a pure kappa or pure iota. Very often you have them on one polymer, this mixture. So though there is no way to separate them.
One more thing, just the poly. I need to shut it down. Since we know the bonding, it is beta 1,3 and beta 1,4 that changes between the carrageenans. Like, have you tried in your lab to uh, tweak the bondings between those anhydro galactoses to maybe alpha 1,6 instead of 1,4, and see what kind of carrageenans are like being synthesized and how their gelling parameters are changing based on the bondings?
Now, this would be certainly very interesting, but we have not done any work towards this.
Okay, thank you.
Thank you very much. I think we are running out of time, so excuse me. Okay, one last question.
Hi. So I am Dr. Debabrata Biswas. I'm a scientist here at Institute of Life Sciences. I think my question is really short, Dr. Seaver. So it's a fantastic presentation. My curiosity is, as a microbiologist, that you have utilized these sulfatases, the galactase, these enzymes in a recombinant form. But do you see that it can be combined into a genetic microorganism and that may be utilized? Will that make it like more portable? And
so you mean whether you can have the seaweed itself producing the sulfatase?
Or maybe some microorganism like a bacteria?
Oh, I mean, we have bacteria. We grow the sulfatases in fermenters recombinantly with bacteria.
Okay, no, I'm thinking about like if we can all the steps, different enzymes, can they be cloned into that, into a single bacterium and be utilized?
Oh, yes. yeah, you can have one single organism, and then when you grow this organism in one fermentation, have already the mixture of all the enzymes being in there. That's possible.
Thank you.
Okay, thank you, Fulka. Wonderful talk. We'll now move on to the next speaker. That
What
is
time do I have to be back? Or do I?
in probably it's about 4:45, maybe 5 p.m. India time. That would be 12:30, 1:30 your time.
Okay. 1:30 my time. Okay,
Yeah.
see you later.
Thank you.
Thank you.
Thank you.
We're running about
Pardon?
15 minutes late. So I would
Yes.
5:15 would also work.
Okay. Okay. I'll notify him before. Okay. Our next speaker is Dr. Mahashweta Saha. She's a scientist at the Plymouth Marine Labs in England. She got her PhD from the Geomar Hemholtz Center for Research in Germany, and she does a lot of work on chemical ecology of seaweeds and seaweed microbiome interactions. Over to her to give her presentation.
Hi, Ari. I need to share my slides, right?
If you can present directly. It's not a problem.
Screen.
You can share your screen directly and present as well. Not a problem.
Yeah. Just a minute. I'm new to this system, so I'm trying to figure out. Can I share the slides with you, Ari?
Sure, absolutely. And I
Because
can
this
do the
system is new
presentation.
for me.
Sure. Just email it to me and I will present it from you.
No, I think I got it. Can you see it?
Yes.
Yes.
Just move on to slideshow, please.
Can you see the next slide?
No, I can see only the editing part of PowerPoint. Maybe you want to stop sharing and start the slideshow. Yeah, from beginning. Yeah.
Yeah.
No.
I'm moving to
I
the
think you
next
need to
slide.
stop the share. You need to stop the share and then share the slideshow itself. This is the editing window of PowerPoint.
Okay.
That comes up as a different window.
Can you see now?
I can still see again only the PowerPoint. You can share the whole desktop if you can. Then everything will come through. Have you started the screen share, the slideshow?
Yes, yeah. I did the from beginning thing, and I'm moving to the next slide.
Now
Can you
what you
see
do is
something?
please stop sharing.
Yeah.
And then when you click sharing again, it'll ask for which window. At that time, choose the slideshow, not the PowerPoint editing window.
I think Do you still see the first slide?
Now we see the second slide, but it's still the editing window.
Uh,
Share the entire screen.
Yeah, share the whole desktop or the entire screen and not a particular window.
Okay, and so okay, the three options. Okay.
You start the from beginning, then you start sharing. You stop sharing now, Maha. Then click from beginning, so your slideshow starts. Then you start sharing screen.
Stop sharing. So there are three options: a Microsoft Edge tab, window, and entire screen. So
Choose the
shall I
entire
select
screen.
the
Yeah,
entire
choose the
screen?
entire
Okay.
screen. Yeah, safe.
Okay.
And then bring your presentation to the front. Yeah, go ahead. We can see your screen now. Just click on the hide button here so that the tab goes away. Yeah, thank you.
Fine?
Yeah, fine. Please go ahead.
Okay. Yeah. So yeah, sorry about the technical issues. This platform was very new to me. So thanks for the invitation. As RA mentioned, I am a marine chemical ecologist, and our group focuses on host pathogen interactions and how these interactions are chemically mediated, and we also work on the effect of biotic and abiotic factors that can control these interactions, and how different stressors like climate change induced stressors can have an impact on these associations. So the way I have structured my talk is that to give you a little bit background about what chemical ecology is before I dive into seaweed pathogen interactions and the kind of work we do. So I like explaining my science in very simple language, and I would just begin with some very basic things like the existence of language in human communication. So if we think about the importance of language, why we need it for our day-to-day life, it gives us identity, cultural background, it helps facilitate social interactions, and it also helps us to have a functional society. But the point that I want to make here is that species other than humans also talk to each other, and that's my job is about. So basically, we chemical ecologists eavesdrop communication of other organisms which are not humans. So as I mentioned, other organisms also use language for their communication, and a very simple example is penguins can find their food via detection of chemicals, which we chemical ecologists call as the language of chemicals, and albatrosses use chemical information to find food. What I've tried to show here through this infographic is that a plume of chemicals going out, and the albatross can detect an area which is highly productive, full of fishes. So speaking of language, we usually call them as infochemicals, which is the language of life, or rather communication in the sea. And also you may be already aware that infochemicals exist everywhere. It's the plant's cry for help. You may have heard about it. So many organisms do talk to each other using these chemicals. And this is a like infographic where I have like tried to show some of the communications. It's just a few examples that happens in the marine environment. So the first one is sea lice detecting the salmon as a host via a plume of chemicals that gets released by the host. The middle one is two bacterial cells communicating with each other. I'm sure you have heard about quorum sensing. And then if you look at the upper right one, that's the fouling of ship hulls and oil rigs. So how bacterial biofilm is formed, and then the further colonization happens. And then at the bottom right, I have the settlement of corals, larvae and sponge larvae, which just are not random, but they're basically guided by these chemicals to determine where they should settle for the rest of their life. And then if we move to the left corner side, that's basically the surface of a seaweed, and like bacteria swimming towards the plume of chemicals being released by the seaweed. It can be any other macrophyte as well, like a seagrass. Now, what, as I just mentioned, that like the fouling is in the marine environment can also be guided by these chemicals. And as you are all aware, that it's a paramount thing in the marine environment. And there is a small story that I would like to share here. This is actually from the Battle of Trafalgar. And I found this story quite interesting. This is Lord Nelson, and it was mentioned by Richard Holsworth that Nelson's victory at Trafalgar was partly due to the superior speed of his clean-hulled ships. So this puts a stress that how fouling can be a really pain in the neck, that it can really determine the victory in a battle. So moving away from the story to the science bit, as like ecologists, we call, when fouling happens on a living substrate, we call it epibiosis. So there are technical differences, and where this is just a image of a Gorgonian coral, which has been colonized by several bryozoans. Now, when epibiosis happens on a living organism, it not only changes the smell, the color, the texture of the organism, but it also changes the senses, the flexibility, the drag, and importantly, the exchange of molecules between the host with other organisms and the environment. Now, the first thing that happens if in an epibiosis process is the formation of a biofilm. So imagine you have an algal host or a seaweed, and you have the colonizer pool, and an initial biofilm is formed. Now, this biofilm, although very thin, has the capability to modulate environmental stress like warming or hypoxia. It can affect the amount of light that reaches the seaweed, the nutrients. It can modulate the fouling. It can influence the gases that are being exchanged, the wastes, and it can also invite infections and predation. So when there's a lot of detrimental effect, one would naturally expect that there would be a defense against biofilm using chemicals. So these are the different layers of our work at the moment. So we work on microbial gardening and how chemically mediated interactions fluctuate with stress, including climate change stressors. We also work on phytoplankton interactions with pathogenic Vibrio. And as I have mentioned earlier, we are also working on seaweed pathogen interactions. And some of our projects are also related to the ecological roles of seaweed volatiles. But today I would just focus on these two topics, which is microbial gardening and seaweed pathogen interactions. So before I dive deeper, I just wanted to highlight whatever I'm going to talk about in the next few minutes. It would be mostly on our chemicals that are located on the surface of seaweed. At the moment, we do not work with chemicals that are intracellular, so we are very much interested in the surface chemistry of our model seaweeds. And this is something I just wanted to be upfront about to avoid any confusion. So the first thing that I wanted to talk about is microbial gardening. You may be aware of this Netflix series, which was Hack Your Health, The Secrets of Your Gut. And this is very well known how humans can modulate their gut microbiota by changes in lifestyle, by the food we eat, etc. So, and we also know is that as I just mentioned, that how different food can affect our microbiota, and how an unhealthy lifestyle can lead to a dysbiosis. So basically, you have your healthy gut microbiota, but you put stress on it, and then you have a community that doesn't support your health so much, and technically we call this dysbiosis. So similar things we wanted to try with seaweeds. Can seaweeds also do such a sort of microbial gardening? And this was the first work that was published in the aquatic environment. We know it from terrestrial plants. For example, Arabidopsis can modulate its root microbiota by releasing certain chemical compounds. And what I was interested to see that whether seaweeds can do something similar to land plants. So I'm not going into the details of this study that we did, but would just highlight the experimental results. So what we did is that this was done on the red seaweed Gracilaria vermiculophylla, which is also known as agarophyton. And it's an invasive seaweed which originates from the Sea of Japan, but now it's widely distributed from Morocco to southwest of Sweden and also along the Atlantic coasts of US. And it's a very famous invasive seaweed, if I can say so. So what we did, we kind of isolated some bacterial strains from the surface of Gracilaria, and via a tip bleaching assay, we identified a set of pathogens and also a set of protectors. So the protectors are there were 19 protectors, and three pathogens. And what we did at a later stage is that we used the same assay and we combined the pathogens together with the protectors. And what we could show is that even in the presence of the pathogens and the protectors together, there was still a protective effect of these good bacteria, if I can say like that. Now, this led to the I can't move to my next slide.
You can close there's a small X button that came up. You can close that small window. Yeah.
Still it doesn't move.
Click once on the slide and then click, it might move. Yeah.
Okay. Yeah. So yeah, so this the study that I just mentioned in my previous slide, this led to like testing of these good bacteria versus bad bacteria. So what we did, we extracted the surface chemicals of Gracilaria, and we did an anti-settlement assay. So this is a log effect ratio. So if it's on the positive side, it means attraction of bacteria. And if it is on the negative side, it means repulsion. And what was interesting for us to see is that like the protectors that I just mentioned, we tested just 7 of them because the surface extract quantity was quite less. But for all 7 of them, we saw that the surface chemistry basically attracts the good bacteria and repels the bad bacteria. So what we could say from this work is that like if we have some protective microbes, and like this is something for future, I would say, if you want to think from application angle, because we do mostly kind of basic science, is that like can this kind of work lead to improved farming? Because if you think about manipulation of chemical defense of seaweeds, there might be an avenue for it. So the next work that I just wanted to present very quickly is the effect of climate change induced stressors and how that can affect seaweed bacteria interaction. So why we need to do this kind of work? Little bit of background is that like when certain chemicals are being produced by your host, whether it's a seaweed or a phytoplankton or anything else, like these chemicals can get altered with temperature stress or ocean acidification, and this kind of work hasn't been done much in the marine environment. I think the terrestrial people are much more advanced than us. So we started doing this work, and when these chemicals are being produced, there are different stages where they can get altered, which we have tried to show through this graphic, is that they can get altered when they are getting produced, released when they are getting transported, reception stage, transduction stage, translation, and the response of the receiving organism. So they can get modified, these chemicals, at any stage. And that can in turn affect the populations, the communities, and the ecosystem functions. So just few examples. This is something that we published a couple of years ago with terrestrial and freshwater chemical ecologists. is that like in the terrestrial world, this is just an example. We know that like when an aphid, you may have seen them in your garden, is producing some chemical, temperature stress can modify those chemicals, and the receiving aphid gets a completely different chemical, which can alter their ecological interactions. And this obviously has an effect on agriculture. So basic science, this is just an example of how basic science can be applied in agricultural sector. Same we saw for freshwater fishes and lobsters, and we also know from the marine environment how ocean acidification can change these chemicals and influence the behavior of the receiving organism. So what we try to see is actually test a local stressor, which is salinity, because some of the areas of the world will be going through desalination, and we picked the Baltic Sea as one of our location or test area because it's one of the world's largest semi-enclosed brackish water seas. And it also has a strong estuarine-like salinity gradient. And in the North Baltic, the salinity can be around 1 to 3 PSU, whereas in the Southern Baltic, it can be around 25 to 30 PSU. So what we did was a long-term experiment of approximately 5 months with 3 levels of salinity. One was a low salinity of 8.5, a medium salinity of 16.5, and a high salinity of 25.5. And we had 3 different sampling points. The experiment started in May, so that was our T0. Then the second sampling point was in August, and the last sampling point was in October. And what I wanted to show here is that like as you can see is that like the bacterial cells per centimeter squared of the seaweed did change significantly at the high salinity levels, especially at T1. And the number of taxa also varied over time and depending on the salinity level tested. There was a community composition change as well over time and depending on the salinity that was tested. And this work was basically a precursor to the work that I'm going to talk about now, is that like we revisited the chemical defense story and the microbial gardening story, because following this experiment, what we did is that like we extracted the seaweeds at the end of the experiment. And we also had extracted them at the beginning of the experiment. No extraction was done during the middle like sampling point, which was in August due to biomass issue. But we had the T0 and the T2 surface extracts. And what we were interested to see that the microbial gardening that I had explained a couple of slides before, can that be manipulated? So the surface extracts were tested against the pathogens and protectors that I had mentioned. And what I wanted to show here is that like if you look at the slightly orangish rectangle, so this is the white bar is the salinity, the chemical defense, and this is from the T0 S0, which is the time point in May, and that was S0 was the salinity of the field at that point. And over time, what has happened is that the chemical defense against the pathogens have changed. And there is a significant difference between the three levels. So what I wanted to show here is that the low salinity significantly increased the capacity of the seaweed to act against the colonization by pathogenic bacteria. However, if you look at the medium salinity, which is the dark gray bar, that has been reduced. So they were doing much better against pathogens in low salinity compared to medium salinity. And if you now look at the green rectangle, so that's for the protectors or the good bacteria. So the white bar is the field collected samples and their surface extract tested against the protectors or the good bacteria. So surprisingly, the defense against the kind of, not the defense, I would say the attractive effect against the good bacteria have decreased over time for both the tested salinities. So one line I would, for as a take-home message from this work would be chemical defense against pathogens can be manipulated. So there is not only a microbial gardening that is happening for seaweeds, but if you look from the perspective of application, one can think about changing abiotic factors and thus manipulate the chemical defense against pathogens when it comes to kind of application potential of seaweeds. So the last work that I wanted to present is done by a PhD student. So this is on pattern-triggered immunity change. So you may be aware is that pattern-triggered immunity is an integral part of the innate immune system of many eukaryotic hosts, and it assists in the defense against pathogen invasions. But before we did this work, so the effect of PTI on the microbiota for non-model hosts, including seaweeds, were unknown. So what Irene did is she took Gracilaria gracilis and she exposed them to agar oligosaccharides and measured the production of hydrogen peroxide. And she did some microscopy and some 16S. I'm not going into the details of methods. So what she saw is that the seaweeds that had were controlled, but the seaweeds that were treated with agar oligosaccharides had an increased production of hydrogen peroxide. And what she could also show is that the number of microbial cells per milligram of the seaweed was significantly reduced, especially at 72 hours for the seaweeds that were treated with agar or oligosaccharides. So she also, as I said, she also did the community composition analysis, but there was no significant difference in terms of the community composition. So what we concluded from this work is that yes, PTI resulted in a significant reduction in number of bacteria, which was about more than 75% within 72 hours after treatment. However, the elicitation did not cause any significant difference in the community diversity or structure. So our conclusion or take-home message is that PTI is possibly a non-selective process, and it can help to maintain a stable microbiota by reducing bacterial loads without having any effect on the community diversity or structure. So from the work that I presented so far, I just wanted to wrap it up, what it means for the health of a seaweed. So what I think I can say, obviously this needs to be tested. Microbial gardening can be a tool to improve the health of seaweeds. As I said, we do basic science. We haven't yet delved into application potential of our work. Manipulation of abiotic factors can change the chemically mediated interaction with pathogens. So if you think about pathogens in seaweed farms, this could be a potential, kind of way for improving defense against pathogens. And my question, or like to you or to the audience, would be, are we thinking about seaweed health in the face of climate change? So because there would be new challenges, of course, and are we prepared for it? So with this open question, I would like to end my talk here and thank my collaborators and students and my funding agency. Thank you.
Thank you, Maha, for a wonderful presentation. I think we are running a little late, so maybe one question, either from the hall or from online participants.
Sure.
Hello, Dr. Saha. That's
Hi.
a fantastic talk, especially from a microbiologist point of view. So, the seaweed model in your study, is it a temperate or a tropical seaweed? The one from Japan Sea, right? That you have been. And my related question is that the microbiome of such seaweeds, are they all mapped through sequencing, or it's just that you have studied this and so you have discovered that consortium of pathogenic and protective bacteria?
So to answer your first question is that like there has been some work like where we had sampled this gracilaria from different locations, including Korea, Japan, and North America, and also Germany and Denmark. So there is some work done on that already. That was basically a follow-up work. And what was your second question, please?
Is the microbiome of such seaweeds, different varieties as well as different types, are they already mapped through
Yes, so yeah, but for certain locations only. They are not like, if I understood you correctly, they are not mapped throughout the entire world at all the locations they have been found. Only certain locations have been sampled for the microbiome.
Okay. And I have a second question, which is that low salinity and medium salinity that you mentioned, does it also affect the metabolism of the seaweed?
I couldn't hear your entire question. It got cut off. Can you please repeat?
I said that low salinity and medium salinity conditions, does it also affect the growth pattern or metabolism of the seaweed itself?
I think it does, but I have to say we did not test specifically how the biomass is getting altered with these different. The reason I say this is that like their morphology, and they're like in especially in Nordstrand, which is North Sea of Germany, they appear as huge masses. They just like growing there. However, if we compare it with some sampling locations In China, like lower saline environments, we have seen a drastic difference. So my answer to a question is yes, but we have not done any specific experiments.
Thank you.
Thank you. Thank you very much for a wonderful talk. And if anybody else has questions, you can please email and Maha will be able to answer your questions. Thank you. And Dr. Syed, I think you can take over for the next speaker.
The next speaker is Dr. Catherine Manohar from NIO Goa. She is a senior principal scientist in the Department of Biological Oceanography at NIO Goa, working in diverse areas of marine biology, molecular ecology, and marine microbiology. And currently, her research focuses on profiling the and characterizing the bacterial communities from coastal vegetations like mangroves. So her talk will be on marine microbial metagenomics and their importance in seaweed physiology, cultivation, and processing. Madam, if you can restrict your talk to 20 minutes, it would be great.
Yes, yeah, sure. I will stick to the time. Pointer. So, good evening to all, and it's almost the end of the session, and it has been very, very enlightening session in the last 2 days, and I'm very much thankful to Dr. Devashish Das, Director ILS, and I'm very much honored to present in front of people like Sri Kumar, personalities in this audience. Without much ado, I will go into the topic. And this topic was suggested by Narayan. I have to thank him for inviting me here. He wrote to me, and we had a little bit of interaction at NIO some time back. So he's given me a very exhaustive topic, and I'm not going to talk on all the topics. My previous speaker has spoken a little bit on the seaweed physiology and things like that. And it's almost the PhD work of 2 or 3 of my students, so which I will be collectively presenting today. So all know, meaning what is the diversity, what is the importance, and what are the different indigenous species and those which can be grown and cultivated. So as oceanographers, we are very excited about the Indian Ocean. I actually put this map for other reason, but I thought it is very useful. This is a bathymetry map. That is, it shows the depth in the entire northern Indian Ocean. So if you see this white patch, it is everything which is 50 meters and below. So if you see, okay, there are a lot of dots. Even if you ignore those dots, you can see that the white patch along the Indian coast is very, very narrow compared to the region which we are talking, where they are growing and cultivating seaweed in large, in large hectares, and the cultivation area is very huge, is because of this, that the region is very shallow and it's very calm compared to the very dynamic northern Indian Ocean region, where we have the monsoonal patterns and it keeps changing, and the Arabian Sea is very different from the Bay of Bengal region. And all these things not only influences the physico-chemical properties or the climate of the continent, but it also controls what is growing along the coastal regions of the Indian coast. So this work, we just compiled all the microbial studies that has been carried out from 1970. That is the International Indian Ocean Expedition, when they realized how important the Indian Ocean is, especially the Northern Indian Ocean, because it is a semi-enclosed basin. Like we have the entire landmass in the northern side, so the connection to the Pacific is only through this Indonesian through flow. And in the last yesterday there were presentation by Dr. Ika, where he mentioned about the golden triangle or the coral triangle, where we have a large diversity of coral in this region. And I actually he was not available online, so I wanted to ask him, like in India, the seaweed, growing and encroaching the coral reef regions is a big question. When it is such a big diversity and a hotspot for corals, and they are cultivating, meaning they are the world top people who are cultivating, and if it is not affecting the corals, then the same applies to us. Maybe if someone can speak to them and get some answers, I think it will be very useful to defend our work in Indian coast. So this is just to talk about these features, and then to say, okay, along the open ocean, people do work. We from NIO do a lot of work in this region, and also there's a lot of concentrated effort that's been happening in the coastal regions, which includes studies on the mangrove ecosystem, coral, and the seaweed ecosystem. Okay, why do we study the bacterial community associated in the marine regions? When they started off in the 1970s, it was just out of curiosity to see whether microbes exist in the marine habitats. Okay, it's it's a halophilic environment, deep sea, barophilic, low temperatures. So can they grow? So that was the first few years they started asking these questions just based on basic enumeration techniques to find out how do they grow and where do they grow and what do they do. This enumeration techniques has evolved over time, and even today we do enumeration, though not with microscopy, with other sophisticated cell sorting aspects. But I just want to bring to the focus how technology has helped us here also to understand the bacterial diversity and the functional profiles. They are just not seeing what are the organisms, the diversity that is present. Why are they studying the functional aspects? Because the bacteria are mainly involved in carbon utilization and in the long term storage. Even though the phytoplankton can fix a large amount of CO2, if, or for that matter, the seaweeds also can fix a large amount of CO2, it can be easily remineralized, and the CO2 can be released back into the atmosphere. But only when the life cycle is extended, when the carbon life cycle goes into the recalcitrant form and then they go into the long term storage, then it is real carbon sequestration and storage which can actually sequester the carbon which is the target. So to this is the experiments which we did in the open ocean. So to move ahead to the metagenomic diversity from a seaweed habitat. So this is just to give in brief what is the different diversity which is present here, which is mostly dominated by the proteobacteria and the cyanobacteria. And we can see a lot of these photosynthetic and chemoautotrophic forms which are present here. And also we have related this to their functional aspects as well. This is to just give a brief on this is from the meaning beach cast seaweeds, which is the seaweeds which are getting degraded, and what are the bacterial communities which are associated with these seaweeds. And so this is just about their the classes, what are the different variations which we have. And at class level, if you can see that the cyanobacteria population is really dominant compared to the other proteobacter and bacteroidetes group which are present in these regions. So we also did the whole genome metagenomic analysis of this beach cast seaweeds, and then we can get an array of what are the functional ORFs which are present. But our interest today will be especially on the carbohydrate metabolism pathways, which we can easily study based on the CASI enzymes, which we have studied on the. And we all know that the GH enzymes and the polysaccharide lyase are the main enzymes which are involved. And when we studied on the GH enzymes which are present, we can see all the familiar enzymes that we know, which laminarin and glucosidases and all these specific raminogalactrin, which are all specific to the seaweeds, which we can identify through these metagenomic analysis. Okay, this is about the polysaccharide lyases, because the whole group of enzymes, the carbohydrate active enzymes, which can break down the seaweed polysaccharides, can be divided into the glycoside hydrolases and the polysaccharide lyases. The alginate lyase, ulvin lyase are those enzymes which belong to the polysaccharide lyase family, and we can see a large representation in these metagenomic samples which we analyzed from the seaweed biome. So this is all very nice, and we also looked at from the from the mags, metagenomic assembled genomes, which are the organisms which are largely associated with these seaweeds, and we could identify cyanobacteria again, Bacteroidetes, actinomycetes, and another photosynthetic bacterial group. And then we can see the major groups GH and PLs which are represented here. So this is all fine and nice, like we know, okay, they exist from the metagenomics, but then when we have to practically apply in the industry, we need the actual enzymes which has to work. So, and why do we need these enzymes, especially to break down these polysaccharides? So just to quickly tell about the polysaccharides, they are different groups, we know. Just like they have specific pigments, they have the specific polysaccharides which is specific to the red, brown, and the green group. And these polysaccharides have a lot of advantages. We know we can use them, but they also have a lot of disadvantages which limit their application in many places because of the low biocompatibility, low permeability, and high molecular weight. And their value increases and their application increases if we can break down these polysaccharides into smaller sugar units called the macroalgal oligosaccharides. Yesterday, Dr. Hemant was speaking about chemical hydrolysis to produce oligosaccharides. So we have used an enzymatic approach to hydrolyze these polysaccharides into oligosaccharides. So because of their low molecular weight and high solubility and the GRAS status which they have, so we wanted to use it for the functional food applications. So there are certain disadvantages of using the chemical means, so and advantages of the enzymatic hydrolysis, especially if you want to use it for health or food applications. So I will just show you a little bit of okay, this is just to show that when we started the work somewhere in 1920, we saw that these oligosaccharides have been worked and many of their activity are reported, and most of the oligosaccharides were worked for agar, carrageenan, and alginate, and not much work was there in porphyrin, laminaria, fucoidin, and ulvin. So we thought it is an interesting thing to find out, and we also wanted to find a oligosaccharide which will have a cumulative effect to use it for multiple, especially the metabolic syndrome diseases like antioxidant and antidiabetic and prebiotic activity. That is what we tested. So we have isolated okay, this is all the enzymes. I think we know about all these things again. So this is the isolation procedure which we did to isolate the macroalgae associated bacteria from the seaweeds. And this is a very simple technique. If anyone wants to follow, it can be easily followed. They are well established techniques which can be used. This I just put for the sake of students. And I just wanted to just point out that we screen nearly 400 to 500 isolates, and oops. Okay. Only from the epiphytic bacteria we isolated, only 10 to 12 percentage of the bacteria showed porphyrinase, alginate lyase, and vulvin lyase activity, which we screened on the qualitative assay plate. And then we did the quantitative assay also for these, and then we identified 2 or 3 isolates which were very good for the activity, and these we have deposited with NCIM. And we have worked on this to use these enzymes to break down. We have standardized, we have done a lot of work to to standardize the activity, especially this from porphyrin oligosaccharide from 284. It is a bacillus species which we have identified. We have checked at different pH levels, temperature levels, and inducers, and to metal modulators to find out which is the optimum condition. We try to optimize a little bit, and then we use this enzyme, purified enzyme, for the production of porphyrin oligosaccharide. And then we also tested for their antioxidant activity and compared it with the parent polymer and with a control gallic acid. And we can see that the yeah, sorry. So we can see that the porphyrin oligosaccharide has good activity even at a low concentration level, and it was much better than the fructooligosaccharides. So we also compared with the commercially available fructooligosaccharides to show the activity of the porphyrin oligosaccharides which we have prepared. And we also tested their prebiotic activity based on the growth of the lactobacillus cultures and their ability to suppress the activity of the E. coli. And we can also again see that they are much better than the commercially available fructooligosaccharides and better than the porphyrin polysaccharide as a whole. So we did the characterization, and these are the uh, oligomers that we identified that they were dimers or trimers and tetramers. We had obtained all of this in the oligomers that we obtained through our enzymatic hydrolysis in the lab. So, okay, this is to summarize. So, people have done oligosaccharide production enzymatically. Some of them have tested for bioactivity, but most of them haven't tested for bioactivity. So, this was the status a couple of years back. And this is the same thing with ulvin lyase. So, we have another isolate, bacterial isolate, which has the activity, and we did almost the same type of test. And we were actually looking for bacterial isolate which can be used to break down more than one oligosaccharide, because when you see the quantitative assay, the same culture can actually do the alginate lyase also and ulvin lyase and porphyrinase, but most of them had good activity when we gave them the specific substrates. So again, we characterized the oligomers, and here it was mostly dimer or trimers. So again, the activity which we showed. So this is just in a nutshell to show like how the reactive oxygen species, any oxidative stress can actually lead to a lot of metabolic physiological disorders, including affecting the immune system, especially inducing diabetes and affecting the gut health. Today the gut health is a priority. So if we can have a combined activity and then control the antioxidant activity and then promote the growth of friendly bacteria, the gut bacteria, and then also we use the anti-glycemic assay to identify the anti-diabetic activity, then it will be can be having an application in the functional food sector. This was the motive of our study. Okay, so that is about the enzymes, and just I want to quickly add that we have screened about 15 indigenous species which are available along the west coast of India, along with some of the cultivated species. And I think maybe it is not very practically possible that we will get those isolates like for example, when we tested, we also did the total polysaccharides, which are mostly studied, but we also studied the phenolic content, which is also widely studied, I think. And then we did all the antioxidant activity. And then this is the anti-glycemic activity, which we tested. And we can see here that we tested it with acarbose. And the activity of this extract MEX10 was quite higher than acarbose, and that too in a very small concentration itself. So as academicians, it's very interesting for us to study all the different indigenous species which are available. So but there are for commercial application or for large scale production, there are difficulties, but I'm sure like with synthetic biology and other approaches, we'll be able to harness the bioactive potential of such wild indigenous species. And we also tested a little bit for the agricultural seed priming activity. And here we also went ahead and tested the minerals which are present, which is also generally done. And then we tried to correlate with the minerals and reducing sugar. And the oxidative stress is also there for the plants. So when these we again tested about 10 or 12 of these seaweeds, and we can see that that when they had a combined antioxidant and this growth promoting activity, then those macroalgal extracts showed a better result. So this reactive oxygen stress is not only for the human beings, but the plants are also having these things. So this is our team at NIO. Dr. Ravi Bagil Singh is there. He was also supposed to be here, but I think due to some reasons he could not join us today. And maybe they were there online. So he has standardized. I have to just mention just a couple of minutes for indoor cultivation of Geladium and Ulva also. And Ashok is now there at NCPR and working on polar seaweeds. So this is our team, and thank you very much for this opportunity.
Thank you, Dr. Catherine. We can take a few questions. Anyone? Okay.
Concise. Yeah. Hi, I'm Dr. Shamim. I'm working as a postdoc here. I see the graph where you have from 2000 early 1919s to till date. You have every year you have enumerated the variety of bacteria that we find in the West Coast, and I suddenly see in 2010 and 2016 or 15 there's a sudden decrease of the number of bacteria that you have observed. Any reason for that?
Those are not bacteria. Those are publications. Those are the number of publications
Reporting.
published in each
Okay.
of the years on bacterial diversity from the northern Indian
I
Ocean.
mistaken it as a number of
No, it's
species
not
or
number
something
of
that is
bacteria
discovered
because
in.
that's too much to comprehend.
Okay.
So we have not done that. It is just the number of publications.
In that particular year.
Yes, yes,
Okay.
yes.
Thank you.
Oh, thanks, Dr. Catherine. Nice work. So I was particularly looking at enzymes for Gracilaria.
Yes.
So did you find some of those good enzymes? And I was also looking at, although you did show some genomics data on categorizing GH families and all that, but I don't know whether you could see how much of those are actually specific for the Gracilaria and all that side. I thought I'll just ask you.
Okay. So actually, we presented this work when Narayan had come to NIO, and they also asked us the same question because we were intrigued by Ulven lyase and porphyrinase, but we have now. We have agarase and alginate lyase, carrageenase. These are the three things I think commercially important.
No, and given the interest in this topic, what I think we'll do is we have a slot for a panel discussion.
Okay.
So I'm going to put you, Shams, Dr. Walker, and onto the panel, and we'll discuss this question in thoroughly.
Okay.
What do we need to do to find the whole series of bacterial polysaccharides, saccharases? Okay, and you can.
Sure.
We'll all be waiting for an answer while you guys debate.
Yes.
Okay.
Yeah. Thank you.
Go on with it.
Yes.
On your 17th slide,
Yes,
there was a map of all the states who are producing how much of seaweed.
No, that diversity of seaweeds.
Odisha was not there. Next presentation, I would like to see.
Yeah, sure. That is the diversity. Maybe it should be there. I don't know. I don't want to go back. Yeah, it is there. Diversity is reported from Odisha, and cultivation work also we are doing here. We have not missed Odisha.
Yes. Thank you, Dr. Catherine. So the next speaker is one of the hosts, Dr. R.A. Narayan from C6 Energy Private Limited. He is the head of the Ocean Agritech at C6, and before that, he was a director of Customized Technologies Private Limited, where he led software development for almost 15 years. So it's quite surprising what made him shift his gears and come into active research. Yeah, and he did his electrochemical engineering from University of Minnesota, and then finished his PhD in chemical engineering from the same university. And today's talk will be on laboratory methods of seaweeds. Thank you.
Thank you, Dr. Syed. I'd just like to admit one thing, that I didn't know that seaweeds existed until I joined C6 in 2020. So it's been a journey of learning for the last almost 5 years now. and I'll share whatever I've learned with all of you here today. This is a brilliant slide that Ian had prepared for one of his talks. And as we all good engineers do, we always take it, we loot good things from others. So I took away the slide from him. And this is kind of a driving force for me as I'm working on Ocean Agtech at C6 and trying to enable large scale farming in the oceans. So what this shows is where we are today, which is primarily adaptive economy. This is basically old style agriculture where people work on small changes by understanding what's the weather patterns, water availability, and trying to improve crop productivity in their farms, and moving towards precision agriculture, where we know exactly how much nutrients we need to supply, what is the weather going to be. So based on the weather, how we should supply different nutrients and pesticides and other situations to get the maximal productivity out of our farms. So there are two parts to this. One is the culture environment, which we don't really have direct control over. And the second part is the crop biology, which is very little is known for seaweeds as a crop. So that is why we are here today, trying to see if we can go up the axis on the crop biology so we can do better and improve, get to precision for economy eventually. So what does precision for economy mean in terms of what we need to do? So we need to know better on tropical seaweed physiology. How do we manage nutrition to the seaweeds? We need to expand genetic diversity and cultivar development. We have had a couple of wonderful talks by Eka Prasadya and Mikey yesterday on these parts. And of course, wonderful talks by Mahashweta and Catherine Manohar on the microbiome and ecological factors that affect seaweed growth. On the technology side, of course, we need monitoring and control. We need to do mechanization and do better ocean farming operations. I will not be talking anything about the technology aspect of it today. Just to emphasize, there's a huge number of factors that affect seaweed growth when we are growing them in the ocean. This is just a small snapshot of a little more than a year in one farm where we are trying to grow Eucuma denticulatum. And you can see the variation in temperature, in salinity, in the number of different seaweeds and fowlers and flora and fauna that are coming and affecting this. Plus throw in a couple of storms in the middle and you have a huge number of variables that affect how seaweeds grow and that affect the productivity of sea farms. So here's a small summary of some of the important factors that affect seaweed physiology. Some of the major things are temperature, salinity, water currents. Obviously, water currents are important because they are the things that bring in fresh nutrients and take away waste products from the sea farm. There are predators. We have seen earlier today people showing rabbitfish and parrotfish and jellyfish coming and giving trouble for us in the sea farms. And of course, there's a lot of fouling by other seaweeds, microalgae, and epiphytes and dirt as well. So how do we start? Where do we go to make some progress on seaweed physiology? So what we saw when I first came and joined C6 was we didn't have a simple and easy method to grow seaweeds in the lab, wherein if I can have it in a reliable manner in the lab, then I can start asking questions. What happens if I add nitrogen? What happens when there is nitrogen deficiency? What happens when the temperature goes up? Because there are so many things changing simultaneously in the ocean, they're confounding the actual experiments that we do, and we end up not being able to have clear understanding of what is actually affecting the seaweeds and what is the cause and what is the symptom of a particular problem that we are seeing. So we said that we wanted a simple but reliable system. It should be of minimal volume so that we can even do the work in a lab in Bangalore, which is not on the sea coast. To eliminate the temperature problem, we decided that we'll have a lab where we'll control the temperature at a constant one. We will try to see if we can stabilize the salinity and it doesn't change over time. And of course, as always in India, so cost is always an important factor. So we wanted to keep it simple and easy. And can we do it away from the coast? So to do that, after trying many different methods, I'm just showing you the final result here. But trust me, it's taken us more than 2-3 years to arrive at this system. But it's very simple. It uses our standard 2-liter beakers that we use in our labs. We use simple low-cost slotted angle shelves. We use the slotted angle shelves because we could adjust the height of the shelves. And as you see, we are using simple LED tube lights here. The idea of the slotted angle shelves was because we could change the height. And as you know, the intensity of light changes at the square of the distance from the source of the light. So to control the intensity of light to the seaweeds, we could just simply shift the height of the shelf and get the appropriate intensity that we needed. So as you can see, the summary of the ideal conditions for Capophycus alvarezi, the Indian strain that we are growing, is summarized in the slide on the left side. To show you in more detail, I've zoomed in on one of the beakers. I hope you can see the small yellow tube here. This is basically a polymer tube. This is a pneumatic tube. We have just made 2 or 3 holes in this, and you can see small bubbles coming out. So this is to control the aeration to the seaweeds, and we are suspending the seaweeds with a string, cotton string suspended from the top, and we just put a simple transparent acrylic sheet to cover the beaker to eliminate evaporative loss. And once we did this simple system, we were able to have a stable growth rate up to 2 to 3% per day in the beaker for up to 45 to 60 days as well. So to understand whether this system could work, would the seaweeds respond to such an artificial system as they do in the natural environment? It is well known that seaweed growth is usually nitrogen limited in the seawater. So what we did, we took natural seawater and we started supplementing that with 10 micromolar of either potassium nitrate or ammonium chloride. And we could see immediately an increase in growth rate on a per day basis when we added either nitrate or ammonium as the nitrogen source. So we could see that the seaweeds are able to replicate the same physiological response as seen in the ocean when people would add nitrogen to the seawater. So what we understood here from this was that we could get a reliable, repeatable and consistent system that was independent of seasonality, and we could get this growth rate curve all through the year, all 12 months of the year in the lab. So we could now look at seeing if we could get increased growth rates and start asking other questions of the seaweeds. The next question we wanted to see was how generic was our technique. So what we did, recently we started working with Gracilaria a little more. So we got some Gracilaria corticata from the wild, from the Tamil Nadu coast. So we put that same 2 grams to 4 grams of Gracilaria in the same 1.5 liters of natural seawater. And we could keep them growing well and decently up 2% plus for about 30 days. So we have done this for 2 subsequent generations of vegetative propagation. And we are trying to extend this to other seaweeds like Sarconema filiforme as well. The next question we wanted to ask was, okay, now we have a nice and simple lab system to grow seaweeds. We wanted to know if we need to feed the seaweeds in the ocean farm, how much nitrogen do these seaweeds need for them to grow, how fast they take up. So we wanted to investigate what's the uptake rate of nitrogen, of nutrients, and what is the storage capacity of these seaweeds. Algae, as some of you may know, have this capacity to absorb nitrogen very quickly and store it intracellularly. and then they update, they use it for biosynthesis later on as and when they need it. So when we went and saw the way the nitrogen nitrate is measured in seawater, this classical method that is very clearly delineated in the ICMM protocol document, it uses 100 milliliters of sample. What we have done is go from thousands of liters in the ocean to just 1.5 liters in our 2-liter beaker. So if I take a 100 ml sample to do one nitrate measurement, I really cannot do kinetics of nitrate uptake. So what we thought of doing was, can we reduce the volume of sample we need, and can we do this assay faster? So this assay usually takes about 7 hours to do about 12 samples, one sample at a time through the column. So to show you only the summary of a long amount of work, about 6 months of work. So we reduced it to a 1 ml sample of seawater, and just using 40 microliters of an expensive optical dye to do absorbance measurement, and we got an R-square value of 0.999 plus, and we could reduce the time of analysis from more than 7 hours to 1 hour and 15 minutes for us to do this experiment. We are next working on scaling this up to do it in 96 well plates, so we can do it for multiple seaweeds and for multiple conditions as well. We have conducted a similar scale down method for ammonium as well. And then we went ahead and did the ammonium uptake kinetics in Kappa ficus. And as you can see, the seaweeds are able to soak up the ammonium in less than 4 or 5 hours. So what we did next, so if you put only 10 micromolar, all the ammonium is disappearing in 4 or 5 hours. So to ask the question of how much ammonium they can take, so we added excess ammonium to the extent of 60 micromolar, and we were able to see that they were able to absorb all the ammonium within 24 hours of adding the ammonium to the seawater. So we could estimate that Kappa ficus is able to take up at about 30 micrograms of nitrogen per gram of fresh seaweed per hour. So this is very important to know, because now you can understand how fast seaweeds are able to uptake nitrogen relative to other organisms in the ocean, and we can design any kind of feeding strategy in the ocean farm better with this kind of estimate. We also found out that the seaweeds are able to store at least 150 micrograms of nitrogen per gram of seaweed. We are investigating this further to see if they can do more than 150. So these experiments are still underway for us. Another interesting question that came to us once we got the system stable was, what is the smallest size of seaweed that we could chop up and that would still grow to be a fully adult plant? Because Kappa ficus grows vegetatively very well and very conveniently. And so this was something we wanted to do. So what we did, we took 0.5, 1, 2, and 3 centimeter sized thalli, and then put them in the beakers and then let them grow. And you could see after 45 days, we saw that 1 centimeter and 2 centimeters were doing very well as compared to 0.5 and 3 centimeters. So we chose the 1 centimeter size as our optimal size for micropropagation. So what does this simple technique allow us to do? So it allows us to do micropropagation. So if you start with just 1 kg of seaweed, for example, after maybe a natural disaster in your sea farm, so you have very little seaweed left, so you can chop it up to small pieces and then start to get lots of individual seedling pieces to expand your seed very quickly in land nurseries. So that's one of the resilience techniques that we are trying to implement in our ocean farms. So, and then of course, what this also allows us to do is, if you have a strain bank of seaweeds, such an ability tells us that we need to have small amounts of seaweed that we can maintain in our strain bank. And so we need to use less water and less change and less labor as is needed to maintain the strain bank. So these are all things that we are trying to implement. So one other crazy thing that we tried to do was we are trying to improve seaweeds. There's only one variety of Capophycus alvarezi that's grown in India, and as all previous speakers have shown before, it's not doing very well in India. There is massive decline in ocean farm productivity in India. So we thought, and these are vegetatively propagated varieties. So it is not been shown to get back into sexual reproduction. So that's another challenge. So we thought, can we make a shortcut and see if we can do a mutagenesis protocol to see if we can produce new varieties of Capophycus that could be resilient to climate change and other problems that we see in the field. So to cut a long story short, we did UV mutagenesis because that was the simplest and easiest with our 1 centimeter thalli pieces. And after a few minutes, 2 to 4 minutes of irradiation under UV, we recovered them for anywhere between 20 days to 200 days. And after they recovered, we put these mutants under high temperature of 35 degrees centigrade for 35 days. Sorry, 30 to 35 days at 35 degrees C. They were constantly at 35 degrees C, day and night at 35 degrees C. And then after 28 to 35 days, we would put them back at 28 degrees centigrade, and repeat the cycle several times to ensure that our mutants, whatever survives these conditions, would be very stable, and we could get a mutant that would survive 35 degrees centigrade. In the natural ocean, you won't get continuous 35 degrees centigrade water in the ocean for more than a few hours, even on the hottest day in the summers. So here's one of the mutants that has grown over the last 3 or 4 months. And what is interesting to see in these mutants is even at 28 degrees C, they are growing at a higher growth rate than the natural wild type. And at 35 degrees C, they'd still show a growth rate of about 1%. So we are in the process of characterizing genetically what are the differences. And I hope with ILS, we can do some transcriptome analysis as well to understand what is giving these mutants the heat resistant capability as well. And so as a part of a new cultivar development process, so usually tissue culture is the best method used today to get clonally consistent new cultivars. But then tissue culture in seaweeds takes anywhere from 9 to 15 months. So by doing this small piece culture in the lab, we have been able to speed up this process by up to 15 months. So we have done the mutagenesis, we have put it through selection pressure. So now we are establishing the stability and we are expanding so that we can get to ocean trials soon. So and release the new cultivar to all farmers in India as well. So what have we learned till now and what's next? So we have a reliable and consistent system to investigate seaweed physiology. So there are a lot of nutrients. We have done a little bit of work with nitrogen. There is phosphate, there is iron, there is many other micro and macronutrients. We need to know what are the essential nutrients for each commercially important seaweed. And we would like to use this to generate new mutants for different low salinity, resistance, temperature, and disease resistance as well, and see what we can get with those. Transcriptomic level studies would be good. We are also looking at whether we can use these beaker systems to extend them to be a disease model. So many people have tried to identify what is the root cause, which is the pathogen causing bacterium, for example, of eye-size disease in Capophycus. There are candidates, there are bacterial candidates, there are fungal candidates, but there is no reliable lab-based system where you can use it to show you can reliably produce the disease and then infect it with a particular bacterium and establish that the disease is formed by after infection with that particular bacterium. So those kind of things can be done if we can develop a disease model. I'll stop here. Thank you very much. And if there is time, I'm ready to take any questions. Yeah.
Ian, do you have a question?
Yeah.
Go ahead, please.
Not more of a comment. I just want to say compliment you, RA, on the fantastic progress you've made in such a relatively short time. It finally got us the capacity to do this sort of work.
Yes. Thank you. Thank you, Ian.
Yeah, Arik, you can introduce the next speaker. Thank you.
Okay. All right. So our next speaker is Dr. Alan Critchley. He is a veteran in seaweed biology. Long before many of us were even born, he did his PhD in seaweed biology from the University of Southampton. Am I correct, Alan?
Portsmouth.
Yeah, University of Portsmouth. Then he was a faculty in South Africa and Namibia before he moved to France and worked in a company called Degussa, which was one of the biggest Carrageenan processing companies. And then he moved to Canada, worked in Acadian sea plants for almost for 16 years, I guess, before. Right now, he's a scientific advisor for us at C6, and he's a great mentor and guide for all of us. So I think he has already pre-recorded his talk. So I think we can listen to his talk, and then he'll be available for any questions. Thank you.
Thank you very much. Good evening, everybody.
Dear Dr. Dash, hello, greetings and good day to you all in the audience. I'm sorry I could not be with you, but I hope to join you online at the end of the session. I greatly appreciate the opportunity to talk to you today. I hope you will find some points of interest in the story I will tell you of.
Did it stop? The sound finish? All right.
Yeah, I'm not able to hear.
Yeah, but it should have advanced.
Yeah, same here. I'm also not able to hear.
Okay, if it doesn't work, we'll switch over to the slides and I'll go through that quickly.
Sure.
Dr. Dash, hello, greetings and good day to you all in the audience. I'm sorry I could not be with you, but I hope to join you online at the end of the session. I greatly appreciate the opportunity to talk to you today. I hope you will find some points of interest in the story I will tell you of how meticulous studies in basic biology, blue sky research, if you will, using the simplest of equipment led to a multi-billion-dollar industry. However, not one without its economic problems, but a success story nevertheless, not unlike some examples from kelp or even eukumatoid cultivation. There are many parallels which I hope become apparent. It was done once, and I believe you could do it again. Now, everything that you're going to hear about Kathleen Drew's work is a true story, but some of the events may have been dramatized for effect. I've got about 80 slides to get through, so I need to go quickly. You'll get a copy of the PowerPoint, so please allow me to go quickly and then cherry pick through some of the slides. So the origins of modern nori gim or hai-tai. These are certainly not the largest by dry weight produced, but certainly one of the most largest valued seaweed products in the world. Sushi rolls are now globally ubiquitous, as well as gim. And the value of nori or gim is in the eye and the taste of the beholder. We'll see that a little bit later. And once nori sheets were suitable gifts for the Emperor of Japan, or used to pay taxes. And I wish I could have done that recently. So this is the story of Kathleen Mary Drew Baker. Her achievements are monumental. She was a lecturer in cryptogamic botany in the UK. She published 24 papers on botany. In 1928, some of her taxonomic work was published in the University of California Publications in Botany. She was awarded the DSc in 1939, and that was well before her noted work on porphyra or lava. In 1953, she became the first president of the British Phycological Society, and in her 33 years of research, she collected over 2,700 species or specimens, and these are now donated to the British Museum. So, her story. This is the story of the cultivation of porphyra. Sadly, with all wild stocks which become popular, there's a tendency to over-harvest, and that's referred to as the tragedy of the commons. And there's not enough supply for the ever-increasing demands. Then came along this old school traditional British lady taxonomist, and then unknowingly, serendipity perhaps, she was able to rescue the porphyra industry in Asia. So Kathleen Drew worked from home. She was working in her UK home laboratory. She had been dismissed from her university because she'd had the audacity to get married to her husband, a Dr. Baker. He was an engineer. Anyway, when she was working away diligently, she was looking at two different red seaweed genera at those times, one known as Conchicellus rosea, the other Porphyra umbilicalis. But they were one and the same thing. They were members of the genus Porphyra. She published 3 articles, 2 of which were just 2 pages in Nature. I'll come on to show you those in a minute. She wrote her findings in letters to eminent Japanese phycologists. There were no emails in those days. They've been scratching their heads over exactly the same issues. And then the rest is her story. The Japanese were able to take Drew's findings and then commercialize porphyra cultivation on a huge scale. And as such, she's known as the mother of the sea and is still venerated in Japan. I thought I'd ask a few people if they would give me some quotes. And I think many of you know Professor Seaweed, Charles Yarish, formerly of the University of Connecticut, now a visiting investigator at Woods Hole. And I asked him for a quote. And he said that the present development of Pyropia porphyra, Neopyropia, nori or gim or zikai in Chinese is due to the scientific discovery of the life cycle by Drew Baker in 1949. Her paper may have been so short, but its impact led to a multi-billion-dollar aquaculture industry. Now, that as a side, Charlie sent me this picture of him. It's one of his favorite memories of visiting the marker dedicated to Kathleen Drew Baker overlooking Ariake Bay in Japan. It's also the favorite picture of himself, which you'll often see online. So let's look at that marker or monument to Kathleen Drew. After her publication, the Japanese literally had a eureka moment, and suddenly it all became very clear and a path to cultivation at an industrial scale was obvious. And such was the appreciation of the Japanese seaweed farmers that they actually venerate and hold various services at a monument for her, which was erected in 1957. This is the Sumoyishi Shrine, Yuto, Japan, and every year on April the 14th, there's a commemoration called Drusai, and then contributions are made, flowers are put out, and discussions of the science that developed the seaweed industry. So she was a traditional taxonomist with a very inquisitive observation, a simple microscope and petri dishes, very elegant work, and she worked from home. Here's one of the pictures of Drew at her desk with her book and looking down the microscope. And then that publication in Nature, and it literally was a letter to the editor. Now, at that time, Drew was working on Welsh lava bread. This is Porphyra umbilicalis, which is foraged. It's not cultivated in the UK. It's picked by hand from intertidal crops, generally in the cooler months, and then treated in various ways. And a particular delicacy in Wales on St. David's Day is to use lava as a jam or as a mixture that goes in with oats and is then fried and would commonly be eaten with a British breakfast. But as you can see, that one there, there's black sausage, beans, egg, bacon, and sausage, and a real cholesterol feast there. So this is it. This is the letter to the editor of Nature in 1949. It's a single authorship. It's only 2 pages, and it's what set the Asian Porphyra industry on its way. Now, notice in this illustration there are shells, and also the top right shows you these small filaments, once identified as a completely different genus and species, Conchocelis rosea, but Drew was able to find them in shells. And this is the first of her 2 pages that really did rock the phyconomic world in 1949. Here's the second. In 1954, she rewrote some issues about the Conchocelis. Again, it's a single authorship in Nature. And again, that shell there is illustrated as a cockle. And one wants to know, does the filamentous phase actually burrow? How does it get into the shell? That's a question that I don't know. There is a third paper, which those of you interested in the story should look it up, Studies in the Bangioideae, about the life history of umbilicalis, where Drew has looked at the culturing of the filamentous phase in various types of shells. Most of those are bivalve shells there, but I will bring your attention to the fact that the illustration number 9 is where she put some eggshells in. Now you can imagine her being at home in the kitchen, doing the kids some breakfast, taking off the tops of the eggs for their dip their soldiers into their eggs, and then taking those little bits of eggshells and putting them in with her cultures. And lo and behold, even those calcareous shells were used as a habitat for the Conchocelis. So let's look at that a little bit more in a little bit more detail. So we know that the Porphyra, the large blade, is the gametophyte. That's the haploid. The blade phase is what's collected, it's what's eaten, and it alternates with a diploid phase, which is called Conchocelis, which literally does look like pink candy floss or pink fluff. Forms as filaments that are able to dissolve the calcareous medium of shells and, for want of a better word, to burrow into those shells. Now, I asked Professor Yang Kim of Incheon University, Korea, please, Yang, give me a quote about Kathleen Drew. And he gave me a little bit of an anecdote as well. Kathleen Drew transformed the seaweed farming industry from unpredictable to sustainable. Her work directly influenced the Korean gim industry, and that led to exponential growth since the 1970s. And Korea now is one of the most successful producers of porphyra gim in the world. Now, as that anecdote, he had a reprint of her first Nature paper published in 1949, which he auctioned at the annual meeting of the Korean Society of Phycology. He got $500 for that. That's pretty good, eh? $500. So don't throw any of your reprints away, especially if they were signed by the first author. And I wonder how many people in the audience know what paper reprints are and the joy of receiving them in the post from the authors. Moving on. So eventually in 2022, Kathleen Drew Baker was honoured in her home city of Leigh in the UK. And there they have these blue plaques that they put on the wall where auspicious people were born or lived. And this one happens to be on the wall of the school, which is in the road where Kathleen was born. So that it took a while, but at least her contributions there as Mother of the Sea were acknowledged. So I asked another friend of mine, Professor John West, who's Honorary Associate at the University of Melbourne. I said, John, give us a quote about Kathleen Drew. And he said that when he first started his PhD program in 1961, he was inspired after many hours of reading in the library about the Porphyra conchosilis. But John actually, as a tremendous red algal taxonomist, he was actually more taken by some of her earlier work. He mentioned the work that Drew did in California in 1928, and this helped John select Rhodocorton and Acrocisium to discover their diverse life histories and seasonal day length. And that has led to a very fruitful 64-year career in culturing reds and publishing the results. And then John has a copy of that paper in the University of California publications, and he treasures it. Let's go back in history. Here I've got the cultivation was around about the 17th century. And this is phyconomy by trial and error, or bricolage. Simply here, the nori farmers are enhancing the amount of substrate for the natural settlement of spores from the fertile blades of the nori. They didn't know they were collecting the spores at that time, but they were perhaps doing a primary selection of cultivars. And then as those sticks and branches became wet and dry during the high and low tide cycles, these kept the blades relatively clean of epiphytes and diseases. This is what harvesting would look like, where the blades would be brought in and then arranged in rough square paper-like sheets. Saw this picture before, the blade and the filament. Here's cultivated tenera. This is the strain that is used commonly in Japan. And here are the oyster shells. Now, in Japan, they moved to using oyster shells for collection of the spores within which the filamentous conchosilis would grow. This became produced on a huge scale. You can see these very large greenhouses with many thousands, if not hundreds of thousands of shells, and then an automated system of wheels which would have nets on them, and the nets would rotate. At the time when the conchosilis was releasing spores, the nets would be the surface onto which the spores would settle. And then those nets would be put out in the open water, and then at times the nets with the growing blade would be washed, and this would be to remove epiphytes from the surface. These would then go into machines, some of these machines costing over $1 million each. And these are turned into now very uniform sheets. And these are the sheets that we're more familiar with, which would wrap various rice, sushi elements. And then here's checking the various grades of the nori sheets. And one would be surprised as to how many grades there are. And it's graded on color and texture. And these sheets then are then bundled. And then those bundles would go into boxes, and then there are very large auctions of these bundles, and the specific grades, the highest grades, can go for very, very high prices. So she sells seashells. Again, what we knew, what Drew Baker figured out, and then there are the oysters shells for the development of carpospores. It actually takes a large area, a lot of investment to have the tanks within which the shells are housed. Back in 2006, people actually began looking at, and this is Pimin Hay and Charlie Arish, began looking at the mass cultivation of free-living Conchocelis. So they're able to grow the Conchocelis now independently of the shells. And this allowed for mass cultivation, not unlike microalgal cultivation. And then that work, the mass production, went into bioreactor production, free-living Conchocelis, and large amounts of Conchocelis can be produced and maintained in bioreactors under various levels of temperature, light, and photoperiod. So it really does speed up the cultivation, and it makes cultivation free from having to have the shell phase. The process is less cumbersome, requires less labor, and mass release of conchospores can be induced and then seeded onto the net. Okay, here's the life cycle of Porphyra. It's considered to be simple, or was, at least when I taught it, it was simple. It was hard enough talking about the Conchocelis in the shells. But if one looks at some recent publications here, 2018 by Varela Alvarez et al., Porphyra is anything but a simple case. You can get 2x, 3x, 4x, you can get triploid, tetraploid, and all sorts of combinations in between. And I would highly recommend that article to you. Okay, Kathleen Drew's findings led to a boom time in production. Demands for materials increased globally. And then, perhaps it was too fast. Diseases and pests are now known within the stocks of various commercial porphyroids. So all is not well in the Nori Gim high tide garden. Let's have a quick look at red rot disease, and these figures were uploaded by Gwang-Hoon Kim, who is perhaps the foremost expert on this. But you can see that the red rot disease causes discoloration and pitting within the thallus. There is a green spot disease, and this is commonly associated with Alpidiopsis. And we can see that this is an infection which burrows within the thallus of Porphyra and leads to this mottling effect in the lower area here. So essentially the thallus disintegrates. Looking at that in terms of phyconometrics, or looking at it in terms of economic impact, the Alpidiopsis disease alone has caused approximately 1.6 million losses, or 24.5% of sales losses in 2012 to 2013. Green spot has resulted in just slightly less, but 1.1 million, a total loss of 10.7 of sales. Regular acid washing of the Pyropia cultivation nets is the most common treatment of all the above mentioned diseases. However, and this is something to note here, that those treatments represent 30% of the total cost of Pyropia seed farming, and it is ineffective for some diseases, especially Ophidiopsis. Now, what do those green tides look like? And porphyra cultivation and, let's say, poor management of the ropes and rafts have been considered to be highly negative influences of green tides in coastal Chinese waters. But without the necessary nutrients present in that eutrophic water, you would never have got such large outcrops of seaweeds. So fire has long been farmed in the area, but in 2006 they started moving offshore. A 2008 bloom of ovoids covered about 5,000 square miles, and 10,000 workers costing $30 million were involved. Even the army, the People's Liberation Army, were brought in to clean up the beaches. The algal blooms often follow massive discharges of phosphates and nitrates. The seaweed farmers, the nori farmers, have been asked to change the way they dispose of their algae that grow on their rafts. And nori farmers in the south have found that their rafts attract algae which get cleaned off each spring into the Yellow Sea. Now, moving on quickly, let's talk about kimbap and TikTok. Kimbap found its way onto TikTok and became a viral phenomenon. And what happened was this put pressure on the production of porphyra in Korea, and there were shortages of gim for gimbap restaurants within Korea, because lots of the material was being sold at high prices to international markets where gimbap had also become very popular in other markets of Europe and North America. So recently, there has been a production surge in South Korea, which leads to prices dropping and also wastes. And it was reported in the Chosun Biz, and this was in January 2025, that 1,200 tons of raw seaweed were discarded into the ocean. Now, that's 10% of the consignment volume. There are also illegal gim farms on the rise, it would seem. So good governance is required in supply and demand. Many of you have heard of Wando. And in Wando, the Marine Police Station recently caught 22 fishermen who were setting up illegal seaweed farms without permits. And the problem with those is they put them in areas where they were concerned about shipping. And as the popularity of seaweed has surged worldwide, the prices have risen, and so illegal aquaculture farms seem to be on the rise. This is another recent February 2025. South Korea's fisheries sector shows that the product becomes popular, the producers flood the market, they drive up the supply only for the prices to crash. The cycle of overproduction and price collapse will continue, and there's a mismatch in supply and demand. Now, these are just some reports I got out of the press. I took some data from the global production for pyropia from the Hatch Blue site. This is from Seaweed Insights, and just a few sort of Economy 101 updates. In the Chinese production, there seems to be unlikely continuous growth. And this is because of government restrictions on expanding into new areas. And you saw the same issue with GIM in Korea in those newspaper reports. For Korea, the production looks like that primarily it will be constrained by limited space in the near shore. And also that there are in Korea now shorter seasons due to climate change, and Kim likes colder waters. But the South Korean National Breeding Program is developing climate resistance and high quality cultivars. So there is perhaps a silver lining. Interestingly, in Japan, where this all started out, the future in Japan is primarily constrained by the lack of labor due to an aging population and also in the pyropia farming section, low levels of automation. We saw high levels of automation in producing the sheets, but perhaps not so much in the farming. And it really is strange to see Japan in a position of declining production. So just as I'm wrapping up here, just a few things. When Elizabeth Gant and many others published, they said that maybe Kathleen Drew wasn't working on Porphyra umbilicalis, but it was almost certainly Porphyra dioica. I asked Professor Juliet Brodie for a quote. She was definitely inspired by the work of Dr. Drew, and she's actually asked herself whether Drew was working on umbilicalis for the 1949 paper. Juliet went on to say that she published the first barcode paper a decade before barcoding was invented, and that more taxonomic genetic information is required from around the world. She's going to create a new Vangelis group, and it all depends on who's still working in that group. Remember that taxonomy is not being taught as much in universities as it once was. So, what's Kathleen Drew's achievements? We've been through this. She had a remarkable career, and she continues to inspire contributions to phycology. So very quickly, 1957, Utah erected a monument and bestowed the title of Mother of the Sea. Drusai is every year on April the 14th. She actually is a patron saint to which the entire community is indebted. Kathleen Drews Baker was immortalized in 2022 with a blue plaque. And then, years after their mother's passing, Drew Baker's two children, John and Francis, traveled to Japan and were treated like celebrities. John Baker, when he was 64, said his mother never went to Japan and never knew what a difference her work would make. She studied the seaweed purely for scientific purposes and blue sky research. And then he didn't know what his mother would make of it now, but she'd be surprised that what she did back then turned into a multi-billion-dollar business. John then donated in 2003 the microscope which Kathleen Baker did her work on, and that now resides in Manchester's New Science Gallery. And that's a picture of John in the bottom right there with the sheets of nori and his mom's microscope. So I'll finish on this, where when asked about what their mother would have thought about the sushi she'd helped to birth, John said, "I don't think she'd like sushi. She wasn't very adventurous when it came to food." And there we go. Her story of Kathleen Drew, Kathleen Mary Drew Baker, Mother of the Sea, and maybe the Princess of the Porphyroids. Thank you very much for your attention. Dear Dr. Dash, hello, greetings and good day to you all.
Not again. Stop. Stop.
Thank you very much. Maybe a couple of questions. Anybody?
Alan, can you hear me?
Yes, hi. Good evening, Sri.
Yeah, hi. So, I mean, we have so much to learn from in history, right? I mean, what goes wrong, you know, you create a whole new industry and then there is pests, diseases, oversupply, undersupply. When we are starting off something new, I mean, and I think, you know, when Ian spoke yesterday, we're also seeing this whole thing happening in the tropical seaweed industry. We're discovering it, except that I don't think it has the basis of science that at least the Porphyra industry has. So, being familiar with this, Alan, what are the missing gaps for us if we really want to build this as a future feedstock? Just take a shot.
I think it's matching the supply and demand, but with any one of the biomasses to have multiple uses. When a product becomes commoditized, even as much as the nori sheet is specialized, it's now become a commodity, and in many cases it's sold then on price. But going back to some of the earlier work that I listened to, if you think about some of the oligosaccharides that you can get from porphyrins, which is the gel, then I think there are a number of immune stimulating properties that could give rise to, whilst also, I mean, I think it's criminal that people have grown over 1,000 tons of seaweed and then they just throw it away in the sea because they don't want to sell it for a low price. They want to try and maintain a higher price in the Korean gim market. So matching supply and demand is important, but I think having multiple streams of use, and I'm very much a fan of biorefinery approaches to seaweed biomass.
Thanks a lot. Ari?
Can you guys see me? Okay. I just wanted to make one small additional comment to Alan's talk. One lesson I get from his talk is how important basic research is. When we in the industry will sit and do all kinds of applied work, and unless you have basic science, there is no technology. Technology is application of science. And if you don't get the basic science done, the technology and the industry is essentially handicapped. You don't know when the next disaster is going to happen. So it's really, really important that somebody does the basic science. And I really hope ILS will take this as an inspiration and take the basic science work forward.
No, I absolutely agree with that. And then going back to the earlier talks, particularly of Dr. Saha, climate change is going to affect even the basic biology. So you can't do basic biology once and then stop. It has to be continuous. And I think one of the things that we'll face seaweed cultivation, it's already underreported now, but diseases and pests are becoming major issues. And then even unintended consequences like those green tides in China come from the seaweed farmers thinking they're doing a good job of keeping their nets clean of the epiphytes. And yet because of the eutrophic waters, you get these phenomenal growths of green tide. So everything is interlinked. But without the basic biology and understanding, we don't know how to address the issues.
Ari, may I?
Yeah, please go ahead, sir.
I think we are naturally getting into the next topic, which is panel discussion. So my question is for all, and I'm Debashish Das, currently director of Institute of Life Science, and I have almost zero knowledge on seaweed biology. Having said that, I have a question to all the people who understand that, given Odisha coast, where I belong to, which is a rough sea and quite a bit of cyclone that comes every now and then, do you think in the open sea doing seaweed growth and for the purpose of generating thousands of tons of seaweed requirement by the industry, should we advise the regulators, the state government to allow that? Or what are the solution from your side, suggestion from your side, that we can give it to the state government policymakers, that how much greed is allowed and after what stage one should start putting a barrier? Ari, you may define the question a little better for better.
No, I think it's pretty clear to everybody. So basically, he wants a policy kind of answer. So what policy advice he can give to the government, given that it's the Orissa coast where ILS is situated, is pretty much open to the larger Bay of Bengal area, and you do get 1 or 2 cyclones every year over there, and the monsoons make the seas pretty rough for about 3 to 4 months every year. So Alan, Ian, you guys have any thoughts that you would like to share?
Yeah, not so much policy, but in terms of engineering, it's possible to construct a seaweed farm that would survive even the worst of those conditions, not necessarily at the surface, but it would need the ability to be sunk and go lower in the water column during the roughest times. But I know of a company called Kelp Blue that's engineering for macrocystis farming in the open southern Atlantic Ocean, which is one of the roughest. So it is possible, but you have to make sure that the value of the crop can actually accommodate all of those costs. But in terms of policy, I think it's a government decision as to what the strategic value is of that seaweed biomass to the economy of the country. And I could just point people to the Mariner program of the Department of Environment in the US, where the growth of seaweeds, various reds and browns, are now considered to be a strategic objective of the United States of America. As such, the Departments of Energy have determined that they can grow seaweed at any cost. Now, we've been through these boom and bust cycles before, because in the 1970s, the Americans decided they could grow seaweed at any cost. And then after about 5 years, it petered out. Will this Mariner program, Department of Energy program, currently peter out? I don't know, but policy, the government determines priorities, and the policy will follow their priority. Okay, I'm done.
She wants to say or ask something. So. Yeah.
Okay. Yeah. I think I thank you for bringing up the Mariner program, because it really depends upon what the Can you, is this better? Can you hear?
Yes, now. Yeah, thank you.
So I think it's very clear that it should be the strategic objective of the country. And in this case, in the case of India, I believe that we are actually caught in a very strategic pincer, because we're the third largest energy consumer in the world. And we don't, and also the largest population in the world, which means we don't have sufficient residual biomass to be able to make enough sustainable fuels. It is actually a very strategic imperative for us that we create a whole lot of extra biomass. But that is very different. The approach there is very different from the kind of approaches we've seen as food, where seaweed is fundamentally a food. Here we are talking of really large scale biomass, very cheap biomass. But you know what? We don't have all of the downstream technologies to actually convert that completely to usable fuel, because it is so different from land-based biomass. So and this is a very good segue, I think, into what I think is a very important question. And maybe I should ask Are to actually call on to the panel people. What would it take to discover all of the enzymes, the microbes, and whatever convert the seaweed into fuel? I think we're running out of battery on this mic, so that's what. So Are, would you take over and actually do the panel discussion on that? And invite?
Yes. Thank you, Sri. I'd like to invite the panelist, Professor Fulka Ziba. He's available online. Dr. Shams Yazdani and Dr. Catherine Manohar.
So should we answer the question, or?
I think I'm just seeing if they can, if they would, if they are ready there. So. Okay. All right. Let's get going. I think we can start with the question that Sri raised. maybe Volker, you can go first on how can do we know that we have the full complement of polysaccharide degrading enzymes, and what is the commercial applications? Like one of the persons also asked earlier when you gave your talk as well, how can we go hunting for these to get a more comprehensive library of enzymes for polysaccharide degradation?
So I believe that most of the polysaccharide to ethanol to fuel processes and analysis are certainly for the land-based plants, and there are many enzymes known that you need to create hemicellulose and cellulose, and even there new enzymes are found because of the complexity. Now with the marine polysaccharides or with the marine sources, certainly other enzymes are necessary, and if you want to go for full conversion, that means that 95% or above of all the carbohydrates are at the end utilized for the ethanol, that you would still need to get some of the enzymes. I mean, all the cellulose you can degrade. It has been shown many times, and you can degrade most of the carrageenans. But even carrageenan is modified, is carrying methyl groups, and you might have other smaller amounts of other polysaccharides. And the smaller the amounts, the more chance that another enzyme is still out there and missing to convert it. And so you have to find the right balance of not putting too much effort in it, but still having a high conversion. What I do not know is all the salt rate that you bring in. Many of the enzymes that you typically use for the degradation of cellulose hemicellulose might not work with the seaweeds, as well as you have different environments, different specifically salts in there.
Yeah.
And here you might also have to engineer or look for enzymes that are active in these high salt environments. All is possible, but I have not. The focus of my work, I mean, we do degrade seaweeds, but we haven't gone for the full conversion of a whole seaweed into ethanol.
Yeah,
it's possible, but I think there has to be some work to be done.
Would it be possible to produce these enzymes in really large quantities?
Well, you might want to look for an fungus in marine fungus that would produce all of the enzymes by itself. You will probably find some terrestrial fungi that can work well with a lot of the stuff that whatever the seaweed brings along, but you might have to add certain enzymes that the normal fungi that you now use for the pre-production of enzymes are not producing themselves. The alternative, which I would actually, which is a more fashionable, more modern approach, is that you design a strain that is degrading your seaweeds, degrading the polysaccharides, growing on this, and then by that producing ethanol with the consolidated bioprocessing. And also for the ethanol production, you need different strains. Typical engineered strains for ethanol from land plants now convert cellulose, that's easy, but have been engineered to convert also the hemicellulose, which you do not have. So it might be that you can actually work with the commercial enzyme mix that produced by typical fungi like Trichoderma, and then feed the not completely hydrolyzed, not completely degraded stuff to an engineered strain that is then capable of doing the ethanol. That's probably what I would pursue.
So it was very interesting, Volker, that you said it would be good to look for a marine fungus. Now, I don't know how many of those exist, but perhaps I can ask Catherine, who's actually done a lot of screening, to give her opinion on this matter. By the way, as an introduction, Dr. Catherine Manohar is from the National Institute of Oceanography, Goa. So,
Thank you very much. Hello, Dr. Walker. So, yes, we are working on both marine and bacterial enzymes for breakdown of seaweed polysaccharides. So, largely we screen for alginate and agarase and carrageenase. But we also do for ulvin and porphyrin. So we have some enzymes from bacteria, like I just presented, and from fungal samples also. But we have two problems when we do it. We try to actually use the fermentation of the enzymes directly on seaweed biomass, on sargassum gracilaria and Kappa ficus. And what we saw was when we use the cultures directly, fungal isolates, they were also consuming the sugars that they were producing. So we were not able to quantify. This is one thing that we wanted to solve. If you have any suggestion on this, then it will be helpful.
So if you grow your strains on your seaweeds, then yeah, the strains would grow on the sugar, so you will not be able to quantify it. But wouldn't you be able to isolate the supernatant of the seaweeds and use it, because this supernatant should contain the relevant enzymes, and use that on a seaweed and then quantify it?
Yes, okay, that is okay. The cell-free supernatant we collect from the extracellular enzymes of fungal, and then we use it for treatment. Okay, we will try that. Then I think we will be able to quantify. And I also you think there is a bottleneck, like in case we want to do a transformation to E. coli. Suppose there are two fungal strains which are producing enzymes, and we want to bring both of this into the same E. coli. You think it will be possible because this is a eukaryotic and a prokaryotic system? So E. coli transformation is possible?
Very hard to say. So many enzymes that are coming from eukaryotes are produced very efficiently in E. coli, and others you struggle for years and you will not get that. We've just done some work for an almost impossible to produce enzyme. That's a plant enzyme that generations have not been able to produce in E. coli, but we got E. coli to do it with doing the right enzyme engineering on it. So usually it's possible, but on the other hand, if you want to have a cheap enzyme, then usually having a secretion system, so having a bacillus or having a fungus for the production is better. Just for studying it now, that certainly E. coli is fine.
So on this note, actually, Shams, would you have something to add about expressing, especially in relation to Catherine's question?
Yes. Okay, so I'll answer that first and maybe then I'll comment on other parts. So, of course, expressing fungal enzymes in E. coli is extremely difficult. We made an attempt. We tried to express 20 of those enzymes, which we thought is much easier. And forget about those complex cellulases. We were trying to express some swollenins and Ig, which were trying to loosen the biomass, but that also we couldn't really express in E. coli. So it's extremely difficult to express the fungal enzymes in E. coli. Some success you can get from the Pichia. So we were able to express some of the beta glucosidases in Pichia because it's relatively easier to express. If we had many models that we made and we expressed them in Pichia. So if the mic is on me, so I will just add on the enzyme part. See, based on the holistic, screening system that we followed on the lignocellulosic biomass hydrolysis, what we really realized that and I'm assuming it's probably the same is true for the seaweeds. There's not one or two enzymes work that will lead to a final metabolizable sugar. It would be probably a complex and many of those enzymes together that are needed. So when you are trying to screen for microbes that are going to digest the biomass, you should have a system that will detect your final monomeric sugar. Okay, and you assay for that. You should have really a good high throughput screening system that will detect the monomeric sugar that is coming out. So I mean, based on some of the previous lecture, I heard that it's possible that these macroalgae can get completely hydrolyzed and disappear in the liquid. So that means certainly there are enzymes and there are microbes that produce those kind of enzymes. But we need a very good assay system to detect those. So there are two ways you can solve the problem. One is that you discovery of enzyme and production of enzyme. So you have to think about these in a different term. If you are happy with 3 or 4 or 5 enzymes, whether it is a fungal or bacterial, if those are enough to convert the polymer into a completely metabolizable monomer, and when I say metabolizable monomer, I'm assuming they are desulfated. They are good enough to be transported inside the microbe, which is going to be fermenting it to ethanol. Now, you need to actually, if there are 4 or 5 enzymes, you can, if they are bacterial based, I will just look at some of the good bacterial hosts like maybe bacillus or something to express and secrete. If at all it is possible, fungal is the better host to make larger quantity of them. But if there are more than 4 or 5, if there are 20 of those that are really needed in a high quantity, I would rather go to that particular host itself and try to see if I can manipulate that host to produce even more and more of that. And if that host is fungus, is nothing like it. So there are fungus naturally has ability. For example, Dr. Catherine is saying that if there is a fungus that can, degrade and consume the sugar, that means it certainly is producing enzymes that are able to metabolize the sugar, convert into monomer, and which is transported inside and it is consumed. So I think these are the microbes that need to be really, really explored in a deeper sense. It looks like it is producing a series of different enzymes. And that's not an issue, how do you characterize the enzymes? That is not very difficult thing to do. Most difficult thing is to first have a good assay. Yeah. So uh, to get to that microbes. So I mean, basically.
Okay, so it's a very good assay system. RA, Dr. Folker, do you have any other comments? This has been a very interesting discussion. We should certainly follow this up, and maybe Dr. Folker, if you'd like, we'd like to follow it up more with you in discussion with the ILS, and maybe even join the kind of consortium that you are talking about. So if you have any closing comments, otherwise we can close this panel discussion.
So I'd be happy to cooperate here, join in the consortium. I have been working many years ago at Clariant, that time Südchemie, to build up the wheat straw to ethanol production. I was a global manager for that. So I can certainly agree what was just said last. You do want to work with a wild strain that will produce many, because you will need many dozens of enzymes. And then engineer this strain that is already producing these many. So that should be probably a fungus. And then engineer a second organism for the ethanol production, where there's not so much necessary. But yeah, how to balance, that's certainly some work to do.
Yeah, as a quick statement from my side, from Institute of Life Science as well as the BRIC family. So Institute of Life Science belongs to a bigger consortia called BRIC, where Sam comes from ICGB, I'm from ILS Bhubaneswar and others. So we are now getting into the seaweed biology. We'll try to understand the basic biochemistry and the engineering side. At the same time, I think the part that you are talking, Volker, about being a bigger family, we'll exchange ideas and understand. Would like you to visit India sometime, and maybe some of us can go to other places and understand. And very soon we will create this awareness in the country and particularly in our state, how to maximize the biomass generation and use it profitably in such a way that the farmers are not at loss, at the ecologists and environmentalists are not shouting at us that you are polluting the ocean or something. So these are some of the guiding principles we'll have, and responsibly we will get into this seaweed biology for the interest of the humanity. Thank you.
Now we have one more talk, right? So yeah, the closing talk. So
what is that ILS is planning to do?
yeah, so this is based upon all our discussion. What is the ILS planning to do? Let's hear what the roadmap is.
So, good afternoon, everyone, and thank you for your patience. I will take a very short time. After this, like 2 days of excellent and highly enlightening talks from expert researchers and entrepreneuring producers of seaweeds, we have a comprehensive outline of both the problems and the prospects of seaweed production, utilization, and various application. In light of that, it is now time to describe and refine the approaches ILS has planned to dive deep into seaweed research. At the onset, I would like to acknowledge the guidance and help of the C6 team, particularly Professor Sri Kumar and Dr. R.A. Narayanan is sitting there, in formulating the primary objectives that ILS has envisioned towards research and development of a sustainable system for seaweed application. So with that, I think, yes. Is my slides are visible for the online viewers? Hello?
Yes, yes.
Yes, yes,
Yes, they are.
everything's fine. Please go ahead.
Okay. So this slide, I guess, is already redundant and well known to us, the types of seaweeds. The only thing that I'd like to point out here, that there are like at present, currently, 11 seaweeds have been reported from Chilica, and none of them have been sequenced or studied in depth for their content composition and content and to their functional analysis. And further, I think we have already discussed on this the global seaweed market, that it comprised of $17 billion in currently in 2023, and of which Asia Pacific actually contributes about $13.21 billion. However, India's share in this is less than 1%. So why is this huge market? Because seaweed has its application in almost a lot of sectors, almost multiple sectors of economic and ecological importance, including food industry, pharmaceuticals, biofuels, agriculture, and has additional environmental benefits like carbon sequestration and reducing ocean acidification. So looking at all of this, and given the coastline, 600 kilometer of coastline advantage that Orissa gives ILS, under the able leadership of our director, Dr. Debashish Das, we have roughly summarized a BRIC-ILS team, which is comprised of Dr. Sanjeev Sahu, who has already worked with seaweed extracts and he has presented his findings. Then we have microbiologists like Dr. Tushar Beuria and myself. Then we have genomics specialists like Dr. Sunil Raghav, Dr. Puneet Prasad, and Dr. Seema Pradhan. We have virologist Dr. Gulam Said. We have a bioinformatician, Dr. Anshuman Dikshit. We have plant virologist as well as a proteomics specialist in Dr. Namisha Sharma. We have also plant engineering, genetic engineering in Dr. Shubhobroto Majumdar. And then we have the able company of Dr. Nivedita Jena, who is the COO of the ILS Bioincubator, providing the entrepreneurship mentoring. So looking at the steam and the strength of ILS, the primary objectives that ILS has thought of to start with the seaweed research, stepping into it and getting into a mature relationship with seaweed biology. I will just read through these. So the first one is like basic biology of seaweed. We have understood that already, that to understand the life cycle and understand the strain specialty specification, we need to get the seaweed genome sequenced. After that comes characterization of seaweed raw material, where we need to understand the composition of the seaweed. I think many people have already signified the importance of understanding the raw material of seaweed as a starting source for certain enzymatic activity. Then we have assessment of pathogenic microbes of seaweeds, where identification of the seaweed pathogens comprise the important aspect. We have bioprocessing of seaweed biomass. I think many of the lectures have really dwelt upon that regarding chemical as well as biological processing of seaweed biomass. And followed by that, naturally, comes the objective of microbial conversion of seaweed sugars that are obtained after the bioprocessing of seaweed biomass. So with these, just to give a kind of an overview of the basic methodologies. So for basic biology of seaweed, isolation of genomic DNA is targeted, followed by both sequencing for long reads as well as sequencing for short reads. And then there will be genome assembly followed by genome annotation. And this will serve the purpose of a DNA barcoding for a seaweed for their identification, as well as elaborating the molecular markers of strains and their different stress tolerance. And this will help in future or to get a molecular breeding for enhanced seaweed strains. Followed by this is the characterization of seaweed raw material. As I already said, that it's an important aspect for bioprocessing. So for characterization of seaweed raw material, seaweed biomass will be extracted and macromolecules will be isolated from there, and they'll be subjected to metabolic characterization. Along with this, on the parallelly, there will be gene annotation from the genomic analysis, from the transcriptomic analysis, and the pathway for the synthesis of those specific macromolecules will be elaborated, and they will be optimized for industrial application in a later stage. Third objective about assessment of pathogenic microbes of seaweeds. So for that, microbial isolation and identification, we have already heard from specialists about this. And then followed by metagenomic analysis, microscopy, and other assays that will show us the impact of these pathogens on seaweed biomass, their mortality, their life cycle, and their bloom dynamics. Identification of the as a next step, there will be identification of candidate genes responsible for seaweed pathogen interaction that would probably help through transcriptomics and metabolomics, that would help us actually to understand the resistant varieties and resistant genes, and then cloning and expression of those genes for characterization and further application in the improvement of strains. Then we have aimed for bioprocessing of seaweed raw material, where sample collection and from the macro environment of the seaweed will be done, followed by bacterial isolation, and then identification of these. So, सुनाई नहीं दिया. So identification of the bacteria through sequencing, and then cloning and expression of the identified useful enzymes from there. The next step will be optimization of expression and activity of enzyme through cloning and purification of the enzyme, and then validation of its activity, as well as optimization of the reaction condition in vitro and for further industrial application. And the last objective is microbial bioprocessing for converting seaweed sugars into value-added products. It's more or less something in a similar way. It will be about locating those microbes that will provide enzymes for their breakdown and taking these sugars and then converting into useful products, industrially useful products, like, for example, as a bioethanol production. So it can, as we can have enzymes probably from bacterial species, microbes that can be utilized to enhance the yield as well as quality of the downstream product. So with that, I thank you for your patience. I've come to the end of the slides, and instead of saying that the floor is now open for questions, I will say the floor is now open for your inputs, suggestions, advice, and guidance. Thank you. Yes.
So that's all. Are, I think you heard us, Alan, all the participants. Anybody has any suggestions? Now give us. Otherwise, you can write back through email. This is what. Yes, Alan.
Sir, I wanted to say that I think there is a lot of things that underpin some of the more advanced, what you I would say was advanced, you said were basic biology, but I think there are issues about sampling technique and sample preparation so that you have representation and replication, because I've seen a number of studies where a person has taken one very small grab sample and then tried to take the analytics of that and almost apply those data to the entire genus. And I think you need to understand that is a lot of seasonality as well as geographic variation, but in the end, your data are only as good as the sampling technique that you've used. And I think we're not being as rigorous, we collectively, hands up, including myself, but we're not being as rigorous with sampling procedure as we should be at times. And I think that part of that, don't forget that as part of your getting the material in which to do the more advanced studies on.
Thanks, Alan. I think the point is noted, and we will do take care of the sampling strategies and whatever resources in terms of manpower, money, whatever you have, can be utilized in a proper way if we do the proper sampling strategy. Point taken.
Exactly.
I think Mahasweta has a...
Yes. Aha, you can go ahead, please.
Yeah, so Mike, you mentioned about isolating pathogens from seaweeds and then doing some assays. I was just curious about the model species that you want to work on as a starting point.
You're asking me specifically, Maha?
No, it's just
to the speaker.
Yes.
yeah,
So
speaker.
yes, Dr. Sa. So to answer you, I mean, I think RA will also be able to answer it, but it is about Kappa ficus that we are looking forward to, the commercial variant that is being mostly commonly used. It will be provided to us by C6, our C6 team, and we will start from there. I think it's Kappa ficus as well as Gracilaria that we will be getting from them.
Basically,
yeah, so,
not yet frozen, but idea is still culminating some industrially important strengths as well as some strategically local variants whose importance we do not know, but they are definitely locally prevalent. A mixture of that will be taken up.
Okay, thank you. Just a follow-up question. Forgive my ignorance. Do you have Gracilaria in the Orissa coast?
Yes.
Yes, it is there.
It's
There is
there.
there is a
It's
presence
present
of
there.
Gracilaria.
Yes.
Okay. Okay. Thank you.
Volker? Dr. Volker?
Yes, so thanks for this overview. It's a very nice program. You mentioned ethanol as a big product, but certainly the seaweeds in the picking up will have many other potentially interesting compounds. So that even the proteins, would you make ethanol out of these, or would you rather go for a biorefinery approach where you then have different product streams leaving the system?
I think we are open ethanol because people around us are influencing that ethanol is the way. Probably the people know the road to go to ethanol, but definitely we are open as we go along.
Other products.
If something interesting, important, easily available, economically viable products come, then definitely we'll be taking those routes as well.
Okay, that's good.
Thank you, Professor. Looks like there are no more questions from online platform.
Okay.
Offline also there.
So if there are no more questions, I will request our director to please come up, say the vote of thanks, and conclude the meeting. Sir.
Okay. So I think this is a concluding session. Online participant can take a call whether to continue or not. I think we have to give some mementos to the speakers who have come here in person. Thank you all.
So thank you everyone for being here, and I would like to request Director Sir to give away the memento to Catherine ma'am for the wonderful talk and bringing us into the microbe world of the seaweeds. Dr. Debobrata Bishwas for giving the vision of ILS. Dr. Seema Pradhan for co-chairing the session. वो repeat it.
And to Dr. Namisha Sharma also for coordinating the entire event.
I would like to thank you all, everyone who contributed to this meeting on a short notice. Speakers, especially organizers, in the team that is playing behind the IT team and other food and other organizers. Heartfelt thanks to all of you. I think we should continue to meet in certain frequency because this topic is new and a lot to learn. And with all those notes, thank you everybody. Bye bye. There is a high tea. Everybody, please, you are welcome to join there.
More time for discussions.